Improvement. A warm welcome to all on this beautiful, auspicious day. Myself, Dr. Monica Joshi, professor in the discipline of Seed Science and Technology and convener for 2 days professor presentation, along with my co-convener, Dr. Shruti Sethi, principal scientist, Division of Food Science and Post-Harvest Technology, heartily welcome all the esteemed dignitaries, faculties and students to the 3rd day of the Convocation Week program. After witnessing the power-packed performances and presentations made by the brilliant students of our institute for the past 2 days, now it's time to listen to the professors of the 26 disciplines who, through their respective faculties, have mentored the students by providing academic, professional, and personal motivation to the path of success. The chairperson for today's session on crop improvement, Dr. Raman Meenakshi Sundaram, Director, ICAR, Indian Institute of Rice Research, Hyderabad, is amongst us today. It's an honor for us to have you here, sir. May I kindly request, sir, for gracing the dais along with our respected Dean Ma'am, Dr. Anupama Singh. And I also invite the co-convener, Dr. Shruti Sethi, to kindly join on the stage. Thank you, sir. A big round of applause for all the dignitaries seated on the dais. May I now request respected Dean Ma'am to kindly welcome the chairperson. Thank you, ma'am. And now may I request respected Dean Madam for her welcome address and a kind introduction of the chairman for today's session to the August gathering.
Thank you. A very warm good morning to one and all, on the dais and off the dais, and those who have joined online. I have been assigned the twin duty of doing the welcome address and introduce the honorable chair of this session to the audience. Since our director is due to some very urgent engagement, he is out of IRI and will be reaching by 11 a.m. and will join the further proceedings of the day and the convocation week. Thank you very much, Dr. Monica. So since, needless to say though, but it's my pleasure and proud privilege, rather humble privilege, to extend a very warm welcome to all of you on this 3rd day of this Convocation Week program, which is of course, as I said, that it is the academic utsav of 6 days, wherein the academic brilliance is at its peak, that display is at its peak, and today, after listening to yesterday the scintillating performances to the extent that Dr. B.M. Prasanna, who was the chair of the jury yesterday, he made a special comment that, you know, the faculty should sit here to see actually how the presentations and the display of the communication has to be there. And that was the display of that acumen by the students was just brilliant, and that is well reflected in the results that have been announced yesterday. So today we have presentation of the significant educational achievements in continuation of the yesterday's program by the professors of different disciplines. There are 26 disciplines, Honorable Chair, and we have in the forenoon session today the first session from the crop improvement disciplines, which have the genetics and plant breeding, plant genetic resources, seed science and technology. I have the honor on my own behalf and on behalf of the director to welcome Dr. R.M. Sundaramji, Director ICAR, IIRR, Hyderabad, and also now an integral part of the Hyderabad hub of IRA Graduate School through Hyderabad hub, who is chairing this session. So in this session of crop improvement, the presentation would be based upon a total of 54 theses comprising 34 MSc and 20 PhD. So therefore, sir, it does not represent the entire spectrum of the research carried out by School of Crop Improvement, but actually gives a window shot of through students-driven research about what is being done in IRI in the form of the cutting edge and the translational science. So now, on behalf of the director, it's time for me to introduce the speaker to the audience. Dr. R.M. Sundaram, Director, IIR, Hyderabad. He completed his BSc Agriculture from TNAU, Coimbatore, MSc Agriculture in Molecular Biology and Biotechnology from GB Pant University of Agriculture and Technology, Pantnagar, and a DBT Research Fellow, and PhD in Plant Sciences from University of Hyderabad as a CSIR JRF. He joined the Indian Institute of Rice Research, Hyderabad, as a scientist in the biotechnology section in 1998, and his studies focused on biotic stress resistance, hybrid rice applications of biotechnology, and product development through transgenic breeding. He was appointed as director of the institute in April 2021. As director of the institute, Dr. Sundaram is serving as the coordinator of All India Coordinated Research Project on Rice and also as coordinator of ICAR Consortia Research Platform on Crop Biofortification. His work is well known for his in trait characterization, molecular studies, and breeding in rice, which has culminated in identification of novel genes, development of several products, high-impact publications, and national awards and laurels. The management skills of Dr. Sundaram, as a matter of fact, this is something which I would say out of the script which has been provided to me. That became so evident, and I became an ardent admirer of him since that day. 2 years ago, during director conference, when DG Sahab was announcing the names of the institutes which do not have any audit para. And he was tempted to call Dr. Sundaram on stage and, give him a big, applause on behalf of the entire audience. So this speaks about, his integrity, his way of handling the affairs, apart from his research skills and acumen that he's holding. Dr. Sundaram also handled and coordinated several externally funded research projects, published his work in many international journals, more than 140, and national journals, more than 30, and has authored more than 40 books and book chapters, technical bulletins, and popular articles related to biotechnology in rice. He is the recipient of several awards and recognitions, including ICAR Lal Bahadur Shastri Outstanding Scientist Award, Indo-US Research Fellowship, Hariom Ashram Trust Award of ICAR, CSIR Innovation Award for Rural Development, P.N. Bahel Award for Excellence in Crop Sciences, DBT Award for Biotechnology Product and Process Development and Commercialization, among many others. Besides this, he is a fellow of National Academy of Agricultural Sciences, National Academy of Sciences, Indian Society of Genetics and Plant Breeding, Associate of NAS, and has received USAID Fellowship, IUSSTF Fellowship, and DST Humboldt Foundation Travel Grant. He served as the editor-in-chief of Journal of Rice Research, academic editor of PLOS One Journal, Journal of Plant Biochemistry and Biotechnology, Indian Journal of Genetics and Plant Breeding, and is also serving as a member of several national and international committees. With this much of credential behind him, I think this session is going to be a very, very interesting and, of course, again, a scintillating one. Thank you so much for giving me this opportunity, Dr. Monica, and I again welcome the chair to this session. Thank you so much.
Thank you so much, ma'am. So as convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentation today. For the session 1, as already briefed by Dean ma'am, we'll have 3 presentations from the professors of the following disciplines in the following order. First will be genetics and plant breeding, to be followed by plant genetic resources and seed science and technology. For all the 3 disciplines, respective time allocation is based on the number of students receiving degree, and professors will highlight the research outcomes of both MSc and PhD students based on the identified theme areas of the respective disciplines. I would also like to inform the chair as well as the House that the respective time allocation has already been communicated to all the professors, and my request to all the presenters to kindly wrap up the presentation in the allocated time. For the ease of the presenter and as an indication of wrapping up the presentation, yellow light will blink 2 minutes prior to the presentation of the complete allocated time, and red light will blink at the end of the time. So with this, with the permission of the chair and Anupama ma'am, can we just move down for ease of convenience? So thank you very much, sir and ma'am. So with this, I will now hand over the session to the respected chairperson for conducting the further proceedings. Thank you, sir.
Good morning to everyone, and thanks to Anupama madam for introducing me. Without much any ado, I would now like to invite Dr. H.K. Dikshit for making a presentation with respect to genetics and plant breeding. Sir, you have got about 25 minutes, sir.
Respected Dr. R.M. Sundaram, Director ICAR, Chairman of this session, and respected Dr. Anupama Madam, Dean and Joint Director Education, conveners of this session, heads of the divisions, professors, faculty members, and students. So I shall be presenting the significant educational achievements in genetics and plant breeding discipline. In genetics and plant breeding discipline, we have 58 faculty members, 46 are in division of genetics, and rest 12 are in hubs and regional centers of IRI. Totally 77 students are on roll, and 20 MSc students will be getting degree in this convocation, and 16 PhD students, total 36. The research work carried out by the students is broadly grouped in 4 major theme areas. First is breeding for higher productivity across all crops. Then second is breeding for resistance to biotic stresses, then tolerance to abiotic stresses, and then finally enhanced nutritional quality. So we start with first theme, breeding for higher productivity. Under this theme, I shall be presenting the work of 6 MSc and 8 PhD students. Gangadhar Kane, he worked on genetic mapping of root system architecture in rice. He has had association mapping panel of 261 genotypes, which were screened for root system architectural traits in hydroponic and pot culture conditions. Genome-wide association studies identified 21 significant MTAs. Novel genes regulating RSA were also identified. 4 marker trait association with root traits were validated in real population, and SSR markers were identified for targeted trait introgression. Harshita mapped genomic regions governing uppermost internode length in rice by utilizing 201 real populations, recombinant ingrid lines at 3 locations. Phenotyping was done, Delhi, Karnal, and Aduthurai. Major QTL hotspot was identified on chromosome 1, having 7 QTLs for different traits, including uppermost internode length. In silico analysis identified 15 candidate genes linked to internode length and panicle exertion traits. 22 QTLs were mapped for uppermost internode length and panicle exertion traits, and out of these, 10 QTLs were validated in F2-derived F3 population. Bhaskar explored genetic and adaptive diversity of Oryza species naturalized in Jharkhand. Total 75 species were collected from 24 districts in Jharkhand, and based on this phenotypic data, he found greater genetic diversity in Oryza rufipogon as compared to Oryza nivara and Oryza sativa. GBS identified 26,267 high-quality SNPs. Population structure using these SNPs revealed moderate genetic differentiation between Oryza nivara and Oryza rufipogon, while Oryza sativa displayed significant genetic diversity. He suggests the use of Oryza rufipogon in rice breeding programs because of the diversity which he got for different traits. I and they worked on genetic diversity and marker trait association analysis for morphobiological traits in colored rice, which were collected from Chota Nagpur plateau. He evaluated 50 land races, including 33 colored grain land races for total phenol, flavonoids, anthocyanin content, and antioxidant content. Black rice, red rice were evaluated, and it was found that black rice was highest in TPC and TAC, whereas red rice had highest flavonoids, total flavonoid content, and black rice group were rich in phenol, flavonoids, and anthocyanin compared to red and brown group. Marker trait association identified 5 SNPs for total flavonoid content, with 2 on chromosome 5, explaining 57 and 46% of phenotypic variation. Then Shiv Kumar Singh, he worked on genetic analysis and marker trait association study for physiological traits in bread wheat. Based on combining stability of using 8x8 parent diallele, he identified good general combiners, parents, and SCA effect was higher in cross, which he has reported for most physiological traits. Then 260 genotypes were evaluated in 4 environments under genome-wide association studies, and 4 genotypes having high yield and stability across locations and time of sowing were identified. 219 QTLs were identified for agrophysiological traits across the 4 environments. Candidate genes linked to development pathways influencing physiological traits were identified. Rohit worked on morphophysiological and molecular characterization of elite wheat varieties carrying alternative dwarfing genes. Alternative dwarfing gene RST18 and RST14 were introduced in 4 elite wheat varieties, and it was found that mean plant height decreased in improved lines carrying RST18 and RST14 as compared to the traditional RST-B1B lines. 12 lines carrying RST18 and RST18 plus RST-B1B gene and 13 lines carrying RST14 were superior in yield performance in more than one condition. The improved lines with RST18 and 14 genes showed better early establishment and performance under restricted irrigation. Aritava Kole, he worked on identification of genomic regions for kernel row number in maize, and based on F2 population of 354 individual. 3 factors with 6 alleles were identified. They also identified 14 metaQTLs which were distributed on chromosome 1, 3, 5, 9, and 10. Shiv Prasad, he worked on mapping of loci governing extra-earliness and multi-flowering trait in lentil. Extra-earliness in lentil is required for pushing lentil in central zone and eastern India, rice fellows. And he was successful in identifying these 3 QTLs on chromosome 3, having 2 candidate genes, LCELF3A and LCFRI. 2 indels were developed linked to LCFRI and LCELF3A, exhibiting large phenotypic variants, and these indels were validated also in the mapping population. Similarly, for multi-flowering trait, 2 QTLs were identified, and 2 candidate genes were identified, and from 1 candidate gene, indel primers were developed. Anand Maurya, he characterized M2 population of winged bean through agromorphological traits and molecular markers. A total 147 families were evaluated for 18 qualitative and 12 quantitative traits. Then these families were genotyped also. This genotyping revealed the polymorphism of the primers. Structural analysis confirmed the presence of 2 distinct subpopulations from mutation of single variety. Then Pratap Ghosh, he worked on identification of genes for seed coat color in winged bean. For seed coat color related QTLs, were analyzed using flanking marker information. Gene prediction from these QTL regions identified 127 candidate genes for QSDC1.1 and 20 for QSDC3.1. In silico annotation of these genes using NCBI database identified 4 key seed coat color related genes. Upregulation of transcription factor MYB113 and W81 related proteins in purple 13 and purple 1 suggest their potential role in seed coat coloration in winged bean. Then Rahul worked on genome-wide association studies for plant architectural traits. This study basically targeted traits for mechanical harvesting. 280 diverse genotypes were of core collection were phenotyped for 10 plant architectural traits. 97 and 51 MTA identified using trait-based and blob-based multimodal GWAS analysis. 17 consistent MTAs were identified, and 2 genomic regions present on chromosome 5 and 7 were found to harbor MTAs for plant height and first pod height from ground.
Dr. Dikshit, just a minute. May I request all the students to please maintain silence? Thank you.
Then Ninawat Krishna Kumar Rathod, he worked on inheritance studies and mapping of early maturity in soybean. From their germ plant, they identified one extra early line, SKF 148. It was crossed with DS 9717 and PUSA 12-13. 2 mapping populations were developed. And then both mapping populations revealed this polygenic inheritance of days to flowering and maturity. 5 QTLs were identified for days to flowering on chromosome 10, 11, and 19, and 5 QTLs for days to maturity on chromosome 1, 4, and 11. Mohit Sharma, he worked on genetic analysis and mapping of seed weight in Brassica juncea. Diallele of 15 parents was was evaluated, and based on that, gene action was predicted. Predominance of additive gene action was reported for 100,000 seed weight, and for oil content, non-additive gene action was reported. Genome-wide association studies of 152 Brassica juncea genotypes identified 16 stable MTAs for 1,000 seed weight. Gene annotation of these MTAs revealed presence of transcriptor factors and other genes involved in regulation of seed weight. 2 intel primers associated with VJU.CKX2 gene were successfully developed and validated. In second theme, that is breeding for resistance to biotic stresses, Amarkant Kushwaha, he fine-mapped major QTL QBK1.2 imparting tolerance against Bakane disease in rice. For this, F2 nil population was developed of 1105 individuals, which was segregating for this major gene. QTL was fine-mapped to a region of 130 kb using SNP markers. Expression analysis and protein prediction study identified potential candidate gene. Then V. Samant Reddy, he worked on inheritance of mapping and genomic regions for brown spot resistance in rice based on F2 population from cross Bala into NIL7, which was phenotyped, and 9:3:4 ratio was there, which indicated supplementary gene interaction and role of two major genes. Two SNP genotypes identified from the study were located within coding region of genes encoding a chitinase-like protein and pentatricopeptide repeat containing protein on chromosome 4 and 6. Manoranjan Senapati, he worked on molecular mapping of QTL governing sheath blight resistance in rice, genome-wide association study with 261 genotype identified 2 QTLs for lesion height and 1 QTL for relative region length. Totally 6 QTLs were identified by using high-density linkage mapping. 3 major QTLs were identified within 1.29 MB region of long arm of chromosome 1. One SNP associated with QTL QSHB1.2 was validated. Neetu Mohan, she worked on identification of heterotic restorers and improvement of maintainer and restorer lines for yellow rust resistance in wheat. 10 cytoplasmic lines and 4 restorers were crossed and hybrids were evaluated at 2 locations. 2A lines which were promising for grain yield, which were good general combinase for grain yield were identified. Similarly, 2 R lines exhibiting good combining ability were identified. Specific crosses with high SCA effect and yield were identified. In addition to this, they transferred YR10 in B line, B365, and YR15 was transferred in R line, R1752, and superior plants were identified. Malvika worked on identification of genomic loci associated with yellow rust resistance derived from Triticum turgidum and Triticum spelta, and their utilization in wheat improvement aided by speed breeding. Yellow rust resistance in TTD533, which is derivative of Triticum aestivum and Triticum turgidum, was reported to be influenced by recessive genes. Similarly, yellow rust resistance in TSD858, derivative of aestivum into spelta, dominant gene was reported. Then they combined this yellow rust resistance in TTD533 and TSD858 with leaf and stem rust resistance from Agilope spaltites derivative lines. For combining, they use this speed breeding. In the process, the entire life cycle of plant was completed in 77 to 78 days. Then Monica Sahu, she worked on genetic studies on tolerance to fall armyworm in tropical field corn. The study was made using a line into tester crosses. All the testers were tolerant to fall armyworm. And 2 inbred lines and 7 hybrids shows yielding ability and low damage score for foliage and crop. Genetic component of variation revealed that biochemical parameters were less influenced by environment. Protein lines with high protein were preferred by fall armyworm, and tannins and phenols caused non-preference for the insects. 2 combiners were identified, good combiners, and 4 SSR marker associated with tolerance to fall armyworm were reported. Mukesh Shankar, he worked on identification of novel alleles for Fourier brass resistance through genome-wide association studies in pearl millet. This High G-into interactions were observed for this foliar blast. Highly resistant and stable genotypes were identified. Out of 34 MTAs identified, one SNP was found to be stable and associated with Fourier blast resistance in 4 out of 7 locations. 14 candidate genes were identified and Cas marker was developed and validated. Hrudaya Vijay, she worked on diversity analysis in soybean with special reference to mung bean yellow mosaic virus resistance in soybean. 142 advanced breeding lines were screened against MYMV, and 63 were highly resistant, 23 were resistant. This is because these are the advanced breeding lines, so advanced breeding lines should have resistance. And then they also said that they identified diverse lines and recommended them for use in hybridization program. 2 lines exhibited low phytic acid content in comparison to the checks. Ankita Singh worked on understanding the complex network of fusarium wilt resistance in lentil using comparative RNA-seq. RNA-seq data indicated differentially expressed genes between 4 combinations studied between resistant and susceptible parents. Disease-related 36 differentially expressed genes were identified and role of 15 was confirmed in wilt infection by QRT-PCR. Transcription factor, LLR receptors, and other factors regulating hormone response and signaling were reported. Third theme is breeding for resistance to abiotic stress. In this theme, 6 MSc students worked. And Shivani Meena, she worked on genome-wide association mapping for combined drought and heat tolerance in wheat. 150 advanced breeding lines were located at several locations and donors were identified for heat and drought tolerance. Total 109 MTAs were identified for component traits of heat and drought tolerance. 8 under control condition, 26 under drought stress, 19 under heat stress, 50 under compound drought and heat stress. 80 QTLs were identified for grain, iron, and zinc concentration, of which 3 were found pleiotropic for both the traits. Aishwarya, she worked on exploring phosphorus starvation tolerance in AUS rice and deciphering genetic basis of tolerance through genome-wide association studies. 181 AUS rice germplasm exhibited significant variability for 7 agronomic and 5 stress-related traits. Genome-wide association studies identified 71 marker trait associations related to P uptake and yield under stress, including key phosphate transporter genes, OSPT11 and OSPHR2. GM Kirti worked on studies on regulatory and functional response of root plasticity to abiotic stresses in maize. Maize genotypes evaluated showed significant variability for adaptive plasticity for drought, low nitrogen and salinity. The significant genotype stress and stress into genotype interactions revealed that there is chance for developing maize cultivars with stress-specific adaptive plasticity. The genotypes CML 579 SKV 671 showed adaptive plasticity for low nitrogen stress, whereas PML 93 and MG42 and CML 563 under salinity. Hub transcription factors were found to be major regulator of root system architecture under the stresses. Rohit Sarkar worked on genome-wide association analysis for traits influencing terminal heat stress in lentil. 158 lentil genotypes were phenotyped for 11 heat tolerance indices and genotypes exhibiting tolerance were reported. Association mapping analysis identified 28 MTAs. These were found to be linked with 150 protein coding genes and 7 uncharacterized genes. Komula Uday, he worked on estimation of genetic diversity for low phosphorus tolerance in paddy land races collected from Chota Nagpur plateau region. 80 Oryza sativa land races were collected, and they were evaluated under low phosphorus, 8 ppm under field conditions. P deficiency increased the days to 50% flowering across the genotypes when it was compared with the control. Land races exhibited low phosphorus stress tolerance index, and molecular characterization using PUP1 specific 15 markers indicated that PUP1 locus marker K5 and K20-1 were present in high frequency intolerant genotypes. Jyotsna worked on genetic diversity analysis for low phosphorus tolerance in lentil. She collected 150 lentil germplasm lines from eastern and northeastern India, and these were evaluated under low phosphorus conditions. Under low phosphorus, mainly seed dry weight was reduced, and however, under low phosphorus, root length and root to shoot ratio increased. She also identified genotypes which were superior to check and others under for PEU traits. Then under enhanced nutritional quality, we had 3 MSc students and 4 PhD students. Mamta worked on molecular mapping and characterization of neurotransmitter compound, gamma aminobutyric acid, GABA, in germinated brown rice. 192 germplasm lines were evaluated at 2 locations, and their GABA in germplasm range from 7.19 to 75.04 mg per 100 grams. And genome-wide association studies identified 6 MTAs on chromosome 1569, explaining 0.06 to 4.15% variation. In silico analysis from RAP database identified this 10 putative candidate genes. GABA in brown rice was half of the GABA content in germinated brown rice. Pratibha worked on a genome-wide association study for identifying marker trait association for nutritional and glycemic index quality traits. In this study, 116 triticum sphaerococcum and 7 bread wheat varieties were evaluated for 2 years. Variation was found for different quality traits, and promising lines were identified. Positive correlation was found between zinc iron and negative between amylose and amylopectin. 49 MTAs for different nutritional quality traits and glycemic traits were identified. Then Duo worked on analysis of genetic variability and molecular characterization and marker-aided enrichment of methionine in maize. For methionine, predominantly additive gene action is important. Major genes for methionine were characterized and sequenced, and inbred lines having these major genes were developed. In addition to this, hybrids were reconstituted with these genes, and it was also found that peak expression of these genes was at 20 days after pollination. After this methionine content slowly decreases. The introgress gene showed higher total root length and seminal roots. Vinay Rajora, he worked on genetic, physiological, and molecular characterization of LPA1 and LPA2 based low phytate maize genotypes. He developed this double low phytic acid mutant LPA1 by intercrossing these near isogenic lines carrying LPA1 and LPA2. Expression of LPA1 and LPA2 in foliage and seeds was studied at different stages. Double mutant had 40% less phytic acid, and it was found that double and single mutant of this had comparable agronomic performance, grain yield, germination, and seedling vigor in comparison to the wild type. So yield, etc., was not affected. Victor work on genetic variability and molecular analysis of folate accommodation in maize kernel. 45 inbred lines were evaluated at 3 stages for folate derivatives, and for folate we will always see effect of genotype, days after pollination stage, and genotype into days after pollination stage interactions. Expression pattern of 3 candidate genes, GTP, CH1, SAM, and CTM governing folate metabolism was studied in contrasting inbreds, and it was found that GTP CH1 plays critical role in early folate metabolism. GTP's observation indicated that expression of GTP CH1 gets reduced 24 days after pollination, and folate biosynthesis is slowed down. Then Naveen, he worked on identification of novel alleles for grain protein and essential amino acid through genome-wide association studies. Based on their study, they identified donors for high protein and grain yield, and they identified 653 significant marker trait associations and 41 candidate genes with key association on chromosome 1, 3, and 5. Miniari Taku, she introgressed LOX2 allele in vegetable soybean through rapid breeding approaches under controlled environmental condition. She utilized the speed breeding condition. Under the speed breeding condition, plant in a year, soybean can complete 4 to 5 generations of vegetable soybean under controlled conditions, and immature plots were harvested over and dried, and was successfully reduced the crop cycle time. However, no adverse effect of germinability and seed growth was noticed on this pod harvested seeds. Off-flavor-free vegetable soybean genotypes were developed using marker-assisted backcross breeding. In honors and award, 8 students received this best poster or best oral presentation award. In addition to that, Malvika Shal received best MSc thesis award. Mukesh Shankar received Young Scientist award and ICAR Nahib Caste Fellowship. Naveen received ICAR Nahib Caste Fellowship. Regarding faculty awards, Dr. Firoz received A.B. Joshi Memorial Award of ISGP and S.K. Vasal Award for Excellence in Hybrid Crops. Dr. Gyan Prakash Mishra received NAS Fellowship this year, and he also got prestigious membership of ISTA. And Dr. Harita and Dr. Rajkumar, they received NAS Associateship, and others got fellowships in different societies. "24 research papers were published from students' work in Division of Genetics. From above 10, it is 5. I have wrongly written 4. And 9 to 10, 8. And then 5 and 6. 8 to 9 is 5, and 6 from 7 to 8. ये काम नहीं कर रहा। These are first 7 papers. Then one paper by Shiva Prasad KM, he was student of Dr. Gyan Prakash Mishra, was published in Plant Biotechnology Journal. NAS score for this journal is 19.5. These are students' publication. And student placement, 12 students were placed, 4 joined ARS, Rahul joined Indian Administrative Services, and others also joined as assistant professor, lecturer, Silk Board also, and so on. Genetics division conducted 2 training programs last year. First was one on genetics and genomic approaches for improvement of stress resilience and nutritional quality in crops, and second on ameliorating quality and productivity of oil seeds and pulses through classical, modern and disruptive technologies. I express my sincere thanks to Director IRI, Joint Director Education and Dean, Joint Director Research, Joint Director Extension, Head Division of Genetics, faculty members and students, and Dr. Vignesh, Senior Scientist. Thanks.
Thank you very much, Dr. Dikshit. You took a little bit more time. It's all right. I now request Dr. Sunil Archit to make the presentation on plant genetic resources. Sir, I hope you'll stick with the time. Okay.
सभी गुणीजनों को नमस्कार. Good morning, all of you. I'm very pleased to make presentation of an account of significant educational research achievements of the students of plant genetic resources. In fact, just to quickly inform the audience here, this is a recent discipline started in IRI with signing an MOU with ICAR NBPGR. So this particular discipline is hosted at NBPGR. And also, it's a matter of pride that IRI is the only place where plant genetic resources is offered an MSc and PhD in India. And which is why we have a faculty of expertise in diverse disciplines. Total faculty members are 41, out of which 6 members are from our own discipline. So the people whom we mentored are right now faculty members. And 27 research guides, out of which 19 are PhD guides. We have 35 students at this point of time, MSc 6 and PhD 29. We also follow BSMA courses, and generally students take genetics as minor course, and in PhD genetics or seed center technology. The the PG students carry out research in these 4 major themes. Ecogeography and systematics, genetic diversity analysis, genomics of PGR, and germplasm conservation. Although some theses can fall, some of the elements of some theses can fall under multiple themes. In this convocation, 7 students have become eligible to receive degrees, so of whom I'll be making a presentation. And their research is mainly in two theses are in area of cryoconservation, and 2 to 3 are on diversity analysis, and 1 on genomics of PGR. And then just a point of note is wherever students are carrying out diversity analysis in morphological characters, we now insist to include nutritional profiling as well, which is very important. I'll start with MSc research, and then at the outset I just apologize to all the guides and students that I'll be very, very brief, so which may not touch all the aspects that you have carried out research in your... Okay. The first student is Rosemary. Under the guidance of Mukesh Rana, she carried out analysis of pea cultivars with a focus on building an identification process, marker-based identification process. She used 66 germplasm lines consisting of 19 released varieties, 8 exotic varieties, 4 elite lines, and 35 germplasm accessions with 15 shortlisted SSR primers. And then she found out that similarity percentage is 43%, among varieties 33%, and among germplasm 41%. She could distinguish all the varieties. And based on her profiles, this is some of the insights in clustering analysis, where the heirloom cultivars from different countries have been clustered together, the variety released are clustered together, kind of things. So this is an excellent outcome of the research. Rahul Jagirdar, under the guidance of Kuldeep Tripathi, worked on wild species of Vigna, which is very, very important to start any kind of pre-breeding program or usage of wild species. To understand, to characterize them is a prerequisite. So we tried to characterize 13 wild Vigna species based on agromorphological traits as well as biochemical constituents. So he combined 49 agromorphological traits based on what we have already information in cultivated Vigna species in NBPGR, from IITA, from biodiversity, etc., and then carried out some of these analyses at CPCT IR as well as crop wild relative garden in NBPGR. So this is a quick view of kind of diversity you have across species. So significant outcome includes an understanding of the biochemical profile, such as, for example, Vigna oblongifolia has high protein content. In fact, all these are qualitatively individual accessions have been analyzed. I'm just giving a highlight. Unguiculata subspecies Stenophila and oblongifolia are rich in zinc and iron together. Vigna subterranea having a content of low phenols, whereas the high phenol in Vigna vexillata. Similarly, for multiple traits, superior accessions have been identified by the student. And one very important from the conservation point of view is the correction of taxonomic identity. Rehan Nadaf carried out his research under the guidance of Dr. Sangeeta Bansal on cryopreservation of Rubia cordifolia, mangista. The objective was to optimize cryopreservation protocol using encapsulation dehydration technique in the species. So he carried out all the particular standard protocols, and then his research outcome is that he has identified best regeneration medium, best explant stage and size, and the exact protocol of the cryoconservation. Some of the basic studies have also been done. Bhagirath carried out his research under the guidance of Amit Kumar Singh in genome-wide association in emmer wheat. We have a lot of information in bread wheat, but little scarce information in Triticum turgidum dicoccum, this tetraploid wheat. And then he has carried out evaluation of these the germplasm, 166 genotypes against pathotypes 11, 40A, and 43 in a glass house. And by applying, using the pre-existing genotypic data, applying 6 models for marker trait association, he could end up with identifying QTNs, 4 QTNs, 2 different stem rust pathotypes, 11 and then 43, at a lot score more than 3. Okay, so this is one, and also he has done functional annotation. Suhail Ansari carried out his research under the guidance of Manjusha Verma, Dr. Manjusha Verma, on deciphering nutritional and molecular diversity in Lupha acutangula. So he used 96 accessions and then carried out the work in CPCT, and then profiled for particularly these. So in PhD research, Shivam Kumar carried out his work under Sandhya Gupta. In fact, he was inherited from, he's a senior student from late Dr. Rekha Chaudhary, whom we remember fondly, and then to Dr. Sandhya Gupta. He worked on, again, on amenability to cryoconservation in Artocarpus. He used 2 species, Heterophilus, where the seed biology is known, and a new species, Lacucha. So ultimately, he could get a moderate success in establishing in vitro culture and hardening plants. Anto James worked under the guidance of Dr. Mukesh Rana on a very interesting topic of identifying molecular genetic integrity in conserved genetic resources. So on the left panel, you can see that he used 6 Gossypium arboreum accessions, which were conserved, and then they were regenerated twice. So that means the original one 2000, 2007, and 2015, and then 1,044 individual seeds, and then pooled DNA, they were all analyzed. And then I'll just present in the next slide what he got. Similarly, on the other hand, he also carried out genetic diversity in 135 released varieties to understand the temporal and spatial changes in the general diversity. So you can see the allelic trends in the genetic integrity analysis, where he could see that the number of alleles, or the Shannon index, or effective alleles, or expected heterozygosity are all, the changes are minor in general overall. However, there are in a couple of cases here, for example, loss of bands were visible. And not much, let's say, significant differences were found across different zones of cotton cultivars released in cultivation, as well as pre-2000 to 2010 or 2010 to 2020. However, minor changes are there. That means we are losing some amount of genetic diversity over a period of time. These are the student publications in 24-25 of these students. And mainly they publish in genetic resources and crop evolution wherever genetic diversity analysis or wild species work is done. Couple of papers are also there in scientific reports where some amount of molecular data is there. And in fact, one scientific report paper is exclusively on ecogeography. So it's a different kind of paper. Student placements are as follows. 5 students were placed in ARS this time. And then 3 are in private industry and 1 guy has joined APEDA. Student awards, Mr. Kunal, who is currently PhD student, he received for his excellence in MSc as KL Mehra Memorial Award. Others have received the best poster, best oral presentation awards, Latif, Bashir, Suhail Ansari, Rehan Nadaf, Vrinki, and Tendral. Honors and awards of the faculty, Dr. Rakesh Singh has received NAS Fellowship in 2024. Dr. Yadav, Eminent Scientist Award in 2024. One I have not included here. Dr. Sandhya Gupta received ISHS Gold Medal in 2024. Dr. Kuldeep Tripathi and Dr. Padmavati Gore, these 2 are PGR students. So they are PGR students, they are core PGR people. So Dr. Kuldeep Tripathi received NAS Young Scientist Award 24 and then Ramanuja Memorial Young Scientist Award as well last year. And Padmavati received R.S. Padoda Young Scientist Award. With this, I also thank the head of the division as well as director of NBPGR, Dr. G.P. Singh, for providing unstinting support, financially or otherwise, and all my faculty members of the PGR for taking a lot of responsibility and in providing me assistance. I also thank, in absentia, the Vice Chancellor as well as Dean for their constant endeavors. They are always after us. And students of the PGR division for their enthusiasm and then cooperation. Thank you. And then graduates to school staff and then PGR professor section staff for all their support. Thank you very much.
Sunil, for making a very crisp but very lucid presentation. Thank you. I now invite Dr. Monica Joshi to make presentation on seed science and technology.
A very good morning to all. So I have requested Dr. Sushma to kindly maintain the time for me. So very briefly, I'll present the significant educational achievements for the year 24-25. So this is a brief of the faculty. Can you switch off the light? We are a total of 23 faculty, well represented across the nation, out of which 12 are eligible for MSc guidance and 9 faculty are eligible as PhD guides. For the students, we have on roll total 48 students, out of which 9 are MSc and 39 on PhD. Apart from that, we have 9 degree recipients for MSc and 2 for PhD for this coming convocation. So this is a brief list of the MSc degree recipients. My congratulations to all the students who are present offline or online. Monica Thakur, Ayushi Yadav, Prajapati Dinesh Bhai, Nikita Rathod, Souvik Chakravarti, Fabina, Riya Biswas, Ashwani and Samrat Raj from Jharkhand. For PhD, we have 2 students, Chaitanya Ji and Narendra Pal. So very briefly, I'll highlight the significant research outcomes for all the students. So this is a brief overview of the various theme areas and the number of MSc and PhD degree recipients due under each respective theme area. So I will take up one by one. With respect to the first theme on the seed production technology, we have only one student from IRI Jharkhand, Mr. Samrat Rej, MSc student, who worked on the effect of seed coating with microbial formulations on seed yield and quality in green gram under the chairpersonship of Dr. Priya Ranjan Kumar, principal scientist. So Mr. Samrat Rej worked on different microbial formulations, and he could very well inference that the seed treatment with microbial formulation resulted in a saving of 25% of the nitrogenous fertilizer. Cyanobacterial consortiums and other Anabenalexena and other things, they resulted in significant improvement in the seed yield, and all these microbial formulations remained viable after 6 months of storage. With respect to the second theme area on the seed quality enhancement, we have one MSc student, Ms. Monica Thakur, who worked on the seed enhancement treatments for improvement of seed quality in onion under the chairpersonship of Dr. Sandeep Lal. She evaluated different onion genotypes and subjected them to specific gravity separator, magnetopriming and osmopriming. And the results revealed that both osmopriming and magnetopriming enhance the seed vigor parameters compared to the unprimed seeds. And alpha amylase and catalase activity was also found to be higher in the primed seed as compared to the control. With respect to the third theme area on seed biology study, we have one student, Ms. Ayushi Yadav. So my congratulations to her for competing in the gold medal, although everyone may not get, but she did very well. So thank you, Ayushi, for the representation. She worked on the prediction of seed vigor in rapeseed and mustard using near-infrared spectroscopy under the chairpersonship of Dr. Sangeeta Yadav. She evaluated 300 genotypes of rapeseed and mustard using different species, and she could very well come out with calibration models which provided robust predictions for the important seed quality parameters like germination percentage, seed vigor index 1 and 2, and the RPD values of more than 2 indicated good quantitative prediction capabilities in the developed model. The other student, also MSc, Mr. Prajapati, he worked on critical moisture levels for anoxia storage in soybean seeds under the chairpersonship of Mr. Manjunath Prasad. So here he could very well conclude his study by saying that hypoxia, that is less than 1% oxygen, could maintain seed viability, seedling vigor, especially for seeds with low moisture content. So this was very important under hypoxia condition, that the seeds should have low moisture content. And also he identified that below 8.5% was critical for retaining the higher viability and vigor. And he identified black seed coat color genotypes which maintained higher viability and vigor during the storage as well. Another MSc student worked on the assessment of the relationship between morphological characteristics, seed germination and dormancy in Triticum aestivum and Triticum durum genotypes under the guidance of Dr. D. Vijay. So she evaluated 253 Triticum aestivum and 102 Triticum durum genotypes. And very clearly, and she could underline the fact that the plant and the seed morphological traits varied significantly between both the species. And for induction of dormancy, 50 micromolar abscisic acid was found to be good for both the species. And also she found that Triticum durum genotypes were more sensitive to abscisic acid as compared to the Triticum aestivum. And she could also identify one strong genotype of Triticum aestivum species. Another student, MSc, Mr. Souvik Chakravarti, he worked on the hyperspectral imaging system for varietal identification and quality assessment of rice seed lots under the guidance of Dr. S.K. Chakravarti. So he evaluated various basmati and non-basmati rice genotypes, and of the various machine learning models which he used, the LDA model achieved the highest prediction accuracy of 82% and 78% with values of 0.81 and 0.76 for the classification of all 16 rice genotypes and 11 basmati rice genotypes. And he could also identify certain good storers and poor storers. And his study also highlighted the fact that the aged seeds had higher spectral reflectance values than that of the fresh seeds. With respect to the fourth theme area on seed longevity studies, we had a PhD student, Mr. Narendra Pal. who worked on the studies on variability for seed quality traits with respect to seed longevity in onion under the guidance of Dr. Sandeep Lal. So here he worked on 30 onion genotypes which were stored under ambient conditions for 1 year, and germination and vigor assessment was done after 3, 6, 9 and 12 months of aging. So here he compared accelerated aging test at 3 and 6 year duration, 3 and 6 days duration, and what he found that 3 and 6 days of accelerated aging test mimicked 6 and 12 months respectively of natural aging. So this was the most important finding which emerged from his study. And he also highlighted the fact that poor storer onion genotypes lost 47% of the seed viability after 12 months of natural aging, as compared to 22% loss of viability in the good storer onion seeds. He also could underline the fact that higher NADPH oxidase activity, higher ROS accumulation, and lipid peroxidation caused decline in the germination and vigor of the onion seeds with aging. Another student, MSc, who worked on the same theme area, she worked on genetic variation for seed longevity trait in diverse soybean genotypes under the guidance of Mr. Manjunath Prasad. Here she used 275 fresh and 212 aged seed soybean germplasm lines which were stored under the ambient condition. And then a Videometer Lab 4 instrument was used to record the spectral absorbance values at different wavelengths. And by using GWAS analysis, she could identify, 52 MTS could be identified. And also she could find significant SNPs for the important seed morphological traits. And for seed longevity traits also, she could identify significant SNPs. With respect to one more MSc student under the similar theme area, Ms. Riya Biswas. So she was an MSc student working on physiological and biochemical basis of seed longevity in contrasting maize inbreds under the guidance of Dr. Vijay Kumar HP, senior scientist. So Ms. Riya evaluated 89 inbreds of maize which she screened for seed longevity, and based on the various germination and vigor parameters, she could very well identify 2 good and 2 poor storers, which were selected for studying further biochemical parameters. And she could underline the fact that in good storers, there was higher antioxidative enzyme activity, especially SOD and catalase, even after 12 months of aging. The last theme area was the seed technological interventions for biotic and abiotic stress management mitigation. So the first student, PhD Chaitanya, worked on studies on seed invigoration treatments to mitigate salinity stress in soybean under my guidance. So she evaluated 73 soybean genotypes and subjected them to salinity stress tolerance at various levels. And 4 priming agents were also evaluated by her. And the selected best dosage of seed invigoration treatment was further grown under hydroponic conditions. And Ms. Chaitanya could find that both black and green seed coat genotypes showed higher salt tolerance up to 9 decicement per meter, and it was due to the fact that the enhanced antioxidant activity and reduced ROS and lipid peroxidation content was there as compared to the yellow and the brown genotypes. Further, for the seed invigoration treatments, that is for the mitigation, she found that 6 hours of jasmonic acid seed priming at 20 micromolar enhanced the seed germination and the salt stress tolerance. With respect to one MSc student who also worked on the mitigation, but this time was the biotic. So Ms. Fabina worked on seed technological interventions and phylogenetic studies for lectin genes to mitigate the bruchid infestation in chickpea under the guidance of Dr. Arun Kumar MB, principal scientist. So she evaluated and compared both Desi and Kabuli genotypes by using different polymer composites. So here she used 24 different polymer composites which were synthesized using natural and synthetic materials, and clearly she could come out with 12 polymer composites which can be used as seed coats and gave full protection against the bruchid secondary infestation. And 82 proteins were also identified across various pulses which confer the host plant resistance. So this was with respect to the students who will be receiving degrees. Apart from that, faculty publications, we have a good number, 48 total publications. out of which 9 are above 10 hours rating, and then 8 to 10 and 7 to 8, 14, and 11 are 6 to 7. So this is a brief glimpse of the publications. Out of 48, 31 are from our division, wherein the faculty is as a first or a corresponding author. So these are a few details of the same. Next. So for the student publications also, we have a good number apart from the faculty publications, wherein 21 student publications are there and 3 are above 10. These are the details of those publications by the students. So next we have the honors, awards and recognitions by our faculty. So Dr. Gyan P. Mishra was awarded the 5th Dr. P. N. Bahal Biennium Award by Division of Genetics and a NAS Recognition Award. So myself, I developed the first product in the division which was commercialized last year, and the copyright is in process. Apart from that, we have Dr. Sudipta Basu and Dr. Vijay Kumar as the best oral presentation in various forums. And Dr. Vishwanath Harvai, he has filed patent for the Speedy Seed Viability Kit, and an MOU has been signed for the same. Not lagging behind is our students, a huge list of all the students, and my compliments and congratulations to all. So last year, Ms. Tohina Ghosh, she bagged the IRA Merit Medal Award. Apart from the best oral presentation, Ms. Khatun best oral, Hina Saktivel, Dinesh Chaudhary, and then for poster we had Anbalagan, Kirbhukaran, Sandeep Ji, Pumani S. Then Young Achiever Award by Dadiradi Shri Nandini, Chaitanya for best oral and most impactful article, Gayatri for the first prize in debate, Saktivel in speech, and Paul Prerna for securing ranks in entrance examination, Miss Ayushi Yadav for best student of the year, Young Researcher Award, Fabina for certification of presentation, and Samrat Raj for the best poster presentation. So this time we had a whole lot of students bagging awards in various competitions. For the student placement also, I'm very happy to share with the House that out of the 6 ARS seats, 5 were backed by our students of our division who got their degrees last year. So they set a very good example for all their juniors. Apart from that, we have our students well placed in various forums as agriculture officers at various places. Our technical and sporting staff also does not lag behind in bringing laurels to the division. We had Mr. Dharampal Singh as the Best Agronomist Award, Mr. Athar, Dr. Ravish Chaudhary, and Mr. Sunil Kumar as the sporting staff of our division. Apart from that, the division also organized 2 international workshop capacity building trainings and 1 national training. A few glimpses of the Hindi Divas, wherein we also invited this time the school students from nearby schools to participate in the Hindi Divas. So these were the Diksha Aramb activities wherein our faculty participated for giving Diksha Aramb to the new PG students of the division. Freshers Day celebrated as a part of the Diksha Aramb. Teachers Day, thank you for all the students for arranging this for us every time. And the visit of dignitaries, Honorable Agriculture Minister, Director IRI, Dean Ma'am, and then officials from various ministries and other places. And as a part of the course curriculum, our faculty regularly takes the students for visits to various seed industries and production field to give a first-hand practical experience to all our students. And last but not the least, the educational tour to Sikkim, which was organized, and I thank the competent authority for the fund sanction, wherein the students had a very good time and they came back rejuvenated. So with this, I would like to acknowledge the contribution of all our faculty and students by first thanking our director, our Dean Ma'am, all the joint directors of the institute, our head, Dr. Gyan Mishra, all my associate dean colleagues for working day in and day out, all the BOS members of the division, technical staffs, admin, and the students. And my special thanks to all these degree recipients of last year by setting up a very good example, leading from the front and clearing ARS and setting an example and proving the fact that education is the passport to the future, for tomorrow belongs to those who prepare for it today. Thank you. And thank you, Dr. Sushma, for maintaining the time.
Thank you very much, ma'am. You are well within the time, and the presentation was outstanding. Thank you.
May I now request Dr. Sundaram for the chairperson remarks, sir? If we can have it on the dais. Gayatri and others, please arrange the chairs. Dr. Shruti also.
Can you give it from here?
Yeah, sure, sir, sure.
My dear colleagues, we had a scintillating round of three presentations, and all the presentations were outstanding. And I would also like to place on record my congratulations and best wishes for all the PG and PhD students belonging to both genetics and plant breeding, plant genetic resources, seed science and technology, for not only doing an outstanding work, but also winning several laurels, medals, and then achieving glory. I also have a few comments about the presentations. First of all, let me come to the presentations on genetics and plant breeding made by Dr. Dikshit. Of course, it was a very voluminous presentation, and kudos to you. You could manage just well within the time. Of course, there were some interruptions also, so we took into account. And you made presentation based on 4 major thematic areas. One of the things what I was finding is there is some overlap between the thematic areas. Say, for example, there was a presentation related to colored rices, which should be more appropriate if presented under nutritional improvement. That was the only point which I'd like to comment with regard to arrangement of the thematic areas. They are all very well distributed and then very logically distributed also. Coming to the first thematic area, increase in production and productivity, this thing. I would like to mention that while most of the presentations deployed some of the most advanced tools of genomics like SNP markers, I was a bit disappointed to see that in crops like rice, we are still using SSR markers. Of course, we can still use them, but they are to be used in conjunction with SNP markers, wherein we can get more information from the analysis. That is one comment. And as regards to the presentations in different crops, I would like to specifically mention about identification of alternative dwarfing genes in wheat and also showing their efficiency. And it's now time for us to quickly transfer them in the background of elite varieties and also elite breeding lines. And the stage is now set. And we also had a series of presentation on kernel rows in maize, improving plant architecture in different crops, and genomic regions associated with rust resistance and others. And I was also delighted to get to know about the progress related to sheet blight and bacane resistance in rice. And I think now the stage is set for exploitation of all these discoveries and quickly transferring them into elite breeding lines, so that our varieties will be, varieties or hybrids will have durable resistance. And as far as the presentation related to maize is concerned, of course, they are deployed, considering the limitation of time, probably they are deployed SSRs. But possibly in crops like maize, wherein very good SNP markers are available, we should start working on them rather than just depending on a few SSR markers. And other important thing is that several major loci have been identified, and a set of candidate genes underlying those loci have also been identified. It is time that the Division of Genetics and Plant Breeding should get in touch with NAPB and work for cloning and then validation of these candidate genes, because a marker developed from the candidate genes, which is basically targets the functional nucleotide polymorphism, will be a better option for deployment in breeding programs, and it will also doubly validate our claims that this could be the candidate gene. Next year, I would like to see some progress in this particular regard. Just going up to candidate gene level is, of course, important for breeding. Equally important is to go forward and then identify or validate the candidate genes through various approaches. It could be overexpression, gene silencing, or through gene editing. And I saw several presentations on association mapping in genetics and plant breeding, and of course, one presentation on in plant genetic resources also. I'm not really sure whose domain it is, whether it's the domain of plant breeding and genetics or plant genetic resources. What my suggestion will be, both the disciplines should join together and take up such efforts, because the input from specialists or scientists who specialize in plant genetic resources is also important, very important, particularly in terms of selection of the association panel. And one important thing, lacuna, I would not like to mention, but one important missing link in most of the presentations related to association mapping is the population structure. That information you must have taken care, but it was not part of the presentation. And I would also, I'm also delighted to note that significant progress has been made in terms of nutritional improvement in various crops, including rice, maize, and all. And I was also delighted to identify that LPA mutants do not have any agronomic deficiencies. They perform with equivalent efficiency in terms of their agronomic performance. And I'm also happy to note very high quality publications, awards and laurels in the, from genetics and plant breeding. Coming to plant genetic resources, of course, in crops like field pea and all, the number of markers are limited. But I don't think 15 markers will make any sense in terms of analysis. Possibly when these studies are carried out, we probably need to do with more number of markers. That's my humble submission. And when you talk about morphophysiological characterization of IGNA species, you have very well taken care of levels of iron, zinc, and proteins. But one more important nutritional attribute, the presence of anti-nutrients like phytic acid should also be measured if it is not too tough to do it. And with regard to cotton genetic diversity, your team has stuck with SSR markers, and I think in cotton nowadays a lot of SNP marker resources are also available. As much as possible, you have got a very good Casper platform also. You can deploy at least Casper markers, which are a little bit cheaper as compared to high-end SNP markers. That will be the markers of choice rather than just playing around with a few microsatellite markers. And with regard to seed sign technology, equally very effective and a very good presentation. And when you talk about microbial formulations, I would like to draw the attention with regard to an important invention made by Indian Institute of Oilseeds Research. They have developed a chitosan biopolymer-based seed treatment, like say, protocol, which they are ready to give it to anybody. Madam, if possible, kindly explore, talk to Dr. Mathur, Director of Indian Institute of Oilseeds Research, and try to explore the possibility of studying the effect of that chitosan-based biopolymer. And microbial formulations will be effective if they are delivered along with the polymer so that their shelf life can be enhanced. And this aspect also needs, they should not be studied in isolation. Any seed treatment for enhancing the longevity or for enhancing the tolerance should be involve a coating with the biopolymer and coating with microbial formulations also. And hopefully in the next year I'll be hearing such presentations wherein seed treatment with polymers along with microbial formulations are combined. And this is still a gray area in one of the like say topics in rice, that is direct seeded rice. And we are certainly looking forward for very good seed treatment formulations. It could be microbial based along with some polymer coating. And it's an area wherein certainly we require the seeds to have very good vigor because of these treatments, particularly for direct seeded rice. And I'm also very happy to note that the progress with regard to the awards, the best poster presentations, and also other awards backed by the student. And I'm also equally delighted to see that a number of students have secured employment not only through ERS but also even in fields like Indian Administrative Services. Overall, I would rate the presentations as outstanding and congratulate not only the faculty who guided the students, the heads of departments, and also the students for an outstanding work. With best wishes, I would like to conclude my talk. Thank you. Thank you.
Thank you so much, sir, for your valuable comments. And we'll take care of all the insights. All the heads, professors, and faculties are here. We have taken down the points, and we shall take care to incorporate all the suggestions. Thank you so much, sir. So it's now time to felicitate the chair. May I request the respected Dean Ma'am to kindly do the honors. May I also request Dr. Brahmanand to please join on the stage? Thank you to all the dignitaries for joining on the stage. May I now request my co-convener, Dr. Shruti Sethi, to deliver a vote of thanks.
Thank you, Dr. Monica. As the co-convener of this session, it gives me immense pleasure to propose the vote of thanks. At the outset, I would like to thank Dr. R.M. Sundaram, Director IIRR, for chairing the session of School of Crop Improvement. We all know that we have 6 schools in IRI, and the School of Crop Improvement has opened up the 2-day event. So I would like to thank sir to chair the event. And I would also like to thank the director, IRI, and Joint Director, Education and Dean, Dr. Anupama Singh, for helping us plan out this 2-day sessions of professor presentations. Then I would like also to compliment all the 3 professors of the 3 disciplines who have given in brief, they have presented the presentations of all their students, the 54 students who are receiving their degree under the School of Crop Improvement. So thank you all the professors, all the students whose work have been presented today in this session. Thank you everyone. And sir, Dr. Sundaram was very critical to point out at every step related to all the disciplines. What are the positive points? What are the lacunas, sir? We will all look into it, how to overcome all these. So thank you for your critical comments, sir. And also I would like to thank the August gathering today here, the PD, WTC, heads of the divisions, professors, and the students who are present in this hall to attend this session, and all other committee members who have made it a success. Thank you so much. And we will break for tea for maybe 25 minutes, and we'll come back for the next session. Ma'am, it was supposed to be at 10, 11 o'clock, but we are... So we'll come back in 15 minutes, please, because already we are running late. I request everyone to join back.
Good morning to all and welcome back to the second session of the day. So myself, Dr. Monica Joshi, professor in the discipline of Seed Science and Technology and convener for 2 days professor presentation, along with my co-convener, Dr. Shruti Sethi, principal scientist, Division of Food Science and Post-Harvest Technology, heartily welcome all the esteemed dignitaries, faculties and students to the second session of the day. The chairperson for today's second session on crop protection, Dr. H.C. Sharma, former Vice Chancellor, Y.S. Parmar University of Horticulture and Forestry, Nauni, Solan, is amongst us today. It's an honor for us, sir, to have you here. May I kindly request you, sir, for gracing the dais along with our beloved Dean Ma'am, Dr. Anupama Singh. Thank you, sir. A big round of applause, please. May I now request Dean Ma'am to kindly welcome the chairperson. May I now request Dean Ma'am for the welcome address and a kind introduction of the chair to the August gathering.
A very warm post-tea good morning to all of you. And I can see a lot of faces from the Crop Protection School. Welcome to this session. This is the second session of the professor's presentation, and Honorable Chair of this session, Dr. Professor H.C. Sharma ji, a world-renowned crop protection researcher and manager. We are having now presentations by the professors of the disciplines under School of Crop Protection, sir. These include disciplines of agricultural chemicals, entomology, nematology, and plant pathology. And their presentations are based on, it's a huge number, 63 theses comprising 37 MSc and 26 PhDs. But as I said earlier also, it does not represent the entire spectrum of the research, sir, which is being conducted under the aegis of the Crop Protection School. But it's definitely a window of the projections of the research which is going on. Dear friends, as you can see, the chairperson of this particular session is Dr. S.C. Sharma ji, for whom, if I start introducing him in from length to breadth, then it is going to take a lot of time. So I will have to be brief on that. And I thank Dr. Monica Joshi, who is the convener, and Dr. Shruti Sethi, who is the co-convener of this session, for so meticulously arranging every minute detail of this particular session. On behalf of Director, since this assignment has been given to me as a duty to introduce the chair of the session to the worthy audience, Dr. H.C. Sharma ji served as the Vice Chancellor of Dr. YSP University of Horticulture and Forestry, Nauni, Solan, Himachal Pradesh. And he served as a principal scientist at International Crop Research Institute for the Semi-Arid Tropics, Hyderabad, and as a visiting scientist at QDPL, Queensland, Australia, and University of Wisconsin, Madison, USA. He has over 40 years of experience in education and research and has carried out collaborative research with scientists in India, across Europe, Asia, Africa, Australia, and USA. Under his leadership, his university was ranked among the top 100 universities in India by NIRF between 17 to 19, and further the university's ranking by ICAR improved from 51st position in 2017 to 11th in 2019. This deserves a big round of applause as a source of inspiration for all of us. Dr. Sharmaji has authored over 750 publications, out of which 8 books, information bulletins 13, and research papers, a unique number of 302. The killing number, actually, sir, which has been provided to us, so a reference bar. Book chapters 80, and the ResearchGate lists his 355 publications with more than 1 lakh reads and 8,584 citations. Based on a study conducted by Stanford University, USA in 2020 and 21, Dr. Sharma's research contributions figured among the top 2% scientists in the world and top-ranked scientist in entomology in India. His research contributions have been recognized by several academic societies, including bestowing upon him 9 gold medals, Millennium Science Award by CRISAT, Excellence in Science Award by Consultative Group in International Agricultural Research, and Hari Om Trust Award by ICAR. He is a fellow of 11 academic societies, including NAS and Entomological Society of America. Dr. Sharmaji has also served as chair or member of several international organizations, namely President Council of International Congress of Entomology, first Asian selected as president of this 112 years old organization. He has also served as president of the 19th International Plant Protection Congress in 2019, held for the first time in India in over 100 years. Also, he has served as member of Governing Board, International Association of Plant Protection Sciences. As I said, this is just a glimpse of his resume, his persona, and his acumen. So with these few words, I once again, sir, on my own behalf and on behalf of the Academic Council Chair, IRI, I welcome you to this session, sir, and I personally thank you for so promptly agreeing to be, a part of this 2 days program, sir, and guide us for further orienting the student-driven research in the field of crop protection. Thank you very much, sir.
Thank you, ma'am. So as convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentation. So for session 2, as already briefed by Dean ma'am, we'll be having 4 presentations of the following professors in the following order: agricultural chemicals, to be followed by entomology, nematology, and plant pathology. For all the 4 disciplines, respective time allocation is based on the number of students receiving degrees, and the professors will highlight the research outcomes of both MSc and PhD students based on the identified theme area of respective disciplines. For the information of the chair and the House, the time allocation has already been communicated to the professors, and my kind request would be to wrap up the presentation within the given time. For the ease of the presenter as an indication of wrapping up the presentation, yellow light will blink 2 minutes prior to the allocated time and red light at the end of the time. So with this, we will begin the session, and I would request the dignitaries on the dais to kindly take the seat below so that it's easy to view the presentation. And I would now kindly hand over the session to the respected chair for conducting the further proceedings. Thank you, one and all.
So, very good morning to all of you. Now we proceed with the presentations of the significant achievements of the crop protection discipline. And the first presentation is by the agricultural chemicals. Dr. Suman Gupta, and of course, your time is 13 minutes. So over to you. And who will keep time? Madam? Okay.
Very good morning and welcome to the presentation of Division of Agricultural Chemicals. In the Division of Agricultural Chemicals, we are 13 faculty members mentoring 36 MSc and PhD students and working under the mandates of development of agrochemicals, agrochemical formulations, safety evaluation, and to develop human resource of excellence. This year, 8 MSc students, namely Shubhijit, Anirban, Neeraj, Amandeep, Arunima, Koyal, Gitanjali, and Astha Priya will be receiving MSc degrees. And 7 PhD students, Samir, Shirasi, Rajni, Shilani, Garima, Kailashpati, and Rakesh will receive their PhD degrees. Under the first theme areas of development of agrochemicals from natural and synthetic resources, 5 PhD and 5 MSc students have worked. First student, Samir Ranjan, has worked on triazolyl strips bases and amides based on naturally occurring citronellal as potential plant pathogen and nematode antagonist in rice. He has attempted for the very first time microwave-assisted synthesis for preparing triazolyl strips bases, and he observed higher yields in microwave method as compared to conventional method. And out of the total compounds, 26 synthesized compounds were novel molecule and reported for the first time. Synthetic schemes for the preparation of these compounds is given in this slide. These compounds were fully characterized using different spectroscopic techniques like NMR, IR, and mass. Prepared compounds were tested for antifungal, nematicidal, and antibacterial activities, and overall 3 strips bases and 1 amide showed good potential to control the fungal diseases and root knot nematode. Rajni Godara has worked on synthesis of prenalated chalcones and their evaluation against plant pathogenic nematodes and fungi in tomato. He prepared 32 prenalated chalcones, out of which 19 were reported for the first time. She synthesized these chalcones using microwave synthesis, which was found to be quick, high yielding as compared to conventional method. Synthetic scheme is given in this slide. And all the compounds were fully characterized by specific chalcone peaks in the NMR spectra. Fungicidal and nematicidal bioassays were carried out. Compound 5E was found very effective against Sclerotium rholopside, and compound 6B was found very effective as a nematicide. Both antifungal and nematicidal activities were higher in soil drenching method as compared to root dipping method. The compounds were also subjected to dissipation studies in soil, and all these synthesized potent molecules were less persistent and with the half-life of 1 to 2 days as compared to commercial pesticides. Kailashpati has worked on synthesis of isoxazole and pyrazole derivatives and their evaluation against plant pathogenic fungi and barnyard grass in rice. He synthesized 14 isoxazole and pyrazole derivatives and tested against Rhizoctonia solani and Fusarium fusicoride, and also herbicidal activity was tested. The compound 6R was found very effective against as a fungal antifungal agent, and as herbicidal activity 5A compound was found very effective, and herbicidal activity was at par with the commercial herbicide bispyribate sodium, and these compounds show no phytotoxicity to rice seedlings. Rakesh Kumar worked on synthesis of imidazolyl chalcones and pyrazole derivatives and their evaluation against plant pathogenic fungi and nematode in brinjal. His was the first attempt for preparing this compound using ultrasonic method, and higher yield and less reaction time was observed in ultrasonication method. 5 compounds out of 40 compounds were novel and reported for the first time. All the compounds were fully characterized using different spectroscopic techniques. In vitro fungicidal activity revealed that P11 compound was most active against Rhizoctonia solani, and another compound IC4 was effective against Fusarium oxysporum. In vitro nematicidal activity found P20 as the most active compound against M. incognita. The percent disease incidence was minimum in brinjal plant treated with P11 at 250 ppm dose. Koel Mandal, under the guidance of Rajesh Kumar, worked on synthesis of substituted imines and antifungal evaluation against Clostridia fungi. She prepared 27 imines by condensation reaction between benzaldehyde and anilines. Chlorinated compound was found most active against Rhizoctonia solani, whereas brominated compound was highly effective against Fazulina. Geetanjali has worked on synthesis and evaluation of tetrazole derivatives against plant pathogenic fungi. She prepared the compound, 74 compounds were synthesized using 5-step synthetic protocol as depicted in this slide. The compound 6R was found effective with EC50 value of 17 against Rolfsei, and compound 6T was found effective against Oxysporum. Sheila Neal worked for PhD under Dr. Aditi Kundu and worked on chemoprofiling of Gymnema sylvestre leaves for potential antifungal action against storage pathogens. She has extracted the leaves using different solvents, hexane, chloroform, ethyl acetate, and methanol, and found that ethyl acetate extract was the most active. She identified 37 phytochemicals based on UPLC Q-TOF method, and major compounds were gymnemic acid derivatives. Ultrasonic assisted extraction was tried with the yield of 359 mg per 2 gram of the samples, and other parameters for the extractions were also optimized. The most active ethyl acetate extract was formulated using Catosan and the binder, and half-life of gymnemic acid was found to be 4.46 days. Edible coating exhibited highest protection against Pseudomonas expansum decay, and coated fruit showed better quality in terms of skin elasticity, stiffness, flesh penetration, and other parameters. Chemical profiling of Angelica, Archangelica roots for potential nematicidal action against Malidogyne incognita was carried out by Anirban Bare. Again, he isolated essential oil to the tune of 0.9% and also prepared hexane extract. Molecular docking studies were carried out to study the mode of action of these compounds, and in vitro nematicidal efficacy showed that nanoemulsion of hexane extract showed higher efficacy as compared to nanoemulsion of essential oil. Quantification and insect growth regulatory activity of nimbolite from the leaves of Azadirachtica indica was carried out by Neeraj Thakur. He took the leaves and extracted with different solvents like methanol and acetone. The extract was subjected to column chromatography to isolate pure compound nimbolite. 0.09% nimbolite was extracted from methanolic extract. The highest amount of nimbolite was detected in the leaves collected in December, and lowest amount was present in the leaves collected in April. The nimbolite showed insect growth regulatory and antefident activity at 5000 ppm against Frugiperda. Bioprospecting of Dhatura species for the control of Bemisia tabacea was carried out by Asthapriya under the guidance of Dr. Supradeep Saha. Again, he took different solvents of different polarity for extraction of bioactive components. Yield of methanolic extract was maximum, and this extract also showed the maximum insecticidal activity. LC50 was 0.22 ppm, and 3 major alkaloids were isolated, which were subjected to molecular docking studies to understand the mode of action. After 14 days storage study, 97% scopolamine was present, remained intact in acyl formulation. Under theme 2 on agrochemical formulation, one PhD student who also received the Prime Minister Fellowship worked under the guidance of Dean Madam. She worked on development and evaluation of drone compatible formulation for smart pest management in maize. For drone compatible formulations, she took 2 synthetic molecules and few botanicals and prepared 2 different formulations. One is synthetic formulation, one is botanical formulation using the protocol shown in this slide. Both the formulations passed all the CPAC tests with a minimum shelf life of 2 years. Bioefficacy assessment revealed no phytotoxicity on maize plant. This experiment was conducted under phytotron facility. Damage score was calculated by conducting the field trial at 3 locations in Punjab. Both OD and EC formulation performed better at 1x doses, and chemical formulation at 1x dose and biological formulation at 1x dose were found superior in terms of grain yield. Experiment was also conducted in ICAR field in both Kharif and Rabi season. In Kharif season, both OD and EC formulation performed better at 1x dose, whereas in Rabi season, 2x dose was found best. There were no residues in the harvest time maize grain sample. Residues were below quantifiable limits, and safety assessment studies revealed no consumer risk for drone applied formulation at the recommended dose on maize crop. Third theme, safety evaluation of plant protection schedule on agricultural crops. 1 PhD and 3 MSc students have worked. First student, the PhD student, worked on bio-augmentation for managing degradation of persistent pesticides in biomixture. Garima Sethi, under the guidance of Neera Singh, observed that highest degradation was observed in 1% wheat straw biomixture for glyphosate and 5% biochar mixture for tricyclazole. Further, bacterial inoculation enhances the glyphosate degradation in biomixture. The study was validated in laboratory constructed biobed, and similar trend was observed. Glyphosate and AMPA degradation was highest in 1% biochar biomixture, and tricyclazole degradation was highest in 5% biochar biomixture, and complete tricyclazole degradation was observed in 90 days. So combination of pre-exposed soil, biochar amendment and bioaugmentation, this can be employed to increase the efficiency of biopurification systems. Shubhjit Rakshit worked on the status of glyphosate residues in waters of NCR region and its sorption behavior in soil. He developed analytical method for glyphosate and its important metabolite AMPA with LOQ of 0.05 and 0.5 ppb in water. They collected 263 water samples from NCR region and different parts of India and analyzed for glyphosate and AMPA residues, and highest residues of glyphosate detected was 0.3 ppm. Sorption studies were carried out in 4 different soils. Highest mobility was observed in antisol soil. Molecular synthesis of molecularly imprinted polymer for removal of triclosan was done by Amandeep. He prepared 32 MIPs and NIPs selective for triclosan and characterized using different spectroscopic techniques. Surface modified MIPs demonstrated improved recognition. Also, magnetic core provided easy separation using an external magnet. Prepared MIPs were successfully used to remove triclosan from fortified samples of water with more than 90% removal efficiency. Dissipation of oxytetracycline antibiotics in honey was carried out by Arunima. She developed the analytical method for tetracycline and chloramphenicol with method LOQ of 0.001 ppm. Feeding of honeybees with oxytetracycline during pre-honey flow period led to higher residues in honey even after a month. Degradation of OTC was faster and slightly increased at higher temperature. Minimum waiting period of 3 to 4 months was required for complete degradation of residues. Coming to the publication, the faculty has published 62 publications. Out of this, 27 student research papers were published with 8 practical manuals, 2 book chapters, and happy to share that 45 research papers were published in NAS rating greater than 8. This is the list of publications. Our students are well placed. 3 students which were awarded degree last year were selected in ARS. Only 3 seats were there. All seats were backed by our students. 2 students who are receiving degree this year are already ARS scientists. 1 student is working as assistant agriculture officer in Orissa, and Devbrata Ghoshal is selected in NABARD. And all 8 MSc students who will be receiving degrees this year have joined the division for PhD degree. These are the honors and awards given to these students. They have bagged various first and second position in various competitions. Dr. Supradeep Saha has received NAST Recognition Award for 23-24 in the area of plant protection, and Dr. V.S. Rana has served as expert member in various committees. Division has organized 2 industry academia meet with Tata Rallies Company and PI Industries for the benefit of the faculty and the students. These are some of the pictures showing the fun moments in the Division of Agricultural Chemicals, Diksharam, Freshers Welcome, New Year celebrations. These are some of the equipments we have purchased in the PG lab to strengthen and from the funds received from the PG school. With this, I thank the Director, Dean, Madam, Joint Director, Extension, Research, Associate Deans, Graduate School, Head Faculty, Student and Staff of the Division of Agricultural Chemicals for all their help. Thank you. Thank you very much.
Thank you, Professor Suman. Congratulations. Well done.
Thank you.
And now we move to the next presentation in entomology by Professor Subramaniam. So over to you.
Good morning. Respected chairperson of the session, Dr. H.C. Sharmaji, and convener of the session, heads of the divisions, professors, faculty members, students. I welcome you all to the presentation of the achievements of the students' research during the year 2024. So this is we have besides the IRA Division of Entomology, we have the IRA Jharkhand, 1 student is receiving the degree, and from NABS and Raipur, 3 students are receiving the degree. From the scientists associated with the ASRB hub and ASRP and pollinators, there are 2 students are receiving the degree, and we have the faculty from all the sections for guiding the students. And 16 research guides are there, no new guides during the year. Last year, Dr. Deb Janide retired. And on roll, there are 52 students who are pursuing their MSc and PhD degree, of which 24 are receiving degree this year. So our division, we have the major sections, biosystematics, insect physiology, toxicology, and IPM. Totally 20 degree recipients, and under which sections, varying number of students are receiving from toxicology and other section as indicated. I'll move on to the individual presentations and the details. This year 13 MSc students are receiving their degrees, including Saurav Chakravarti, Arindam Kumar, Sharad, Pradeep Raja, Raiza Nasreen, Durve Kanchan, Vinota, Akash Kotra, Dev Kumar Mandal, and Komal Fia from IRA Jharkhand, and Devanjan Basurai, Chaitna Kumar and Sneha Sharma from NABSM Raipur are also the MSc students who are receiving the degree, along with the students from Division of Entomology here. And this year, 11 PhD students are receiving degree, including Dr. Anil, M.N. Rudragowda, Hemant Kumar, Kalyanu Sai, Aishwarya Lakshmi, Mahendra, Tanmay Kumar, Padala Vinod Kumar, Gundreddy Raja Reddy, Pinlin, Chaitanya, Rakesh Kumar. So let us move on to the MSc research studies conducted by students. First, we'll move on to the achievements of the section under the biosystematic. Saurav Chakravarti has worked on the utility of especially the AI tool by complementing the biosystematics with the traditional taxonomy, how the AI tools can be integrated. Yesterday, he also made the presentation and the merit medal and also won the award. What he did actually was, he did under the cruciferous crops, recorded these different crops that are infesting them, doing the taxonomical studies, and this using these taxonomical keys, identifying the insects. Then he captured the images from the real-time field conditions at different growth stages of the crucifers, and these images have been integrated into the database, especially the YOLO5. You only look once, that is the platform you use, out of which the model 5L based detection model was developed, and it is detected, and it's also the model which was developed is portal and the ICAR Krishi portal. and it can be integrated with a traditional diagnostic tool. So a total of 20 common insect species were identified, and 14 of them have been redescribed and diagnosed comprehensively with 60 illustrations. This is the first part of his work using these traditional taxonomic tools. The second part of the text is only he has developed this diagnostic tool, and it's available, and it is a scope for developing it as a technology. The next student worked on the isothermal nucleic acid amplification assays for detection of Thorimoya absoluta. Earlier it was referred to as a Tuta absoluta. It is very common tomato pinworm. The very need for is that since it is a quarantine pest and also invasive pest in many areas, there is a need for the quick diagnostic techniques. So the student tried to do the loop mediated thermal amplification technique as well as the recombinase polymerase assay for ease of identification. The LAMP assay could easily identify this within 45 minutes, whereas recombinase polymerase amplification technique could identify the insects within 25 minutes. Such kind of quick diagnostic tools are needed, especially in the quarantine centers and others, for easy identification of the non-novices also. And second work is by the Raisa Nursing, guided by me. She worked on the honeybee gut microbiome float. This year the focus was continuously we are profiling on the gut bacterial diversity of the honeybees. The focus given was actually on the insects which are considered as the beneficial symbionts, and in continuation process we specifically devised the media for isolating those which are the symbiotic bacteria, so that the information derived and profile one we can develop a probiotic consortium. So we have enriched some more isolates during this year, especially this year the student focused on the lactobacillus strains, and 3 important strains have been identified in the lactobacillus genera, and also another symbiont Bambala apis. So with the enrichment of this, few more beneficial symbionts, there is a possibility of developing a probiotic consortium for sustainable bee health. Next, this is a work from a student from IRA Jharkhand. Here, the student tried to work on the interaction of the plant biochemical compositions vis-a-vis the growth parameters of the Photoleucopsis. And this is a kind of how the plant biochemical traits are interacting with the pests. And various studies have been taken up on the dynamics of the pests at the different growth stages, and also they evaluated the different varieties, and also they added the information how the gut bacterial communities also interact with the pest-plant interaction mediated by them. They identify the wide diversity of the bacteria as well. Further studies are needed to have some valid inferences on the interaction of the pests and also the role of gut microflora, especially in dealing with the resistance or susceptibility status of the pests on the liche. Pradeep Roja worked under the Suresh Navapre. He focused on the behavior modulating plant volatiles, especially focusing on the leafhopper Amrasca bigitula. This is a major pest in many vegetables, cotton and other crops. So different techniques have been used for capturing the volatiles, and he identified some of the volatiles as indicated here. The main focus is now the plant volatile based chemicals have been used for deriving behavioral manipulating chemicals. The lead obtained from them by using the different studies of the alpha-titometric studies or the semi-field studies and lab studies, give an indication the possibility of exploring them as an attractant or as a repellent, or also as an oviposition studies. Some leads have been obtained there. For further studies are needed to validate them and also to explore their utility for the pest management. So the student guided by Dr. Mohapatra Dev Kumar Mandal worked on the termite scenario in the maize residue based conservation. Normally the crop residues actually pose a threat for the development of the termites, especially as a pest in the subsequent crops. How we can manage them, how different cultural practices or others actually influencing the termite intensity, and are there any interventions by the other agronomical practices or the cultural practices, how they affect the damages. And they studied under the conservation ecosystem, and they studied that both tillage practices and nitrogen treatments significantly affected the termite infestation. That is really an important one. It's further studies have to be taken up how the termite management can be done in the conservation agriculture, where actually we promote the crop residues as well there. So there again there is a possibility, or flip side is how to manage the termites in those scenarios. And the student Chetan Kumar worked under Dr. K.C. Sharma. This is from the NABSM Raipur. He worked on the effect of potassium silicate against pink stem borer. Silicon is one of the one, it gives the hardiness to the crop varieties as well as on the resilience of the crop varieties to the pest. Here they work under the utility of these things, especially the amendments, how the supplementation can help the pink stem borer incidence in the wheat. And they tried different amendments and found that there is providing some kind of resilience, and elevated carbon dioxide without silicon actually increase the larval survival and others. Devajan Basurai worked on especially the screening of Vocro germplasm for the resistance to yellow vine mosaic diseases transmitted by the whitefly, because the whitefly vectored diseases are a major problem in the windy diseases. The screening of the germplasm gives some leads which are the germplasms actually provide not only the resistance to the pest and also how we can contain the transmission of the diseases in the Vocro. The genetic diversity and population dynamics of Samsakta biguta, again this is a work carried out at the NABSM Raipur under the guidance of Dr. Sridhar. Snagha Sharma worked actually on there are leafhopper population, especially in the sucking pest, the insects occur as the species complex. There are the population level variations which cause the varying intensity of the pest in different crop scenarios. They worked on the genetic diversity of the different leafhopper populations and they came up with a clue how there are diversity of the haplotype diversity variations, and are there any impact on these population variations occurring on the different populations collected from the Chhattisgarh, and also they carried out the population dynamics and damage scoring in different regions of the Chhattisgarh. Another important study is under the guidance of Kumar Nag. They worked on the utility of the pollinators in the pollination service and identifying the right kind of pollinators for the right kind of the cropping system. There is a recent incident, besides the honeybees, there are other pollinators playing a major role. So there is a need for recording the role of different pollinators and their pollination services. And the students have come out with a good work. What are the different pollinators, native pea pollinators? They are abundant in the ecosystem, especially in the IFS model system. And stingless bees and xylocopa, they are more abundant in the IFS models. And another important services is the pollination service. This is gone unnoticed by many policy makers as well. Recently, the general survey done by the ASRP, honeybees and pollinator, and the project director, Dr. Sachin Suresh, came out with the wonderful information. The pollination services done by the bees and other pollinators can be worth at least to the value of 50,000 crores annually. They are doing the unpaid service to the farmer. This is a kind of one of the bio inputs, and there is a need for assessing the pollination services by that one. This student worked wonderfully how in the pollination services can be valued. For this particular cropping system, the value in the IFS model by the pollination service is worth 53,000. In another one, 51,000. More studies are to be entertained to have a policy intervention. The pollination services done to augment the crop productivity. Again, another work is in especially vegetable crops, the role of the pollinators are more emphasized, especially in the cross-pollinated crops. And which are the crop pollinators are doing the services, and are there any difference with the hand pollination, or are there differences whether there is a quality or quantity of the fruits are influenced by the pollination services? They have come up with a very interesting ones. The stingless bees are more efficient in the protected condition than in the open condition. That is a one. Better fruit quality is that when the flowers are pollinated by the stingless bees than the hand open, and conversely the fruit set is higher in the hand pollination. This is a message for the vegetable breeders or the vegetable researchers, how effectively we can use the pollinators for improving the fruit quality. So this is the important achievement. Just I highlight some of the notable publication. Saurav Chakravarti came with a very good publication in the smart agricultural technology based on his MSc thesis. Next we will move on to the PhD research projects. Here again I will go systematically with different disciplines. First with the biosystematics. Pinlin, guided by the Devjanide, work on the pollinator diversities. They explore the pollinator diversity, especially from the members of the Apoidea in the Meghalaya. And their studies recorded especially 2 new species of the Apidae. And their family Collichidae and family Antrinidae, they have recorded the new records. And these pollinated diversities and are also the new records of the thing that augment use of the newer pollinators, identifying them and use them under the cropping system, for which these biosystematic studies have provided a wonderful information. And they also recorded what are the different plant species they are visiting and what are the different pollination diversity, their phylogenetic association, variations in the region-wise is documented by the student. This is an important publication from the student, and she also received awards and also recognition in national and international conferences. Dr. Anil, guided by me, he worked on the role of the symbiotic bacteria, especially in the case of white dwarfs. They have a specific adaptation of the fermentation chamber. They harbor the microbes. They play a major role in the nitrogen cycling of the insects. He carried out by doing the work in Himachal and other areas, and what are the different gut bacterial diversity in the white dwarf species. And we also tried to work out how the different nitrate reductase or nitrate reductase enzyme, especially in the gut of the insects, play with the nitrogen cycling of the medium, because the level of the nitrogen content, especially the applied nitrogen, affect the crop in terms of the white dwarf incidence. What is the role of this gut bacteria and the nitrogen cycling was elucidated by undergoing the different biochemical, molecular and microbiological studies. So, Dr. Rudrakoda take out the work for the genome-wide analysis for identification of the odorant binding protein and the chemosensory protein. Yesterday the student presented in the IRA merit medal presentations. They have developed a perfect computational pipeline for identifying the genome-wide analysis and characterization of the odorant binding proteins and chemosensory proteins. We identified 14 odorant proteins, of which 6 are reported new to the science. 14 chemosensory proteins have been identified and 10 ejaculatory bulb proteins have been identified. These are also homologous to the chemosensory protein. The role of these proteins are actually, they play a major role in interacting with the plant volatiles. These are the frontline molecules interacting with the plant water. They have these chemical cues only intercepted by these proteins, and they act up. Then there is a possibility for identifying a novel attractant, repellent, as well as the oviposition deterrent. Extensive functional characterization have been carried out, and the leads obtained from the studies as can be taken up for developing a newer technologies based on the behavior manipulation of the whitefly. The student came out with the 5 publications. And next is the work done by the Hemant Kumar. Under the climate change scenarios, the increase in the temperature affect the pests differently, especially their reproductive system is manipulated, how it is manipulated, and what are the changes occurring in the reproductive physiology of the insects. He explored it. And he came out that the longer mating duration and elevated antioxidant levels occur. And there is a balance. The insects actually divert the energy for the survival rather than for the reproduction. So this kind of the trade-off between the physiological mechanism provided the resilience to the pests. Those pests which are exposed to the thermal stress actually give them in the next generation the resilience for the survival as well as for the further build-up under the climate change scenario. Further, more studies are taken up how the climate change scenario influences the physiology, especially the reproductive physiology of the insect. That will give a clue about the abundance and pest distribution in the coming era. Aishwarya Lakshmi worked on the plant resistance to fall armyworm in the maize jump blossoms. She has done especially on the biochemical basis of the resistance in different maize lines. And they identified especially the A lines, R lines of the different maize jump blossoms and their level of the biochemical defenses against the Spodoptera frugiperda. What are the varying levels of these plant biochemical constituents, and the defense kinds are altered in different jump blossoms, and they come out with a good publication, Induced Biochemical Variation Maize Parental Lines, how it affects the life table and a specific reproductive potential of the fall armyworm. Another student, same on the maize crop, he worked on the other Spodoptera frugiperda, how it is giving a fall armyworm, and what are the constitutive as well as the biochemical in the Cheiloportalas. Sorry for the mistake in the title. They worked on the Cheiloportalas pest. And similarly, how the phenological traits as well as the photosynthetic pigments interfere with the preference and the biological fitness of the lines. They identified that especially the developmental duration of the chylopotellus was relatively longer, whereas some of the other parameters were relatively lower. So different biochemical constituents modulate the different growth and developmental features of the pest differently. So there is a need for understanding how each of these biochemical constituents in turn influences the pest development. Tanmay Kumar worked on the characterization of the wild relatives. In the case of Brassica, for development of the newer varieties, there is a dependence on the wild relatives for especially the developing the resistance against the pest. So he worked on the different crop cultivars as well as the wild relatives, and he extensively conducted studies with the 32 genotypes comprising 29 from the wild crucifers and 3 from the Brassica junctia, and how they respond to Lyphovis erasimi. The interesting leads are, they identified that wild cultivars like Brassica fructuosa and others showed the higher resistance to the Lyphovis erasimi. Similarly, certain lower nymph aphid preference was found out in some of the wild Brassicaceae lines. So based on these studies, they come out with the 2 important publications as indicated here, one in Phytoparasitica, and another publication is on the how the phenology and morphological traits actually influenced by the wild crucifers vis-a-vis the infestation of the Lyphovis erasimi. Padala Vinod Kumar worked on the rice crop phenology and the brown plant hopper, especially the role of BPH-induced plant volatiles. There is a tritrophic interaction, the plant, the volatiles influences the herbivore. Another one, the volatiles released from the insects, they influences the predator and parasites abundance. He studied those interactions, how different crop phenology influences the BPH at one stage, that is at one level. And another one, how the varying intensity of this pest level influences the predator abundance. By using the chemical ecology studies, they identified that spiders prefer the BPH damaged plants under the ambient as well as the elevated carbon dioxide condition than the undamaged plants. They don't find any difference between the BPH damaged plants and the ambient and the others. So Gundreddy Raja Reddy worked on the mass multiplication of the Metarrhiza anisoclea, and he standardized the technique. And Chaitanya came out especially on the foraging potential of the ladybird beetle, and this is as well very much important to optimize the release of these predators under the augmentative biological control. Such basic studies are needed. How many predators can be released at a given conditions? Rakesh Kumar similarly worked on the role of the predators against the Babesia tabaci, which stages of the predator are more active and which stages of the predators are actually can be used for the biological control of the thing. He found out that Chilomenus sexmaculata, especially 3rd and 4th instar grubs, were more effective and can be used for the augmentative biological control. Such basic studies give what is the stage of the insect which can be released, which can be used. These are the salient publications, 42 publications last year by the students, and of which more than 10 rating were 9 publications, more than 6 rating were 32 publications. These are the details of the publications. This is a student placement record. Besides the ARS services, they also placed in the agriculture department and also the private establishment. Most of the MSc students who have completed joined the PhD program. Students have received awards and recognitions. The notable one is the Rudragoda yesterday received the gold medal as well as the Best Outgoing Student Award of PhD award. And he is also nominated for the Guru Prasad Pradhan Medal for the Best PhD Student from Entomology. Saurav Chakravarti received the MSc Gold Medal. These are the other goals and awards and recognition by the students. Our faculty also got many recognition. Our head received the Fellow of Royal Entomological Society, and I received the fellowship from the NASI membership as well as the NASA fellowship. And our other scientists also got NASA fellowship, Young Scientist Award, and laurels and recognition. Vibrant activity from the students actually give color to the division. They are involved in very engaging activity themselves as well as with the faculty. There are different training and extension activities conducted by faculty to the students. These are certain colorful activity by the students and various programs, Deepavali, Holi, Freshers Welcome. The students make the divisions more colorful. I thank the support by the Director, Joint Director of Education, Research, Extension, Joint Director Administration, Head of the Division, fellow faculty members, faculty from other hub and others. All of you, I say a big thank you. The support given by the PG School is tremendous and excellent. Last but not the least, the contribution of the student and research scholars actually give the facelift to the divisions. They actually reflect the excellence of the division. Thank you all students for your contribution. Continue to do the same in the coming years as well. Thank you.
Thank you, Professor Subramaniam.
Thank you.
A big applause to all of you once again. Now the next presentation is by Professor Anil Sarohi in nematology. So over to you, Dr. Sarohi.
Thank you, sir. Good afternoon to the House. I stand before you to present the salient findings of the students of Division of Nematology. And we have a faculty of around 17, and we have 26 PhD students and 3 MSc students, of which 5 MSc students are taking degree this year, and 2 PhD students are also taking degree in this convocation. Coming to the themes, we have 4 major themes in nematology under which the research is being conducted. And of the first theme, that is nematode diversity and biosystematics, we have 3 students, of which one is Srinivas Gowda. He has basically looked around for how plant parasitic nematodes in the protected agriculture get impacted by soil properties as well as the under climate and all that. So with respect to the studies, it was found that when we went looking at the status of these varieties of crops and the soil pH, soil EC and all that, so it was found that root knot nematode, that's Meloidigyne, basically prefers organic soils and it's straightway associated with potassium and nitrogen. So healthy soils propagate Meloidigyne. Platylincus is situated to the vector near to the pH. The pH influences Platylincus considerably. Hoplolymus also pH influences. Helicotylincus is again close to vectors of phosphorus and nitrogen, so appears to benefit with these nutrient-rich soils. And Tylerorhynchus is strongly associated with organic carbon, so carbon-rich soils basically propagate Tylerorhynchus. Coming to the next study, that is by Sharad under Dr. Bharat. They intercepted consignments of paddy seeds and looked for the Aphelencodes baccii, which causes the white tip disease in rice. And they found that in around 18 out of 150, that is around 12% of paddy seeds or the consignments were found to be infected with this nematode. And the infested or the infected samples also, if you look at many of them, were all infested. Every seed of paddy was infested, especially Rayagad area and all that. But this range varies from say 50 to 100. And even consignments coming from USA and China, they have a considerable infestation levels. So we need to be careful with respect to seed because this nematode spreads because of seed. Also, they confirmed this, that it was Aphelencodes baccii using the molecular markers and phylogenetries. The study conducted by Vijayalakshmi Saikia was regarding the cyst nematodes in the campus. And she studied in different fields of IRI, and she found that only 2 species of cyst nematodes, that is Heterodera aveni and Heterodera cageni, have been documented from this IRI campus. Other nematodes which were tried using the molecular mechanism, Cephalipja and all that, Z, so they were not found to occur. So as of now, we are infested, our fields are infested with these 2 cyst nematodes. And the nematode management theme, we had a student, Mr. Suman Panja, working with Dr. Pankaj, and they came out with a very interesting outcome, wherein they used nano formulations of garlic oil and other oils and compared in vitro as well as in vivo. In the in vitro studies, you can see that the nano emulsion formulations of garlic oil are giving very good LC value, and same sort of results were observed when in vivo studies were done using the root dip. And so if you look at that is T2 treatment, this is the garlic oil nano emulsion. And another thing which needs to be observed is that the nano emulsion is giving better results as compared to the macro emulsions. That is, the smaller the molecule size, the better the results for management of nematodes. This is the gall number formation of the root knot nematode in rice, basically. Similarly, when you look at the soil trench treatment, there also we had the similar kind of results. So, and the results with respect to chemicals, that is the vellum prime, are also comparable at 30 days. So this looks to be very promising, and I believe there has been some technology transfer of this with the company. Then coming to the fourth, that is entomopathogenic nematodes. One of our students, he worked to find out native entomopathogenic nematode populations in Dakshin district of Canada, which is a major areca nut growing area. And in the samples they collected, they found that around 4 to 5 species of different 2 strains of Heterorhabditis indica, Oschkias species, and Metarhabditis were observed by them. And if you look at their capacity to kill insects, then you can see that the mortality of Heterorhabditis indica is far greater than Oschkias isolates. It's at a lower number, it is able to give you LC50 values. And this was against 2 insect pests, that is fall armyworm, and the lower graph is with respect to the what you call as areca nut root grub. Then coming to the PhD degree recipients, we have 2 of this. Artha Kundu worked on identifying the genes responsible for chemoreception, and as you all know that nematodes basically communicate using chemical signals, and the G proteins are the major proteins which are responsible for switching the information which is perceived at the molecular switches inside the system, and then the nematode starts behaving as per the perception of the surface receptors. In this area, they worked out a complete bioinformatic line, because our department has done the genome sequencing of this EPN, as well as this genome is already available. So comparing the genomes and using biotechnological tools, bioinformatic tools, we were able to identify 4 different genes which are responsible for chemosensory responses in EPNs. Further, using the tools, they synthesized the proteins and they tested whether they were stable and robust. So all those tests which are associated with protein designing, they confirmed that they were very robust and stable. Later to this, they went on for docking and binding affinity, and the results indicate a high degree of affinity and binding, so that this model can be used for generating things which can really modify the behavior of the insect, these nematodes, towards insect parasitism. Their functional validity was done using the RT-qPCR and the RNA silencing. Both the results confirmed. This was regarding expression analysis of the nematode genes, basically after exposure to insects, and this was by silencing the gene. When we took these studies to the validation with C. elegans, and we used the gene, and it was observed that these 2 signals, odorants basically, which are either attractants or repellents. So if you mutate this gene in the C. elegans, the behavior absolutely changed. So if you can look at, so we have confirmed that these genes really work well, and possibly they can be used further to improve upon the predatory behavior of the EPNs. Coming to the last student work, this is Manish Kumar. He worked with me, and here it was interesting to work with a nematode which has been documented first in the history. This is plant parasitic nematode, and it has been known that even after 32 years lying in as a cockle, it can revive and parasitize. So we studied its life cycle as well as interesting aspects where it survived in different stages. And all 3 stages were used for generating the transcriptome of this. And if you look at it, this is an interesting part that if you have a wheat crop which is going even up to 150 days, the nematode is basically aligning itself according to the time period and ultimately matures along with the crop maturity. When you sowing late wheat crop, ultimately it results in formation again of completing a life cycle within say around 100 days. So it is able to shorten its life cycle depending on the conditions of the crop. and possibly it's the growing degree days and the sowing and the day temperatures will impact this life cycle. Now coming to the assembly, this is the assembly around 132.2 MB base transcriptome was generated with transcript levels of around 1,05,606, and there were also differential expression was observed with respect to the anhydrobiotic and male females with respect to upregulated and downregulated genes. And the interesting part is that when we compared the transcripting data with plant parasitic nematodes and the EPNs, especially EPNs, if you look at it, around 88 genes are common, and this is where possibly we can use this information for extending their what you call is shelf life, because today we don't really have a longer shelf life with EPNs, so possibly the information generated through Anguina can be utilized for this. So another aspect of the study was that we are able to identify two important genes, DAF2 genes, which basically help in longevity and as well as anhydrobiosis and stress management. These are common with C. elegans. They are there within this treatise, as well as we have genes which are molecular clocks, so they can be used for establishing the phylogeny as well as the timeline when these kind of genes got incorporated. Publication around 49 of this, majority are in the higher impact factors journals, and we had book chapters. This was both faculty and students. Then awards, Dr. Pankaj was awarded Distinguished Scientist Award in 20 by the Society of Plant Protection Sciences. Dr. Datta completed his Fulbright Fellowship, studied at UC Davis. And 2 of our patents got granted last year. And many of our students received different prizes at separate functions in conferences and all that. And not only academics, they had participated as PGSSU members as well as in sports. So we had a vibrant student community with us, and you can say that they had a tour to Darjeeling as well as, and some happy hours. So they celebrated Teachers' Day with us, and that's all. 2 of our students graduating in PhD, they all have got jobs. So thank you very much, ma'am. Thank you, sir.
Thank you, Professor Sarohi, and congratulations for the job well done. The next presentation is in plant pathology by Dr. Andre Kumar. So over to you.
Most respected Chairman and my dear colleagues, it's a pleasure for me to share the students' achievement from the Division of Plant Pathology. I am Dr. Andre Kumar. I am a professor in this Plant Pathology Division. Let me get into the presentation. As of now, we are 23 faculty. We need to deal with 105 students, including the 20 students who are going to take degree this year. So the ratio is really a tough challenge for us. In fact, we don't have enough faculty to guide our students. So the presentation is made in the 4 format: mycology, bacteriology, fungal pathology, and virology, the classical theme, what we started doing for years. And we deal with a variety of topics, starting from taxonomy, pathogen characterization and diagnosis, disease, of course, detection, and disease diagnosis in epidemiology is another major trust area in our division. Host resistance, susceptibility, both now we are dealing with. Susceptibility is now back into focus because of genome editing tools are looking for more susceptibility genes which can be harnessed in the future. And pathogenomics and metagenomic and microbiome is a new area which we have already started working for last 15 years. Now we see more and more students started taking up this kind of research now. Biocontrol, genome editing, and nanochemicals are another new line of work most of the students started doing, and some of the work I'm going to highlight here. The pathosystem, what the students have carried out in the last few years, is highlighted here. We deal with the nationally important fungal pathogens, and bacterial pathogens, viral pathogens, and also the plant-associated microorganisms, They are going to help us in the climate resilient crop protection in the future. And these are all the pathogens what the students have handled during the thesis projects in the few years. And the notable one is the kernel bund is again and again is a botheration for us, and the students are again and again putting a lot of efforts. And fusariums are always a polyphagous disease in many hosts. Alternaria is emerging. And post-harvest pathogens, it's nice to see that students started working on post-harvest pathogens in the last few years. And Puccinia is again a perennial issue, and the students are continuously putting a lot of efforts. Bacterial pathogen, of course, Xanthomonas is a major problem in monocots and dicots. Students are putting a lot of efforts. Viruses always evolving, and that gives a lot of nightmares for the agriculturist, and students are putting a lot of efforts. And these are all some of the microbial agents people deal. I'll just take it forward. From here on, I'll be taking you to the tour of the students' achievements to start with MSCs, the research outcomes of the master students. The theme on mycology, there are 2 students put a lot of effort in mycology research, Ankita and Pallavi. Ankita worked on fusarium diversity in post-harvest disease in many fruits, and Pallavi worked on a new kind of biocontrol system that is yeast, plant-associated yeast, guided by Amrita Das. These are all the 2 pathogens, the 2 systems the students worked. Ankita's work is very clear. Fusariums are re-emerging or emerging as a major pathogen, not only in the pre-harvest system, but also in the post-harvest system. Whether it is the climatic fruit like mangoes and bananas, they are going to create a lot of problems, and the students have put a lot of efforts. And what she found was the new species of fusarium that is going to create problems, that is the Fusarium pernambucanum. That's going to be an issue. And Fusarium verticillidus, normally we found in monocots, now reported in bananas, and Fusarium irregulare in citrus are going to be problems. And Ankita's work going to be a future for post-harvest disease management using a plant-associated yeast, Hansinia fora, that the scientist is continuously giving this problem to the students. Maybe in the future it come out as a technology. And this is all about the detailed characterization of this yeast. And she brought out a lot of new volatile compounds the yeast is synthesizing to suppress the pathogens. In fungal pathology, large number of students handled the projects on fungal pathology. Hemant, he worked on Puccinia striformis in yeast and identified the new resistance. Smita, yesterday, she made a presentation for award. She handled kernel bund, and Shivaraj worked on fusariums, guided by Dr. Vaibhav, Dr. Malkan, and Dr. Saharan. And Shreya and Chaitra, guided by Dr. Bagh. I'll come to that later, what they have achieved. And Jyoti was handled by Dr. Shanmugam. In terms of a student from Jharkhand, guided by Lakshman, we'll come to that later. These are the pathogens the students handled, the fungal pathogens. I already told. I'll just take it forward. Hemant worked on stripe rust of wheat and identified several resistant genes in those cultivars. And also he found a very strong correlation among the resistant parameters. And also he advocated the adult plant resistance as the way forward for sustaining the wheat rust disease management. And Tilicia indica, yesterday she made an excellent presentation. The resistance always should be blended with the other method of disease management to stop the pathotype evolution. That is one of the highlights of yesterday's presentation. Resistance plus biocontrol could be one of the way forward to stop the emergence of new pathotypes. I think that's a highlight of her presentation yesterday. And she published her work in Journal of Micropathological Research. Shivaraj, he worked on head blight of wheat and screened many wheat cultivars and concluded that none of our Indian wheat cultivars are resistant to this disease. And either they are moderately susceptible or highly susceptible. So that means a lot of efforts are needed from the future students to identify the resistant cultivars. Shreya worked on, this is a very interesting project. This is something like new use of a known agent. So Beauveria bassiana, we know that it's a kind of organism which can suppress insects, but they are trying to attempt to control plant diseases. This is a new line of work, the new use of known agents. And again, she identified some resistant cultivar in chickpea. This once again against the Sclerotium sclerotiorum. That's one of the pathogens. Chaitra again worked on the similar line, coming out with a new concept, like the same concept like how best we can use a known agent for seed disease management or seed pathogen management. And she come out with some excellent agents like Trichoderma asperillum, procured from ITCC as a new agent. Bavaria bassiana once again for management of seed-borne diseases, I think guided by Dr. Bagh. Jyoti Kumari from Jharkhand, guided by Dr. Shanmuga. And again, she found that a new species, Pernambucanum, is a major pathogen. And come out with a clear-cut explanation. We always deal with this problem in wilt diseases. The material is either resistant or it is escape. I think she come out with some explanation. It is purely environment-induced. Under certain environmental conditions, plants do not get affected. And she called it as escapes. And again, they correlated with the season, her Kharif and Rabi. That's a beautiful finding from the students' work. And Tanisha, again from Jharkhand, guided by Dr. Lakshman, and come out with a new concept whether bacteria and fungal agents can be mixed together as a common a formulation to control plant diseases. I think she come out with some baseline data and analyze the metabolome profile of individual organisms in combination, how they really work out. That's again one of the finest work. And that was all about fungal pathology. And virology, we had two students who worked on two important pathogens. One is citrus triticum, another one is a new disease appeared in Maharashtra. And both the students addressed very nicely, come out with them beautiful explanations. To come, guided by, of course, Dr. K.K. Biswas and Dr. Nirban. To start with the first work, Prantik Mazumdar, yesterday he defended for a gold medal. And he come out with a clear-cut etiology of this particular disease that was really bothering tomato growers and market at large. The tomato prices have gone up like anything because of this particular disease. And the shoestring disease, he clearly come out with a new viral agent responsible for this disease. That was a finding from a master's thesis. And he published this work in Plants. And Chandrakesh and worked on citrus triticum. He mapped the disease in Northeast, come out with the variations in the pathogen. I think that is one of the reasons why the disease is still around in citrus garden. And come out with an excellent molecular explanation for high rate of recombination-based variability in this particular pathogen. That was all about master's thesis, although master's thesis is only for 8 months, at the best 10 months, but still they come out with an excellent findings from their work, and some of them they really published. I'll move on to the doctoral thesis. Fungal pathology, again, 5 thesis projects were carried out in fungal pathology, dealing with the nationally important pathogens. And Jagadish worked on the histopathology of rice when it is with fusarium interaction. And Sangamitra worked on Helminthosporium, a well-known disease in wheat, come out with new findings. And Vineet worked on microbiome of maize to find some new solution for rhizoctonia, guided by Vishnumaya Saharan and Dr. Gogai. And Sudipta, and she worked on microbiome, endophytic microbiome of maize to find a solution for Bipolaris maidis, one of the foliar pathogens of maize. And Deep Narayan Mishra, he worked on nano formulation generated through microbial interactions, trichodermas, and come out with the new findings. And guided by Dr. Robin and also Lakshman. Little bit details. Jagadish come out with the finding that the fusarium, the finer interaction between fusarium fujikuri and rice, he did a detailed microscopic analysis how exactly the pathogens are colonizing different plant compartments, come out with the new findings, and he come out with a good publication in Journal of Agricultural Research. So Sangamitra's work is an excellent work. She really handled the genome of several pathogens, which is responsible for leaf blight. And the popular notion is Bipolaris Sorokiniana is a pathogen, but she come out with a few other pathogens associated in the complex and come out with an excellent finding. That's the highlight of this particular work. And of course, this project was supported by many other agents. This paper was published in Frontiers. And Sudipta's work is a new line of work for maize microbiome. She handled the microbiome of maize in order to identify the potential agent which can suppress the bipolar maize, and she came out with the finding Stenotrophomonas multifila and Brevandimonas and Pseudomonas aeruginosa as a potential agent. Of course, this will be under further scrutiny to come out with the best one which is safer for agricultural use. I think somebody should take it forward in the future research program by students. And she also had a visit to Australia under NHP cost, and she published her work in Indian Journal of Agricultural Research. Vineet Vijayan, again, did a new line of work for the entire division, microbiome of maize, once again, come out with a finding like Penicillium oxalicum is one of the agent, can suppress the rhizoctonia is a problem in maize, and considering the safe nature of this Penicillium oxalicum, I'm sure this agent can go to the production, mass production, and can be a technology in the future. Deep Narayan Mishra is a completely the next-gen disease management using nanotechnology. He came out with a trichoderma-assisted generation of zinc oxide nanoparticles and used this to suppress Alternaria brassicae, is a problem in Brassica family, as a new option for disease management as a foliar spray. I think he did a very in-depth study and he could publish his work in Frontiers. So that was all about the fungal pathology. Now I will move on to bacteriology. There are 4 theses under bacteriology. The students dealt with the pathogens like Xanthomonas in rice, Xanthomonas in pomegranate, and also some of the foliar adapted fungal, beneficial organisms like Pentoya. So Karishma, yesterday she made a presentation here, guided by me, and Kalaivanan, he was guided by Dr. Kalyan, and Mrityunjaya, again guided by Kalyan, and Rashmi, again guided by Kalyan. In brief, I'll tell what they have done. Karishma, her project was supported by Prime Minister's Fellowship, come out with an excellent finding. Foliar adapted epiphytic Pentoya can be one of the options for foliar disease management. In the long run, it can reduce the amount of fungicides which is applied on rice crop. Indirectly, a way forward towards organic method of foliar disease management in rice. I think this paper was published in Microbial Pathogenesis. Mrityunjaya come out with the finding the gene HPA in Xanthomonas is actually responsible for causing all kind of damages in rice, pomegranate. And she handled this particular gene of Xanthomonas actinipodes punicae and attributed the role of this particular gene for a variety of other physiological malfunctioning in the plants. So that is a finding. And this is one of the fine, good piece of work the student has carried out using the technology for conferring resistance in rice, using the CRISPR technology, CRISPR-Cas. I think one of the pioneering students, this work, who handled the genome editing as an option for disease management. She handled CRISPR-Cas technology to confer resistance in rice against bacterial blight by targeting one of the genes, what is called as sweet genes. I think the protocols, the technologies, whatever she developed, I'm sure it will go the long way, assist future students to take up such a projects, right? The work has been already completed, and she has already published her work in Australasian plant pathology. Kalaivanan, and again, I think Kalaivanan was a senior to this particular student. He came out with the genes which is conferring susceptibility in rice, that is sweet genes. And the next student followed it up to confer resistance. I think these 2 theses can be linked, and that is how the follow-up work can lead to something which is very useful. Yeah, he also published that work. So with that, I conclude the achievements of the students. From here on, I'll take what the division has achieved in the past, research publications and other accomplishments. I just brought only the students' publications here, 19 of them. And most of these papers appeared, 15 of them appeared NAS rating 8 and above, that is impact factor 2 and above. So that's a very good score. I think these papers appeared in a variety of journals, and some of them are APC journals, and most of them are the subscription-based journals. So that's a very good point, because you don't have to pay any money on that. So these papers dealt with the pathogens, what is highlighted on the side. So we made a small attempt in the last 3 months, I would say the last 2 months, to generate the Google Scholar account of the Division of Plant Pathology. So I added 3,000 papers in this particular Google Scholar, right from 1905 And this is a highlight of this particular divisional profile. The divisional publications are one of the indicators how the division fared in terms of academic publications. So this is a Google Scholar account of division of plant pathology. And you can see here the paper, whatever appeared right from 1907 and right up to 2025 is getting auto-updated. So far, I have added 3,000 papers. And one condition is either the paper author should be the first author from the division or it is a corresponding author from the division. No other papers are included here. So this is, I think, we added all these papers in this particular work. Honors and awards faculty, I think large number of awards were backed by our faculty. And I think I just go back to the previous one. This is getting auto-updated automatically. So that's a good point. And again, a lot of scope is there to, if you want to retain the best papers, you can always eliminate the paper with less impact. I can put the new paper there. So that showcases how this division fared in the academic environment. So this is awards, large number of awards backed by our student, our faculty. So the entire list is here. I don't want to read it here. And the students who made imprint in the academic arena, large number of students, they bagged one or other awards in conferences and in the society conferences or the best paper, and that's a list. And faculty who visited abroad, either by invitation or on their own, I think 4 of our faculty, they visited, myself visited Seattle upon the invitation of Gates Foundation to discuss with endophytic microbiome. Dr. Prakash visited Bangkok, Crawford Fund. Amilendu visited US under Fulbright Fellowship, and also Sri Lanka for study in India fair. I think that was our faculty visit abroad. I must tell here, in addition to ICAR Senior Research Fellowship, the students have brought laurels to the division. They brought fellowships from other agencies also. The competitive fellowships, I think the list is already here. It is INSPIRE Fellowship, or it is an Netaji Subhash Chandra Bose International Fellowship, or UGC, JRF, NFST, and FIAM Fellowship. So that's the laurels that the students brought to the division. And I must tell here, the students not only did work here, they also went abroad to top up their research. I think this is how the students went different countries. I think 8 of our students, they traveled all over the world to acquire knowledge, and they learned many things. And upon their return, they implemented all those new learnings, and their perception and their output started completely changing. So this is the laboratories where they visited, right from Spain, Australia, Japan, UK, Netherlands, and also Malaysia. So that is achievements of the student, and thanks to NHP COST that brought new dimension for students' research in the last few years. And we also conducted a lot of programs. I'm not putting all the programs here, only the key ones. And this is a 10 days program in NHP COST. Once again, Mushroom training program is ongoing program, plus several programs we conducted in collaboration with the industry, mostly to make the student to understand how to use the particular facility. I must mention here, even offered a training for handling the genome sequencing using nanopore technology. And after learning, the students started doing the genome sequencing on their own. So we also invited 3 internationally reputed scientists to deliver our faculty. Professor Savari is adjunct professor here, and he regularly comes and take classes and address trainees. And Papu is from Seattle again, and he Washington State University, and he comes here and deliver a talk. And Frank White talked about the genetic aspect of Xanthomonas. Patents and technology, we are second to none. Patents have been granted for some technologies from virus, and the technologies in collaboration with other institutes have been converted into a kind of commercially viable things, varietal development in soybean. And I must really recall the funding agency support for supporting all our students. I think 14 projects, and these projects brought around closer to 4 crore rupees to the division. And that really supported all our students directly or indirectly. So thanks to the funding agency for liberal funding of many projects. So with that, I'll close my presentation, but not before thanking Director, Dean, and Joint Director Education, Joint Director Research, and admin, registrar, head division of plant pathology, and head division of collaborating divisions, because without collaboration we can't do anything. MOHR, funding agencies, and several funding agencies who supported us. Thank you all.
Thank you, Dr. Kumar, and congratulations to your group.
Thank you, sir.
So, thank you.
Thank you to all the professors for the crop protection session. May I now request the chairperson, Dean Ma'am, and the co-convener to please join us on the dais. Okay. I would now like to request the chairperson, Dr. H.C. Sharma, for his kind remarks.
So a very good afternoon to all of you. I think all of you have made excellent effort, made excellent presentations, the research outputs, and my congratulations to all the degree recipients in the 63rd convocation of ICAR, and of course the congratulations to the mentors of these students who guided them to uphold this ICAR flag and the flag bearers of research and future. I have probably written as much notes as probably the students have written in their thesis, but I will just focus on a few observations that probably are relevant to most of the researchers and our disciplines. Of course, some of the comments could be relevant or only to a particular discipline or to a topic, and of course they should be read as something like we synthesize several novel compounds, and I know this research for the last 50 years because I was a student here in 1976. So we have to actually do a SWOT analysis, how many of those compounds that we synthesized were well characterized, then do we register them like the genetic stocks of the varieties were registered, and how many of them were patented and how many of them actually but then commercialized by the industry. If it happened, yes, we continue this research. Otherwise, I'm not sure we can match the money that the industry puts in synthesizing the novel molecules, and then the amount of money around to 1,500 millions that is needed in commercializing one chemical compound for crop protection. So we must be sure, but we must take stock of this situation, what we actually do. Do we do something that actually finds application, or we do it and then probably we stay there. Some observations again specific to neem about growth regulation, growth regulator properties of neem bolide or neem and neem constituents. I think this research was also done in the 80s. Probably much of it is described in the proceedings of the first International Neem Conference in 1983. I think you must analyze what advances we have made since then, or we are just retreating and taking that path again and again. And again, a specific comment about phytotoxicity studies in the phytotron. I think that dynamics will be totally different in phytotron than what we do in the actual field conditions. And what happens in the actual field conditions where the UV rays proportion is different through the day dynamics and on different seasons of the year, probably we must take a stock of those things. I think AI-based diagnostic studies, I think we must focus on it in future, but we must make sure that they are user-friendly. There are a lot of money that has been spent on this AI-based or expert systems to identify pests and pathogens of their nature of damage, but how many of our crop protection scientists actually go to the field every day to be able to identify a pest either from the nature of damage or a pest, then what damage it will cause, and of course what levels of that pest incidence or disease incidence are needed to actually for us to focus on those things. So, something like there was a lot of noise on actually fall armyworm. Do we know that what levels of incidence we need to cause economic loss? Even if we remove 30 to 40% of the leaf area, if it is damaged or consumed by insects, it does not reduce the yield significantly in cereals. So we must take cognizance of such effects. Then a good effort has been made to study the role of microbes in the gut, microbes that sit in our gut and also in those of the insects. They play a very important role. One is in case of honeybees, of course, we look at the positive effects, and so is the case with human beings. But in case of insects, these microbial organisms that are sitting in the gut, they also play a very important role in potentiation or detoxification of a molecule, for example, Bt proteins. If we kill the gut microbes through antibiotics, for example, in Helicobacter from my own studies, the insect becomes resistant to Bt toxin. So that kind of information must come out for us to plan our future focus on developing either transgenic crops or develop a technology so that we can delay the development of resistance in insects to Bt transgenic crops or any other gene that we deploy in the crop. So that kind of understanding has to come out for us to plan a long-term strategy. Then demonstration of the value of insect pollination. Actually, this is very important. That is where probably we should not only study this thing, publish the papers in the journals, but we also put this information in the general, digital, and print media so that the general public, the administrators, and of course the politicians are available of the effort that we need to make. First, in conserving the insect pollinators, that if certain insect pollinators are become extinct because of climate change, many of our crops will fail to set the seed, or those plant species will also become extinct, or our yields will be reduced very drastically, and that has been demonstrated. For example, in apple in Himachal Pradesh, now farmers ready to pay 2,000 rupees per hive during the apple blooming period to increase pollination, fruit set, and of course the fruit quality. And such information is also there for pomegranate. We must generate this information for all the crops and highlight the value of insect pollination and the importance of conservation of insect biodiversity, in particular the parasites, predators, and of course the pollinators, so that we focus enough attention on this. Exploitation of wild relatives. I think that is gold mine for us to look for genes that confer resistance to insects, diseases, the nutritional quality, and of course adaptation to drought. And all these studies should not be by an entomologist or by a breeder. This should be interdisciplinary research. In fact, I will again say, Madam Dean, PG School, IRI, you must ensure that many of the topics in research that are undertaken, we must ensure that we work across disciplines, across labs, across institutions, and of course, I'll cover this topic again. We should also encourage our students to go to the other labs. Within the ICAR system, you have 101 institutes, you have CSIR institutions, you have CGIR, the international institutes. If you collaborate, or if the student works here and also works in another lab, he gains double experience of how to work in a different environment, what resources are there, and how he should approach his research in future. So that interinstitutional collaborations within India, across institutions, and of course internationally, that part is also very important. And of course, one thing that came out from Andy Kumar's presentation, lot of students, we must maintain a proper ratio between the number of teachers and the number of students. There are guidelines from the ICAR which it implements while it goes for evaluation or accreditation of the state agriculture universities. If you don't have 45 faculty on board, then the university cannot conduct an undergraduate course. Do we apply this rule in IRI? We must apply it. I had been part of your Research Advisory Council for 3 years, and I highlighted this aspect every time we spoke, that for undergraduate studies, you need a dedicated faculty, you need large laboratories, large lecture halls so that you can accommodate 50, 60, or 70 students. And then if you are taking 1 or 2 courses in a semester, that teacher doesn't have much time to focus on research. So there should be dedicated faculty for undergraduate research, and of course a proper ratio as per ICAR norms between the number of students and the number of faculty. Of course, that aspect I think should be looked into, and plant pathogen antagonistic compounds produced by yeast is very important. Actually, the biological activity of all the chemicals, agrochemicals or pesticides, that depends on the leaf surface profile of the microbials that we have there. And if you want to know, we had one paper on Bt toxicity on different crops on different crops in different regions. So that aspect we also must focus on, and these are very important. Can we just spray yeast on the plants and reduce the disease? Reduce the disease or the insect incidence. So these are the kind of simple kind of interventions that we probably can look into. And for me, it's good to be a student again here at IRI albeit after 49 years. So I was a student here in 1976. So to be in my alma mater, and thank you, Madam Dean, again for inviting me for this 63rd convocation and to be part of this, because I missed my convocation in 1980 to receive my degree in person because I had just joined ICAR, so I thought I shouldn't be asking for a tour. I had no leaves at that time. So thank you very much for this opportunity. And of course, again, a big congratulations to all of you to continue that excellence in science, do good work, train those extraordinary students to undertake research in futures, and good luck to you all. Thank you very much, and have a nice day.
Thank you so much, sir, for your kind remarks. So since the heads and professors of all the 4 disciplines are here along with the faculty, we shall take care of all your suggestions, sir. Thank you so much. So now it's time to felicitate respected chairperson. May I request respected Dean ma'am and Dr. Brahmanand sir to please join us on the dais for the felicitation. Yeah. And for a group photo, I would like to invite all the professors and HODs of all the 4 disciplines. And we also have Dr. Rashmi, who has been a stalwart in plant pathology. So we can also invite ma'am. May I now request my co-convener for a vote of thanks?
So we have wrapped up with the second session of the professor's presentation. I would like to thank Dr. H.C. Sharma, former VC, Y.S. Parmar University of Horticulture and Forestry, to chair the session, sir. Thank you for your appreciation as well as critical comments, sir. We'll try to, sir, incorporate all your suggestions in future line of work. Thank you so much. I would like to thank our director for guiding us for organizing this event. Also, I would like to thank our Joint Director Education and Dean Dr. Anupama Singh for very critically managing all the steps for organizing this event. Thank you, ma'am. Thanks are due to all the professors of the 4 disciplines, agriculture, chemicals, entomology, nematology, and plant pathology, for presenting the work of the students during the past year in a very timely manner, what Dr. Monica had given. So thank you everyone for following the timing. And it is a pleasure for having all the faculty here in the audience. I would hope that we will have more number of scientific staff and students in the upcoming sessions. And also from our sister institutes, the head of the departments, the PD, everyone is here. Thank you so much. And all those who have joined us and who are watching us through the YouTube channel, thank you everyone for participating in the session. And we will start the next session at 2 p.m. sharp. So I would like everyone to please be seated here before that so that we can start in time. We will be having the School of Natural Resource Management and School of Basic Sciences post-lunch. So we will wait for the audience here to be seated here in time. Thank you so much. 2 p.m. ma'am. All to be seated before 2 p.m. Okay. So 2:15, as madam is suggesting, we'll start at 2:15 p.m. sharp. Thank you so much. Thank you, everyone.
I request all the Dr. Brahmananji, all HODs, professors, faculty of NRM disciplines, I can see many of them. Those who have joined online. Now we are having the third session of this 1.5 day overall long program. I welcome you all. This session will witness presentations by the professors pertaining to 7 disciplines, and these presentations will be covering 109 theses comprising of 57 MSc and 52 PhD dissertations. As I always repeat, and I have been, it is customary for the dean to always say that it does not represent, sir, the entire spectrum of the research which is conducted by the NRM school in IRI, but of course, it will give us a glimpse of or what I would say that it's a window shot of the research domains, cross-disciplinary, multidisciplinary, cross-institutional, that are being conducted through the students as the future researchers and as our current instruments of operation. So now I request, so before that I have this assignment of introducing the chair to the audience on behalf of Director IRI. Sir, may I kindly be allowed to perform this duty? Dr. B. Venkateshwarluji is former Vice Chancellor, Vasant Rao Naik, Marathwada Krishi Vidyapeeth, Parabani, and former Director of Krida, Hyderabad. He is an agricultural microbiologist by training and has served in ICAR for over 35 years under different capacities in research and research administration, and for a brief period in ICAR. His main areas of research are biological nitrogen fixation, role of microorganisms in abiotic stress tolerance, dryland agriculture, and climate change. As director at Krida, Sir conceptualized and implemented the NICRA project in ICAR. He has published over more than 250 research papers with total citations of 13,500 with an H-index of 55 and has 2 patents to his credit. Dr. Venkateshwarluji is a fellow of National Academy of Agricultural Sciences and Telangana Academy of Sciences. He has several awards and recognitions from ICAR to his credit, the important ones being Fakhruddin Ali Ahmed Award, Vasant Rao Naik Award, and Hariom Ashram Trust Award. Sir delivered J.N. Mukherjee Memorial Lecture in Indian Society of Soil Science and is a recipient of 7th International Crop Science Congress Award and FAI Golden Jubilee Award. Dr. Venkateshwarluji has been recognized as Honorary Fellow for his lifetime contributions to dryland agricultural research by Indian Society of Dryland Agriculture and Arid Zone Research Association of India. He has served in different committees and task forces in DBT, DST, ICAR, and Niti Aayog related to agricultural research and policy. He was also a former member of GEAC, and he chaired QRT and RACs of KAZRI, CICR, NIASM, and is as a member in few other ICR institutes. Currently, Sir is the chair of the expert committee of NICRA project, member empowered committee of the NASF of ICR, and is the president of Agri Biotech Foundation, Hyderabad. But nevertheless, I would have to say that this is just a brief of of his towering personality, and let us give him a big round of applause. Thank you very much, sir, for accepting our invite and request. And may I now request Dr. Monica to take on further proceedings.
Thank you so much, ma'am. So, as convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentation. For session 3, as already briefed by Dean ma'am, we are having 7 presentations from the professors of the following disciplines in the following order: agricultural engineering, agricultural physics, agronomy, environmental sciences, microbiology, soil science and agricultural chemistry, and in the end, water science and technology. So, for all the 7 disciplines, respective time allocation has been based on the number of students receiving degrees, and the professors will highlight the research outcomes of both MSc and PhD students based on the identified theme areas of respective disciplines. For the information of the chair and the house, the respective time allocation has already been communicated to the professors, and it would be my kind request to all the professors to kindly abide by the given time. For the ease of the presenter and as an indication of wrapping up the presentation, yellow light will blink 2 minutes prior to the allocated time, and red light will blink at the end of the allotted time. So with this, I now hand over the session to respected chairperson, and I would also like to request the dignitaries so that we can occupy the front row for the ease of convenience, sir.
Thank you, Monica. Thank the organizers for inviting me. As the program looks, it's very challenging. It looks time frame, 3 minutes, 4 minutes, 8 minutes. It's kind of very tight. But I think most of you are senior people, senior professors who are in this system for quite long, and manage your time. And more importantly, I see this danger light, red light, green light. So obviously that will warn you. So with that, without taking much time, few comments I would like to make at the end. But right now, we can perhaps start with the presentations. So first is, I think, agricultural engineering. So
if it is convenient, we can see that is what we have been doing.
Yeah, it's okay. Every time going up and down, we can see the slides.
Otherwise, that board is there.
We are okay. Because there also it is there. No?
Yeah, there also it is.
Oh, then why? That is meant for this only.
Okay.
So please go ahead. Yes, sure.
No.
Yeah, eyesight.
Very good afternoon to all present in this august house. I extend a very cordial and hearty welcome to all of you on the occasion of 63rd Convocation of IRI, the third day and third session. From the School of Natural Resource Management, I will be starting the presentation, and I am Professor of Division of Agriculture Engineering. I am A.K. Mishra. Honorable Chairman, Sir, Dr. V. Venkateswarluji, Honorable Vice Chancellor, Parvani University, and also ex-Director of Krida, respected Madam Dean and Joint Director Education, Madam Anupama Singhji, Coordinator of this session, Madam Joshi and Madam Shrutiji, Honorable School Coordinator, Dr. Brahmananji, and the Project Director WTC, all respected Head of the Divisions and esteemed Professor colleagues, all my student friends and persons from the media. This is a pleasure welcoming you this morning. As I have already introduced myself, so I will skip this slide. The Agricultural Engineering Division has got 3 major disciplines: farm machinery and power engineering, processing and food engineering, and soil and water conservation engineering. We have got a sanctioned strength of 24 scientists, against which we are now currently 13. So around 50% of us are really vacant. So and also 3 years back we started a graduate program, BTech in Agricultural Engineering, and as per BSMA guidelines, for full faculty strength we need 123 faculty members, and here we are running the program by drawing the faculty from other sister disciplines and sister institutions. So here I request to authorities that our strength should be compensated so that we can successfully run our programs. As on today, we have got 61 BTech students, 18 MTech and 59 PhD scholars at Division of Agriculture Engineering at IRI. At CIA Bhopal, we have got 6 MTech and 18 PhD scholars, At Niasam Baramati, we have got 2 MTech and 1 at ICAR RECR Patna. This convocation, we are going to impart degrees to 7 MTech and 6 PhDs from IRI New Delhi, 8 PhDs from CIE Bhopal at NISM Baramati, 2 and 1 MTech at, from ICAR RCR Patna. So, starting the theme-wise presentation, and the farm machinery sub-discipline has got the most important theme of precision farm equipments. And here we have got our student Mr. Pradeep Kumar, and who has worked with Dr. H.L. Kushwaha, and he has developed and designed an embedded system-controlled site-specific seed cum fertilizer applicators. You all know that the commercial seed drills are costing more than ₹1 lakh, and all farmers are not in a position to purchase, and that results into the delay in seed, sowing operations, and sowing operations are really costing a lot as far as the produce and production of the commodities are concerned. So one very interesting machine was developed by this student, where the microcontroller and hopper has been designed, and this is the machine design of this particular machine, and the performance was evaluated. It was found that the precision index could be as high as 93%. With the D2 diameter of 13 millimeter and a speed of 2 km per hour. Also, the germination percentage was very high, up to 94% at optimized speed of 1.5 km per hour. The cost benefit ratio of this machine is very high at 2.2%, and also the machine cost only 11,000 rupees, a meager one, and you can see that the operation cost of the machine is less than 1,000 rupees. So you can imagine millions of the farmers, they can be benefited using this retrofitable and scalable technology. The second student, Mr. Munde Arjun Naik, worked with Dr. Adarsh Kumar to develop a robotic pesticide applicator for greenhouses. You know that greenhouses are fast upcoming for high value cash crops, and this robotic system has been developed. It can detect the diseased plant and also design the amount of the chemicals that has to be sprayed, and also this can work in horizontal mode. So the machine was tested for its performance, and performance was found to be very high, and also the theoretical field capacity was very high, up to 0.2 hectares per hour in the greenhouse conditions. So this is going to ease out the problem of the greenhouse operators and for high value commercial crops. Now one point has to be emphasized here, that the cost of the robot is only 1.2 lakhs as compared to, of course the other robots are also not available for greenhouse cultivation, but the cost is very meager, and the break-even point can be only 3.5 years. And the operation cost of this machine is less than 1,000, only 665 rupees per hectare. Our gold medal aspirant, Mr. Manojit Chaudhary, designed and developed crop canopy reflectance-based real-time variable rate fertilizer application system. He indigenously designed the sensors which can sense NDVI, NDRA and RVI. As compared to the traditional sensing devices, this is a very cheap intervention that has been designed by the student. A vehicle for carrying this particular sensor was designed. The machine was tested in the DSR rice, and this particular machine can assess very precisely the nitrogen availability in the crop up to 96% to 100%, and accuracy up to 98%. The machine was optimized, and the final result that show that in DSR this nitrogen use efficiency could be increased up to 53%, and yield could be increased up to 12%, cost 25%, and fertilizer saving 28%. Associated with are many other environmental benefits. The student has applied for a patent. He has got the technology certificate. He has published extensively in very high rated journals, and also a lot of people are after taking this particular technology for commercial uses, and he has presented this work for gold medal. And I think other students who are aspiring for gold medal, they have to think something beyond that, so that they can seek or get a gold medal from IRI authorities. This student from Bhopal, Mr. Abhishek Upadhyay, worked with Dr. Narendra Singh Chandel, developed computer vision based disease management system for tomato crop. Now this is the spraying control system that was designed. This machine was tested for performance, and this performance was rated as very high as precision recall, and F1 score was quite close to 90%. The pesticide and water solution that was being applied traditionally could be reduced to 1/3 to 1/4, reaching to 257 liters per hectare with this machine. The take-home message is that this is a very highly economic intervention that was designed to operate at 82 rupees per hour cost, and operation cost per hectare is less than 1,000 rupees. And the saving of the agrochemical could be more than 40%. So wastage was reduced for different diseases that was tested, and it could be increased substantially. For pomegranate crop, the student Deepak Thorat, working with Dr. C.R. Mehta, the worthy director of CIA Bhopal, he developed a canopy volume based variable rate spraying system for pomegranate orchard, which was tested for 2, 4 and 8 years old pomegranate orchards. This is the variable rate spraying system that was designed. Electronics, lot of electronics has gone into this one. Response surface methodology was adopted for testing the machine. The performance was high. Field performance at optimized parameter combined resulted at VMD as high as 320 micrometers, spray coverage of up to 41%, droplet density up to 85 droplets per centimeter square, and spray deposit reduced to 2.4 microliter per centimeter square. The developed variable rate sprayer reduced chemical uses by 46%. And as compared to conventional air blast sprayers, and also 34% as compared to the same sprayer without variable rate function. So this is a very good machine that has come out of this research work. Farm machinery and power engineering. The second thematic area is resource conservation and technology. Our student, Mr. Dera Vikas Reddy, working with Dr. Rauf Ahmed Pare, and he has developed a non-destructive adulteration detection protocol for saffron using advanced imaging techniques. We all know that saffron is very costly commodity, even costlier than gold as far as the weight of this one is concerned. It is very, I think, prone to adulteration, and it is very difficult to find out what are the different adulteration material or adulterated samples. It is very difficult to detect. So this particular research work has used total as content, color values and aprocotinoids as the dependent variables for finding out the adulteration. The machine learning ensembles were used, 5 of them were there, and advanced imaging techniques using hyperspectral were used and validated here. So the time taken by the most appropriate ensemble, MobileNet V2, demonstrated superior performance and with only 37 milliseconds. So it is quite possible that with this particular strategy or the technology that has been developed by the student, the saffron adulteration can be immediately detected, and many people are after this research. The government of J&K and many other private vendors are after this, but the team is going for a patenting and other formalities before really giving it to the exact users. So Mr. Omkar Sunil Karale, an MTech student working with Dr. Murtaza Hassan, he has worked on AI-based nutrition management modeling for vertical hydroponic systems. So this is his greenhouse where he has worked. This is the methodology that was followed. This is the vertical hydroponic system where the student has worked. He has recorded all the parameters of the pak choy crop for the entire growing season. I am showing only one result here, and that was validated for all 7 machine learning ensembles and techniques. So the student reported that the KNN model was the most superior as far as the efficacy in forecasting the impact of nutrient dynamics is concerned on all plant growth parameters, yielding exceptional accuracy and minimum error margins. Shilpa worked with Dr. Murtaza Hassan once again on AI-based crop growth modeling in vertical hydroponic systems. These are her experimental setup details. 8 vertical hydroponic structures were used. 2 recirculating system was about 100 liters per second. Nutrient solutions were 2, and one among these is, I think I would proudly say that one scientist sitting among us, Dr. Hassan, has developed this nutrient solution. Hassan's et al. N2, it is called as 2 supporting media and 2 structures, A frame and S flatbed they were used. Advanced instruments were used to record the data. This is the lettuce crop and vertical hydroponic system. Now the point is why this particular type of machine learning based, AI based ensembles were required is because that all other crop growth models, they require the soil data. Here hydroponics, we do not have soil, so this was required to develop. And the student reported that random forest emerged as the most accurate model to predict all crop growth parameters of this particular experiment. See, the third thematic area is ergonomics, and the student Sajja Pujit has worked with Dr. Adarsh Kumar, and the day before yesterday he has presented his result, and he worked on ergonomic assessment of power cylindrical lawn mowers for enhanced operators' comfort. We all know that lawn mower operation is very, very tedious, and it creates a lot of noise and vibration, and those are resultant to the psychological, physiological and health hazards, occupational health hazard of the operators. Sometimes noise goes as high as 95 to 100 decibels and vibration as high as 23 meters per second square. So a retrofitable acoustic enclosure was designed, and also few sheets added interventions were designed and retrofitted on this machine, and it was again tested and found that the noise could reduce substantially up to 80 decibel, so as the vibration that has reduced to 9 to 10 meters per second square. So the take home message of this is that the hand blanching can be increased for the time for hand blanching can be increased for 2.5 times or even more. The operator's comfort can be increased. The operator can work for more longer period without losing either the hearing ability or heartbeat or any other problem with this retrofitted system. The student Manisha Jagdale, working with Dr. K. N. Agrawal at CIA Bhopal, studied the cranking operation for women workers. You know that so many women are working, and they are again used in many strenuous activities. These are the points where it hurts most: neck, ankle, trunk, knees, elbow, and the wrist, as well as the shoulder. So all these forces were measured using load cell, EMG and load cell meters recorded. The force diagram was done, and the performance evaluation of this whole system was done. These reports are that Python based angle measurement system can be used as an alternative to digital geonomic methods, and 6 muscles groups they can be eased out by designing and optimizing the cranking system to 910 millimeter cranking height, 249 millimeter cranking length, and 38 watt power. That gives maximum or minimum physiological response to the operator, especially women. The second area of our specialization is processing and food engineering. We have a value addition of agriculture produce as theme 4. Engineer Monica worked with Dr. Mahapatra towards infrared pre-treatment for milling quality improvement of kodo millet, and you all know that it's a very good millet having very high protein and fiber contents, but the milling quality is very poor. So IR applicator was used here. Many other treatments were there. DM simulation was there to design this fabrication of infrared applicators. Infrared applicators were done, and then the whole device was tested. Dehusking efficiency increased to 6%, and also the head grain yield to 7%, etc. And increased protein, fiber and ash contents are reported here, and the moisture reduction was also noticed. Phenol, flavonoids and antioxidants increased substantially, and also the cooking properties was increased, crystallinity was increased. The second student, Ravi Kumar Sahu from CIA Bhopal, he has worked on conversion of chickpea stalk residue into supercapacitor, and he has used this chickpea using pyrolysis with KOH. He created these electrodes, and that you all know that capacitors can store the charge. The machine was tested, the nanoporous carbon CT3 prepared at 750 degrees Celsius with 90 minutes had a high specific capacitance, high energy and high specific area and pore volume also. So the take home message from this particular research is that uselessly burning the biomass waste can be made profitable if they can go for making capacitors out of these stalks. Here he has used chickpea stalks, we can use pigeon pea stalks or mustard stalks or any other organic bioproduct of agriculture. The second thematic area of our processing and food engineering was quality storage of agriculture produce. Mr. Ajay Mudgal has developed a hot air dryer coupled with biomass fired steam heater. So there are certain commodities which are having only regions, they evaporate when dried normally. So a dryer was developed which uses steam, and this is the inside view of the dryer. Overall dryer efficiency was 42%, boiler thermal efficiency achieved was 33%. Curry leaves were dried from a moisture content of 93 to 10.5% within a short span of only 190 minutes. And this particular dryer was at par with the traditional electric dryer. Now what is the advantage of this? It can save a lot of electricity bills to the farmers. Why? Because it uses the biomass that can be used for heating up the boiling the water. Bagola Paravallika, working with Dr. Saroj Kumar Giri, she worked on stabilization of rice bran with infrared heating of brown rice. We all are now eating rice bran oil. So she developed this particular IR heating treatment system in which she treated the brown rice and gone for bran removal, and this is the removed bran. This has got high storage properties, more than 7 weeks, and minimum free fatty acids are reported. They do not increase when stored for long duration. The only 60 seconds of heating time is required for maximum heading rice yield up to 91%, and other benefits associated with this. The third area of specialization of agri engineering is soil and water conservation engineering. We have got students working on precision irrigation. My student Vijan Majumdar tried to develop depth integrated type soil moisture monitoring system, and that can schedule the irrigation on real time basis. And also he tried that the data could be transmitted immediately to the user's mobile or computer through internet or satellite uplinking. So these are the electronic circuit diagram that has been used. Capacitance sensor was used. This is display device. This is the machine. It has got one small storage where all the electronics components are housed. This is the report of one particular data that was collected. The machine was tested in different soil and depth combinations. Systems integration with mobile alerts and automated monitoring improves irrigation management, conserve water and reduce labor for farmers. That is the outcome of this result. The student Ms. Arati Kumari worked with Dr. D.K. Singh, and she has developed a comprehensive system. It had sensing device for irrigation scheduling once again, but this device had soil moisture sensor, infrared sensor, temperature and humidity sensors. So the device was tested in 4 seasons in 4 crops: net house maize, open field wheat and capsicum grown in greenhouses. And the performance was at par with the traditional TDR and handheld infrared thermometers. So the device was very, very good, and the device was a low cost one. So she has also recommended the crop water stress index for all 4 crops he has experimented with, and these are the values given for irrigation scheduling. This device can be popularized very nicely, very easily among the farmers. Saranya has worked with Dr. Wang Chure in NISM Baramati. So to alleviate the water stress in garlic crop through deficit irrigation and plant growth regulators in Deccan Plateau region. She used 7 plant growth regulators, and now you can see the result. This is her experimental plot. It is very well known fact that the stress can reduce the yields, but PGRs associated with the irrigation scheduling like IC, NU, NSW are very effective when the moisture stress is slightly low to medium conditions in Deccan Plateau. This can be translated in entire Marathwada region also. Subha Madhual working with Dr. Nangre in the mango orchards. Again he has gone for deficit drip irrigation system clubbed with plant regulators, PGRs, and I think these results are to be assimilated by our horticulture groups, that partial root zone drying 50% plus SA plus Na boosted antioxidant activity, phenolic and flavonoid contents along with 40% rise in proline accumulation. So this particular research result is applicable to the mango orchard growers. Student Abhishek Patel, working with Dr. Ramana Rao on Upper Tapi catchment, developed suitable rainwater harvesting zones for the Upper Tapi catchment using hydrological modeling of the system, and he has also gone for the assessment of the long-term impact of climate change and given the values for Kharif and Rabi crop water requirements of major crops of that region.
Who is maintaining the time? I mean, are you monitoring?
Yes, okay. Yes. Thank you. So, Mr. Vikas Mangilal, working with Dr. D.K. Singh with Eco Crop model, has shown that water productivity of wheat could be increased substantially, and result showed that the precise irrigation scheduling aided by real-time soil moisture monitoring can lead to substantial water saving without any yield penalty. Pratyusha Misra, working with Dr. Santosh at Ranchi Center from ICAR Eastern Region, she has worked with Hydus 1D model towards modeling water and nutrient dynamics of mustard crops, and with deficit irrigation, she again reported that nutrient productivity was high at 80% irrigation treatment, and also the water productivity could be raised. Uma Shankar, working with Dr. K.V. Ramana Rao, developed a hydrogel applicator machine and tested the machine and proved that the machine developed was having very high capacity, 0.37 hectare per hour. The effective field capacity was as high as 77%, and he has also worked out that shaft placement depth should be 10 centimeter, that can result a very good yield for spinach crop. These are the total publications coming out of the research work of scientists and students. I think 1/3 of the publications are from the students' work, and you can see that 10 and above we have got 21 publications, 17, 8 to 10, 6 to 8 we are having 14 publications, less than 623. Few important publications. These are the publications shown by our gold medal aspirant. You can see once again, these are very high rated journal having very NAS rating, very high NAS rating, and one has been accepted in a NAS rating of 12.7. Intellectual property right, he has applied for a patent, and so as many other students who are developing machines and all. We have got a registered copyright, a design patent, and also people are after us asking for the technology to be given to them. Student has attended a lot of training and workshop and got certificates of appreciation. And also we have demonstrated all our technologies in G20 spouse visit, Indian Mobile Congress, Kisan Mela, and everywhere. No student of us are unemployed as on today, and I acknowledge very sincerely the students, their chairpersons, members of their advisory committee, heads of the divisions, and faculty members of sister institutions, directors of all 4 institutions, project director WTC and school coordinator NRM, dean and joint director education, joint director research and extension, collaborators, technicians and workers, funding agencies like ICAR, DST Industry and AICT, and many others, unknown reviewers and examiners. And thank you, Chairman Sir, for giving us a very patient listening and the time. Thank you very much.
Good presentation as indicated by the applause from the audience. But I think there is no scope for discussion because time is, but time is so tight that really, I mean, otherwise there would have been a lot of inputs from the hall also. But anyway, so with that we move on to, thank you, Dr.
Thank you for
the presentation. And you must really thank your partners in Niyasam and Bhopal and all. You have thanked them, but always.
Yeah, thank you.
So, agriculture physics, Dr. Krishnan, Professor Krishnan.
Good afternoon. Good afternoon. Respected expert of today's session, Dr. Venkateshwarluji, my senior and junior colleagues, and my dear friends. Today I am here in front of you to present the significant educational achievement in the discipline of agricultural physics. Agriculture physics is a small division. We have got about 11 faculty, and during the year 2024, we have 6 students receiving the MSc and 3 PhD students will be receiving their degree. And the whole presentation is divided into 4 different themes. Coming to the theme 1 on drone technology and remote sensing, the student Supriyo Dara worked with the chairperson Dr. Rajiv Ranjan, and in his study he has developed a simplified crop water stress index, and he has monitored it in different irrigation and nitrogen stress conditions using drone-based monitoring with multispectral and also the thermal cameras. And Mr. Supriyo has clearly shown that the drone-based measurement of temperature was very much in correlation with the actual temperature measured with the infrared thermometry, and also his simplified water stress index which he has developed has got very good correlation with the stress indices like RWC and also stomatal conductance. The next student, Ms. Abita, has worked with Dr. Rajiv Ranjan. She has evaluated the drone-based and knapsack-based sprayer for spraying nano urea, prilled urea, and nano DAP, and she has got 16 different combination of treatments in her study. And Abita has very clearly shown that drone-based spray has got a lot of advantages compared to the traditional knapsack sprayer. And she also quantified the crop condition in terms of the indices like NDVI and NDRE and LCI, and she has very clearly shown that when the crop is sprayed with drone-based sprayers, the condition of the crop is far better than that of the knapsack-based spray. The next student, Sunny Arya, has worked on the artificial intelligent high throughput phenotyping of wheat with Dr. R.N. Sahu. Sunny Arya has worked on the chlorophyll fluorescence, which he has attempted for the very first time for characterizing the nitrogen and moisture stress in wheat in both under controlled and also in the open field conditions. And he has used many RALs. As you all know, RALs are very difficult in segregating the traits among them, and based on the chlorophyll fluorescence Fe by FM images, and he could classify the RALs into different categories, and based on PCA he could, and also the yield indices, he had categorically classified the nitrogen stress into 5 different classes based on their stress tolerance and also the yielding abilities. Not only that, he has developed a machine learning and deep learning based model for differentiating the traits of these RALs related to water and nitrogen stress. In his study with the machine learning approaches, he has identified that the gradient booster regressor was the best methodology to predict or simulate the yield in wheat crop. And from the QTL analysis based on the interval mapping, he has used the ICI mapping software, and based on the SNP and the phenotypic data, he is very meticulously able to position the QTLs in the chromosomes, especially 12, 15, and 11, and 21 for nitrogen stress. Similar work he has done for the water stress also. The next theme on biophysics and nanotechnology, the student Preetham Chanak has worked with Dr. Monica Kundu. In his study, Mr. Chanak has used different soil samples collected from in and around IRI, and he has found them to be highly contaminated with heavy metals. So he has developed an integrated electrochemical system for removing these heavy metals in the contaminated water. Based on the response surface methodology, he could identify the maximum efficiency in terms of the time and also the voltage and other parameters of the system he has developed. He has applied the central composite design fitted model for characterizing the removal efficiency in cadmium, zinc, and cobalt and manganese. And based on that, from the collected samples, he could very clearly verify that the efficiency of removal of his system was to the tune of about 80%. The next student, Shreya Sharma, who has worked with Dr. Achelal Yadav, and she has synthesized the zinc oxide nanoparticles of different sizes of 20 and 30 nanometers of different concentration of 40 and 60 ppm, and she has compared it with the traditional zinc sulfate application under the same concentration of 40 and 60 ppm. She has used the green synthesis of these zinc oxide nanoparticles. She has synthesized it from the eucalyptus and also the Arjuna leaves, and based on her synthesis, she has also characterized the size particles, nanoparticle size using the TEM, and using the synthesized nanoparticles, she has very clearly shown that the relative growth rate, absolute growth rate, and net assimilation rate in treatments receiving the zinc oxide nanoparticles around 40 to 60 ppm with nanoparticle size of 20 nanometer were very performing good in terms of biochemical parameters and also in terms of the root parameters. And not only that, she has clearly shown that the biofortification of zinc is possible in the grains because it could increase the biofortification of zinc content in the grain from 30 milligram per kg to an extent of about 50 milligram per kg. The next student, Swagata Das, has worked with Dr. Ananta Vasish. She has very meticulously taken about 55 different magnetic field treatment of seeds in different duration of 30 and 60 minutes. And Swagata Das has characterized the germination percentage, speed germination, and also on the total dry weight and the vigor index. And she very clearly identified two of the treatments, especially those which are having 100 millitesla and 150 millitesla of exposure for 30 minutes, has performed well. And this she has taken it to the field to test their ability to tolerate the moisture stress conditions, and she has clearly shown that the seeds which are exposed to magnetic field could have better root length and root surface area and also root volume under the moisture stress condition. Not only that, the treated seeds could have about 10% advantage of increased yield under field conditions. The next team area on agriculture, meteorology and crop simulation modeling. Madan Mohan Meena has worked with Dr. Joydeep Mukherjee, and he has characterized the effects of high temperature and nitrogen stress on 6 different rice cultivars. And he has meticulously monitored the different parameters like LAI, total dry matter, root length density, and also on photosynthesis rate, root mass density, and radiation use efficiency. And he has characterized the yield changes in the rice crop under moisture, under nitrogen stress and elevated temperature. He has observed from his study that cultivars which are having long duration seems to be highly susceptible to moisture stress and nitrogen stress and compared to those which are of short duration. He has meticulously incorporated the parameters like cultivar parameters, weather parameter and soil parameter infocrop model, and he could find that the model could have the efficiency of simulating the total dry matter and grain yield with a minimum error of 10%. The next theme on soil physics and geoinformatics, the student Animesh Panda has worked with Dr. Pragati Maiti. He has meticulously collected about 150 literatures of published literatures on CA treatments and CT treatments in and on the rice wheat conservation agriculture system. He has collected about almost 4,248 points of 26 different soil parameters based on his, he has also characterize the treatments from the long-term experiments which are conducted in IRA under the CA, and based on all these things he has collected the parameters on physical properties, chemical and also biological parameters, and he had applied the machine learning methodologies for predicting the yield and identified the ANN to be the best strategy for predicting the crop yield using these parameters. Not only this, he has applied the principal component analysis and generated the soil quality index both for the global data and also for the localized field experiment, and found that the agreement between them to the tune of about R square of 0.896. The next student, Ranjita Mandal, has worked on the quantification mapping of soil ecosystem services under the guidance of Dr. Pragati Pramanik Maiti, and she has worked on the Kannal and Kaital district where the conservation agriculture has been practiced intensively. And she has quantified the ecosystem services, and based on that she has made the map of provisional services based on grain yield and byproducts, and also on the regulating services and on the supporting services. In addition, she has also calculated the cultural services and also the, what is happening?
It's okay.
Okay, ecosystems services, and based on this she has calculated the total ecosystem services. And in the study region she could very meticulously classify the system into 5 different classes, and she could identify about 67 villages in the study region to be under the degraded classes. Not only this, she has developed a web-based ecosystem service calculator which can be used by others for their calculating the ecosystem services. Though the division is very small, we have about 21 publications during the year 2024. I am very proud to say that we could reach the publication of even 18 NAS rating, and the students' contribution in the publication is more than 67%, and more than 50% of the publications are in NAS rating of 9 and above. We have conducted 4 different training programs, and the students have also actively participated in the students' tour. And I am very proud to say that 2 of our students, Selva Prakash, has availed the SERB Overseas Visiting Doctoral Fellowship, and he has got about 18 months training in Purdue University, USA. And he has come back. And next student, Mr. Abradeep Sarkar, has availed a similar fellowship. He is now in Purdue University and will be undergoing a similar training for 12 months. And coming to the honors and awards, last year our student Ram Narayan Singh has received the IRA Merit Medal and also the K.N. Singhals medal from the Soil Science, and also the SSRA Best Thesis Award from the Agrophysics Society, and also PD Mystery Award from the Association of Agrometeorology. Apart from this, our students have got Best Poster and also the Best Student Award from different societies and conferences. Our faculty has received the scholarship from International Soil and Tillage Research Organization and also has received the Best Academician Award during the year 2024. Our students are not only dependent on ICAR and IRA fellowship, they have been bestowed with fellowship from DST, NFSC and UGC. Almost 1/3 of our students are getting fellowship from other funding agencies. I'm very proud to say that the students' placement of this division has been very good. Out of the 6 MSc students who have passed out, all of them have joined back into PhD, and 3 of our students could get selected in the ARS 2023. Thank you very much for your patient listening.
Okay, Professor
Thank you.
Krishnan, for being on time and also sharing the valuable work of your students. So we move on to environmental sciences. Here it is, list is environmental sciences or agronomy, sorry, I'm sorry. So Dr. Shiva. Yours is a big division, but you are given less time, I don't know. He has less students or what? Maybe student-wise. Student-wise, carry on.
Very good afternoon. Namaskar, one and all. Honorable Chairman of this session, Dr. B. Venkateshwarluji. I see Dr. Anand Kumarji, P. Anand Kumarji, former Director of National Institute of Plant Biotechnology. Coordinator of this session, Monica Joshi ji, Dr. Shruti Ma'am, Coordinator of the NRM School, Dr. P. Brahmanand ji, Head of the divisions, professors, students, ladies and gentlemen. I am before you to present the significant achievement carried out in the division of agronomy during last 1 year. In the division, we are having 24 scientists, including 14 principal scientists, 6 senior scientists, and 4 scientists. Sorry. क्या हाथ में रखना है? And we are also having a good strength of students. 71 total students are there, including MSc and PhD. And so far, 1,262 have graduated, including associateship from the Division of Agronomy. And if you see this year students, those who are going to receive their degrees, there are only 18 students. 9 for MSc, 9 for PhD, and out of these 18, 3 are from the IRA Assam, and rest of them from the IRA New Delhi. Coming to the students, those who are going to receive their degree in the MSc program for this convocation, I have enlisted the student photograph and name, roll number, then theme area, title of the thesis and chairperson in this list. Coming to the theme-wise significant achievement of the student. Our students for the MSc, they have worked under the broader area of 3 themes like crop diversification, nutrient management, weed management. Coming to the first student, Astika Pandey, she worked under the supervision of Dr. Subhash Babu on effect of land configuration and cereal legume integration on system productivity, resource use efficiency, and soil health. The major outcome of her thesis are integration of the cereal legume systems resulted a 2 to 3-fold increase in the system productivity, and also carbon sequestration 2.2 to 3 times, and reducing the carbon footprint by 2.5 to 3 times across all scale compared to the maize wheat system. Ms. Rimjam Chaudhary worked under the supervision of Dr. Y.V. Singh on integrated nutrient management in baby corn intercropping system. The major outcome of her thesis is INM resulted in the highest co-yield, whereas the baby corn plus cabbage intercropping system under INM achieved the highest baby corn equality, that is more than 10 ton per hectare. And Mr. Navneet Kumar worked under the supervision of Dr. Ishwar Singh on response of wheat to chemical fertilizers under FIM and also biofertilizers integration. The major outcome of his thesis are the highest grain yield was achieved with the combination of FIM at the rate of 5 ton with biofertilizers and 75% recommended dose of chemical fertilizers. This combination of integrated nutrient management is the best option for sustaining the wheat productivity. Mr. Subham Yadvanshi worked under the supervision of Dr. S.S. Rathore on biochar and solid digest effect on productivity and nutrient use efficiency in mung bean under mung bean wheat system. And the major outcome of his thesis were maximum net gain in terms of available nitrogen and phosphorus was recorded with 100% RDA plus solid digest at the rate of 2 ton per hectare. However, the maximum available K was recorded with 100% RDA plus biochar at the rate of 3 ton per hectare. It is because of that their nutrient composition. Hence, it can be recommended as a nutrient source in the targeted areas wherever these materials are available. Coming to the next student, Mr. Chandrakant Yadav, he worked under the supervision of Dr. Kapla Shekhawat on nutrient management in finger millet under alluvial soils of Assam. The major outcome of his thesis were around 33 and 19% grain yield enhancement was recorded with the application of FIM at the rate of 5 ton along with the biofertilizers as compared to the other treatment, and around 41 and 34% increase in the grain yield was recorded with the application of 60 and 80 kg nitrogen respectively over control. Ms. Sayantika Sarkar worked under the supervision of Dr. Praveen Kumar Upadhyay on precision nitrogen management in maize using Android-based mobile application of nitrogen. The major outcome of her thesis are split application of nitrogen through Android-based push or nitrogen doctor app at the rate after 35 and 45 days after sowing of the crop was the most appropriate, and the highest agronomic efficiency in terms of nitrogen agronomic partial factor productivity and apparent recovery was recorded with the lower dose, that is N 50% along with PK and F-based treatment. Coming to the Sneha Bhardwaj, yesterday she presented a merit medal and she got a gold medal overall. I am really happy, and then I must congratulate her. She worked under the supervision of Dr. Parihar on analyzing the yield gap of rice in hilly ecosystem using biophysical modeling for different nitrogen levels. And the outcome of her thesis are due to the limited nitrogen application, the yield gap was 2.8 ton per hectare and 3.1 ton per hectare for this because of low nitrogen application in that area, and for Sasrang 1.6 ton per hectare and 1.76 ton per hectare for Mandri under rain-fed and irrigated conditions respectively. And during drought years, supplemental irrigation increased the yield by 11 to 17% for Sasrang and 5.5 to 11% for Mandri varieties of rice over rain-fed conditions. Mr. Huchappa Jamakandi worked under the supervision of Dr. Shivdhar on response of maize to the nutrient sources in Assam soils, and the major outcome of his thesis are among the nutrient management, integrated nutrient management, including nitrogen, phosphorus, potash, along with the 300 kg lime per hectare resulted in the highest productivity compared to the other treatment. Last student for the MSc degree program work on weed management under the supervision of Dr. Rajveer Sharma, Mr. Hamesh Kumar Meena on integrated weed management in lentil. And the major outcome of his thesis are weed management practices increased lentil yield by 36.6% to 93% compared to the VD check. And the application of the pendimethalin at the rate of 750 gram per hectare is a pre-emergence plus one hand weeding at 35 days after sowing resulted in the highest grain yield, followed by pendimethalin plus maize straw mulch at the rate of 4 ton per hectare. Coming to the theme-wise significant achievement of the PhD student, our PhD students, they have worked under 5 major themes, including conservation, agriculture, crop diversification, irrigation, nutrient management, and weed management. So again, I have enlisted the student, you know, that name, then roll number, theme area, title, and their chairperson. Coming to the outcome under conservation agriculture, Mr. Akshay Galotra worked on residue, tillage residue and nutrient management for enhancing productivity, profitability, resource use efficiency in maize wheat system. The major outcome of his thesis are zero tillage plus residue increased productivity, resource utilization efficiency as compared to the conventional tillage, and zero tillage. And further, you know that nitrogen management, subsurface application of the nitrogen management led to the 20% saving of the nitrogen under this study. Coming to the another student, Ms. Gunturi Alakya, she worked under the supervision of Dr. Professor T.K. Das on cotton wheat system under conservation agriculture, and the major outcome of her thesis were permanent broadbed residue with 100% nitrogen practice gave higher system productivity, system net returns. However, the permanent broadbed residue with 75% nitrogen treatment were comparable with the 100% in most of the parameters, and it indicates that 25% nitrogen saving can be done by this. And further, this CA-based practice led to, you know, that reduction in the greenhouse gas emissions, greenhouse gas intensity, and as compared to the conventional tillage. Coming to the crop diversification theme, Ms. Priyanka Saha again work under the supervision of Professor T.K. Das on pigeon pea wheat system under different production scenario, that is conservation agriculture and organic production system. And the major highlights of her thesis outcome are approximately 6.2, 8.7 and 12.9 times higher carbon sequestration rate was observed under CA conservation agriculture based permanent furrow bed over conventional tillage. Further, 38% reduction of the field scale carbon footprints under conservation agriculture over conventional tillage. However, organic farming showed approximately 64% more field scale carbon footprint over conservation agriculture. Coming to the next student, Mr. Arjun Singh, he worked under the supervision of Dr. Anshul Das on nitrogen and crop residue management in maize under subsurface drip fertigated maize wheat green gram cropping system. The major outcome of his thesis are 100% recommended dose of nitrogen in 4 splits, resulted in highest grain yield productivity. However, it was statistically at par with the 75% nitrogen recommended dose of nitrogen in 4 splits. And further, these varying nitrogen treatments increase yield by 55%, 53% and 55% again over control, and 16.6 and 13.5% and 15.6% over conventional method. Coming to the irrigation theme, in this Mr. Srikant Reddy, yesterday he presented for merit medal, but he could not make final. Although he has a lot of publications and other, you know, that technology development by his study being carried out at the, you know, that IRRI hub Varanasi. He worked under the supervision of Dr. C.M. Parihar on productivity and resource budgeting of rice-based production under different irrigation methods and nitrogen management. The major outcome of his thesis is integration of conservation agriculture with subsurface drip fertigation performed superior in both rice-based cropping systems with 10 to 15% higher productivity, 34 to 45% higher profitability. And further, he, that advocated that rice-based system recorded higher, approximately 10.8% system productivity, and he highlighted that winter maize cultivation can be promoted in the eastern India based on his findings. Coming to the nutrient management theme, in this theme, Mr. Madam Vikram Arjun worked under the supervision of Dr. S.L. Meena on moisture management and also zinc fertilization. And the major outcome of his thesis are hydrogel at the rate of 5 kg per hectare resulted increase in productivity of palm millet and mustard, and also zinc concentration in the seed. And among the zinc fertilization treatment, he reported that soil application at the rate of 12.5 kg zinc sulfate per hectare plus foliar spray of 0.5% at flowering maximum vegetative growth recorded highest productivity in terms of the grain and seed yield of palm millet and mustard, and also zinc concentration in both the economic part. Coming to the K.S. Reddy, he worked under my supervision on performance of nitrogen sources and zinc fertilization on productivity and nutritional quality of wheat rice system or sequence. So he highlighted that recommended dose of nitrogen is important for sustaining the wheat rice sequence, and when we decrease the nitrogen in conjunction with the nano urea, there was significant reduction in the productivity of both the crops and even also benefit cost ratio. And among the zinc fertilization, 0.1% foliar spray of nano zinc oxide was better in increasing the productivity and also nutritional quality of both the crops. Coming to the Ashok Kumar, he worked under the supervision of Dr. K.S. Rana on moisture management and also potassium management. Among the crop residue management, he highlighted that the crop residue at the rate of 3 tons per hectare plus Pusa decomposer. As you know that one of the technology of ICAR is the better for increasing the productivity of palm millet and also wheat. And among the potassium management, 100% recommended dose of potassium plus potassium solubilizing bacteria were better in terms of increasing the productivity of both the crops. Coming to the last student, G.D. Saniket, he worked under the supervision of Dr. Kapla Shekhawat on nitrogen management in rice wheat system under various crop establishment protocols, and he highlighted that highest wheat productivity was observed under zero tillage wheat with residue, which was 7.33% higher than conventional tillage without residue. And higher nitrous oxide emission were recorded under dry direct seeded plot in rice. However, wheat yield nitrous oxide emission were observed under zero tillage wheat plus residue. And he further highlighted that higher nitrous oxide emission were recorded with LCC guided nitrogen management in both the crops. Coming to the students' honors and awards, our students have won several honors and awards during last year. I am really happy to share that one of our students got International Fertilizer Association Emerging Scholar Award 2024, and also he was bestowed with the Young Scientist Award. Then Kamlesh Kumar R.P. Singh Memorial Award. Satyam Rawat last year got a merit medal of IARI, and also R.P. Singh Memorial Award, and then Dr. Rishiraj got Best Thesis Award last year. N. Sarkar Best Poster, then K.S. Reddy Best Poster, then K.S. Reddy again Best Researcher Scholar Award, Abhijit Mandal Best Oral Presentation, then Prakash Sonath Best Poster Award. Not only awards, but our students have bagged many, that scholarship other than IARI, and like Ran Moy Patra Fulbright and Fulbright Clam Fellows, Again, Ayan Sarkar, then Nitin Kumar served in ANRF, UGC Junior Research Fellowship, CSIR, some of the students, they have bagged. And then our faculty is not lagging behind than the student. They have also won awards, several awards and honors. Last year, Dr. R.S. Bana received HK Jain Young Scientist Award. Dr. Subhash Babu has been bestowed with the NAS Fellowship. Dr. Vijay Punia, NAS Associate, and CM Parihar, and I also got 1 or 2 awards during last year. Our division has organized 3 training programs, 2 led by Dr. S.S. Rathore and 1 led by Dr. S.L. Meena. Our students have published nearly 41 research articles. I am really happy to share that 14 articles out of the 41 were more than 10 NAS rating, and 6 to 10 NAS rating 19, and less than 6 NAS rating only 8. This is the list of the publications which I have been given here. I will not go through. Our placement of the student and getting that services. Out of the 8, 5 got selected from the Division of Agronomy in ARS. Few subject matter training, technical, agriculture at the department. Even one student has on ERI. With this, I would like to thank the Director IARI, Joint Director Education and Dean Madam, Joint Director Research, Joint Director Extension, Head of the Division, Dr. S.S. Rathore, Graduate School, Professor Das, UG, Scientist Faculty, Agronomy, and collaborating divisions, because this reflects not only agronomy but collaborating divisions' contribution in terms of the student thesis, students and other staff of the division. Thank you one and all.
So thank you, Dr. Shiva. I think his style of presentation is the best. Student name, thesis, title, and major. Very brief and very pointed. Both he summarized the student-wise achievements and theme-wise. I mean, that should be the real. Thank you, Dr. Shiva.
Thank you,
Honorable Chairman of the session, Honorable Dean and Joint Director Education, Dr. Anupma Ji, NRM Coordinator, Dr. Brahmanan Ji, PDWTC, Session Coordinator, and then heads, professors, faculty members, and dear students. I am here to present the salient academic achievement of 24-25 and in the 63rd Convocation. So the mandate of our division is, as you know, that India is ranked 5th most climate vulnerable nation, and then climate change performance index 10th position, and then SDG performance index 109th. So with these backgrounds, the mandate of our division is to provide basic and strategic research for environment resident agriculture, impart post-graduate education, and then provide advisory services on environment and climate change in agriculture. So ours is a multidisciplinary subject. We are having scientists, environmental science, agriculture, chemistry, economic botany, microbiology, soil science, agronomy, organic chemistry. The major thrust area is environmental microbiology, ecosystem services, remediation, environmental pollution, waste management, simulation modeling, and climate change. Since the inception of our division, 10th of January 1993, we have all the thrust areas and number of projects which we receive from different funding agencies. So I'm not going to detail. So we have different linkages, like international linkages, national linkages, farmers and industry, and different ministries. So as far as MSc is concerned, till now 118 students have graduated, and then PhD, 66 PhD degrees, and students on roll is 16 for MSc and 34 PhD. And then our students are placed in ARS, SAU, CPCB, State Pollution Control Board, FCA, DDA, different agencies, even abroad also. So the area of student research 24-25 is, as far as climate change is concerned, we are having this year, student will be getting degree 7 MSc and 4 PhD in climate change, and 2 MSc and 1 PhD in waste management, 1 MSc in ecosystem services, and 1 MSc in environmental pollution. So I am going to present with the PhD work first. So effectiveness, Ankita Paul, PhD student, she worked with Dr. Aarti Bhatia on effectiveness of modified urea fertilizers in reducing ammonia, nitrous oxide losses and increasing the nitrogen use efficiency in rice. So they have used different urea coating like sulfur coated urea, phosphogypsum coating urea, current coated urea and double inhibitors. In this they have used neem coated oil as well as limus. So this is the material method of their study. And the main findings are that nitrogen use efficiency increase with phosphogypsum urea, and then the major impact is as compared to the prill urea, double inhibitors reduces N2 emissions by 23.7%, methane by 11.9%, and ammonia volatilization by 29.3%. Overall, double inhibitors where they have used neem coated urea as well as limus. It lowers the global warming potential in the rice cultivation to the tune of 17.1%. So this particular treatment got a lot of potential in reducing GHG emissions from the rice field. Another student, Mr. Alesh Kumar, he worked with Dr. Manoj Srivastava under the phosphorus dynamics under different tillage and residue management practices in maize wheat cropping system. So the result shows that available phosphorus was recorded significantly higher under no-till and crop residue management treatment in 0 to 5 and 5 to 15 centimeter soil depth. Macro aggregates percentage were found higher under no-till and crop residue management treatment. Grain and biomass yield were recorded significantly higher under no-till and crop residue management, and nitrogen 150% recommended dose. So coming to the third PhD presentation, nitrogen dynamics, Mr. Sivanand Darji, he worked with Dr. Renu Singh on the nitrogen dynamics in natural farming system under rice wheat cropping system. This study was conducted in Gautam Budh Nagar at the farmers field in the close collaboration with the KVK near Gautam Budh Nagar district. So with the objective of assessment of nitrogen losses from rice wheat cropping system under natural farming, effect of Jivamrit, Ghan Bijamrit and soil microbial properties, growth and yield of the rice farming practices, and to quantify the cost benefit ratio of rice wheat cropping system under natural farming. So these are the treatment details, and they have modified natural farming also. So and the summary of this particular research is the natural farming inputs demonstrated significant reduction in nitrogen losses to the tune of 60 to 65%, and the natural farming practice lead to notable improvement in soil microbial population, 45% increase in bacterial population, 58% increase in fungal population, and 48% increase in actinomycetes population under rice crop. In wheat, 53% increase in bacteria, 62% in fungi, and 52% in actinomycetes population control. While natural farming inputs resulted in lower yields compared to the conventional, but the cost of the cost of cultivation was less. That's why the net returns were at par. So coming to the next PhD students, Helen Mary Rose, she worked with Dr. Nivita Jain on the life cycle assessment of maize and wheat products. So they have assessed the life cycle of wheat flour and maize flour. So in that they have quantified the GHG emission pre-farm, on-farm and post-farm activities. They have highlighted the what kind of emissions there in the form of input formation and the fertilizer formations, and they have system boundaries and different scenarios. Okay. So the major finding is that maize flour had higher global warming potential and freshwater eutrophication impacts on most of the scenarios compared to the wheat flour. They have taken 4 categories like climate change, eutrophication, freshwater as well as marine water, and acidification potential. The study highlighted that the conventional milling is more environmental friendly than the processing compared in industrial milling. So Shravani Shanial worked with Dr. Vidisha Chakravarti, field and simulation studies on the interaction of effect of elevated carbon dioxide and temperature on ST1 durum wheat. So they have assessed the durum and ST1 wheat, two durum wheat varieties and 2 ST1 varieties, the impact of carbon dioxide and CO2. And they reported that temperature rise by 2.5 degrees centigrade reduces yield by 11.2% in ST1 wheat and 7.2% in durum wheat as compared to the ambient treatment. Elaborate carbon dioxide concentration of 550 ppm was able to compensate yield reduction by 5.7% in ST1 and by 3% durum wheat varieties. protein content in wheat grain decrease in elevated carbon dioxide plus high temperature treatment in both Estem and durum varieties. So, simulating wheat yield and nitrogen uptake in wheat varieties using infocrop model. When the crop was exposed to high temperature of plus 2.5 degrees centigrade and elevated carbon dioxide, yield reduction in STM wheat varieties range from 8.4 to 16.7% in northwestern India and 7.1 to 8.9% in central India. In the case of durum wheat, and reduction of central India varied from 4.2 to 4.3. So, it means that durum wheat is more resistant than STM wheat varieties. So, coming to the list of MSc degree recipient, Ms. Reenu Verma, she worked with Dr. Ajay Kumar Singh, Niasam Baramati, and she screened large number of genotypes on the temperature stress tolerance, and with biochemical and physiological marker they could identify that varieties EC 39607 is the most tolerant varieties. And the other study, Ms. Anju Chaudhary, she worked with Dr. Anita Chaudhary in the impact assessment of microbial and non-microbial plant biostimulants for increasing the chickpea resistance in the dry spell. So, in chickpea dry spell is very, very common. So, they try to evaluate the trichoderma as well as humic acid. So, they found that when they are treating with this treatment, it can tolerate the dry spell. And then Mr. Arun Meena, he also worked with Dr. Anita Chaudhary, impact assessment of plant biostimulant and phenolic compounds for water stress condition in wheat crop. So, they demonstrated that the seaweed extract as well as the phenolic compounds, they could increase, they could enhance the tolerance to the temperature wheat crop. Oh, sorry.
I think you please summarize now. You not go into so much detail. Data is not required.
Okay.
This way.
Yeah. Okay. And then Mr. Asemul Hassan, he worked study of the. I'm not able to see. I'm not able to.
अब आ गए ना? अब आ गए ना?
Some blurring is there.
ये आपका चश्मा है क्या नहीं?
चश्मा
हाँ,
I forget my specs.
You are not able to see?
Okay, madam, we will take it as read, it's okay, because it is there in the book now. If he has is a health problem or?
No worries. Actually, we forgot this.
Okay.
I had thought.
I thought he has some.
All well?
Yeah. But I'll summarize.
It's okay. It's okay.
Yeah. So, Mr. Pawan Kalyan, we worked with Dr. Manoj Srivastava on Convocation and zeolite nanocomposites. So, they find that Convocation and zeolite nanocompound, they can increase the economic efficiency by 40 to 45%. And then Ashwani Yadav, she worked with Dr. Ashish Khatirwal on the problem of isolation characterization of biosurfactant and their impact on hydrocarbon degradation in different soil. Yesterday she presented for the gold medal and they have isolated some microbial strain, and in that particular thing study they have highlighted that how the microbial strain, the oil contaminated soil can be remedied with the help of microbial cultures. And then Mr. Rajdeep Das, he worked with Dr. Renu Singh on the preparation and performance of analysis of bioplastics synthesized using vitamin dioxide nanoparticles with corn cobs. So in that particular study, the student, they have made polymers from the corn cob, and the result of the product bioplastic of several ratios of cellulose, plastic hazard and nanoplastic using response surface methodology. Okay. And then Mr. Ramakrishna Panda, we work with Dr. Deepak Kumar Gupta from IRI Jharkhand, and they characterize the IRI Jharkhand soil carbon content, soil carbon study, the mapping and of variation of soil organic carbon stock within IRI Jharkhand. So they have done all the study, what is the carbon status of the IRI Jharkhand soil. And the last study, Mr. Aishwarya Lakshmi, she worked with Dr. Nimita Jain on mitigation, nitrogen and water footprints of tomato cultivation under different irrigation and nitrogen management practices. So they found that, am I audible? Sorry.
Yeah, it's okay.
So drip irrigation significantly reduced the nitrogen footprint.
Kindly let us hear to the Professor Environment Science, and let's maintain little silence.
Among nitrogen treatment, the farmer practice showed the highest nitrogen footprint, while the no and control had the lowest. The highest yield scaled nitrogen footprint was observed in the farmer's practice, with a significant reduction in treatment in the recommended dose of fertilizers. The drip irrigation significantly reduced water usage and water footprints compared to the ridge and furrow irrigation. Drip irrigation used 43% less water and exhibited lower green and blue water footprints. Sorry, there was one study also. Yeah. Sorry. Miss Anjali M. She worked with me on quantification of soil ecosystem services from coal, a coastal agrarian wetland in Kerala. In that study, we tried to quantify what kind of agro-ecosystem services provided by coal wetlands. Although coal wetland is just 6.5% of total Kerala rice agriculture area, but it is providing 20% of rice production from Kerala. So in that particular type of agro-ecosystem, we try to quantify what kind of ecosystem services is provided by the coal agro-ecosystem and what are the emerging threats in coal agro-ecosystem. So we find that lot of metals are coming from the nearby industrial sector as well as sewage. And the Nisha Khatri, she work on greenhouse gas and particulate matter emission from sugarcane residue burning using measurements and remote sensing. With Dr. Nimita Jain, this is a very important study where we are just focusing only rice biomass burning. But in western Uttar Pradesh and different part of the country, farmers are also burning different crop residues. Savli district, it is in NCR, Delhi NCR, they have observed 202 burning events were detected, and then burning of sugarcane residue resulted in the loss of 70.61 and 0.55% 0.55 kg 1000 tons of carbon and nitrogen. Developed, and this is by the help of this study, they have developed emission factor. A mapping of burn area in particular region can be used by policymaker to formulate different policies. Yes, sir. Yeah. So as far as student publication is concerned, our student has published 15 publications. And then as far as faculty publication is concerned, this year my colleagues have published more than 100 publications, and then the last year we have published only 70 publications. The 47% increase is there. Okay, so these are the copyright IPR generated in our division, and then the ICAR certificate of technologies. 8 technologies we have generated as a main developer. And then these are deputation abroad by our scientists, some pictures of deputation.
Okay, thank you.
And that we have organized 4 trainings and 4 trainings for the farmers as well. And these are some of the pictures. And we have also conducted one special talk by the Padam Shri Dr. Sharad Kaleji. And these are students award and recognition. And then our 2 students got ARS, 2 out of 2 seats bagged by our student, and 1 our student got TPCB scientist. And then these are the e-resources generated by our division. And these are some of the awards and recognition to our scientists. Our scientists are from APCC lead author. Some environmental day celebration, student study trip, and then freshers welcome, annual sport meets, and then industrial visit also organized. And then these are the involvement of our students in the cultural activities. And then thank you all. I am really thankful to all. Honorable Director Sir, Dean Madam, and this year Dean Madam has provided 10 instruments for our PG lab and different ICT facilities to our division. Thank you all. Thank you so much.
So thank you, Dr. Saab. I think let us be little sensitive about timing because we should not walk into the other session so much. Keep it okay, but thank you. Dr. Kaushik.
जेब में रख लो चल रहा है Respected Chairperson Dr. Venkateshlu, our beloved Dean and Joint Director Education Dr. Anupma Singh, Coordinator for this session Dr. Monica Joshi, and NRM Coordinator Dr. Brahmanan. Yeah, I am here to present the significant achievements of the students in the year 2024-25 from the discipline of microbiology. In our division, there are 17 faculty members, out of which 8 are eligible to guide PhD student and 10 are eligible to guide MSc student. And we have at present 39 students enrolled in PhD and 6 students enrolled in MSc this year. The work carried out by the different students is divided into 3 broad themes: plant microbe interaction for crop growth promotion, microbial management of abiotic stress in agriculture, and bioprospecting microorganisms for value added products, carbon sequestration and bioremediation. We have total in 7 MSc students this year and 8 PhD students. Out of all the students are from IRI New Delhi only, and no student is there from any other hub or sister institute. So these are the 7 MSc students. So I will come one by one. The student, Mr. Abhijit Pal, under the chairmanship of Dr. Bhaumik, worked on developing a biostimulant assisted medium which can promote the development of pure culture of mycorrhiza in vitro. And he found that the addition of certain phytohormones like BAP and auxins and secondary metabolites like flavonoids and catechin significantly improved the AMF colonization in pure culture. Mr. Deepak worked on the mycorrhiza helper bacteria on Pseudomonas aeruginosa SS113, and he found that it has a biocontrol ability against bipolaris Sorokinina, which is causing disease in the wheat, and this mycorrhiza helper bacteria, if inoculated with mycorrhiza, is assisting in controlling that disease by 72.75%. Ms. Kamali worked on the exopolysaccharide, pure form of exopolysaccharide as a biostimulant for growth promotion of wheat crop or pearl millet, and she extracted this exopolysaccharide from 3 different bacteria, Altinibacter, Bacillus and 2 strains of Bacillus, and found that it was a good promising source of biostimulant for growth promotion under water deficit stress in pearl millet. Then Mr. Sarthak Patil screened different cyanobacteria strains of Anabina and Nostoc for exopolysaccharide production, and finally he selected one strain for EPS production by Anabina, and then he optimized different culture conditions and physiological conditions for maximum recovery of EPS, and he found that red and white light with optimal light intensity of 5400 lux and with having magnesium sulfate of 0.1 to 0.15 gram per liter along with calcium sulfate in the medium increase the yield of EPS production by Anabina. Ujjwal Gowda, under the chairpersonship of Ms. Anju Arora, worked on standardizing the process of bioethanol production for using corn cob stover, and the student found that he used the alkali and acid pre-treated corn cob for fermentation by using Candida tropicalis and Saccharomyces cerevisiae, and could get the ethanol yield up to 2.66%. Mr. Bala Murugan worked on carbon sequestration, and he studied the different diversity of methylotrophic bacteria in different crop, phylosphere of different crop, and he extracted secondary metabolites from those microorganisms and found that they were having a good similarity, significant similarity with the soil humic substances. And Mr. Prajwal, under the guidance of Pranita Jaiswal, worked on that if on the control of degradation of atrazine, then they found that in the low concentration of atrazine, if it is used as in the combination with cyanobacteria, it also promotes the growth and by mitigating herbicide stress. We got 8 PhD students this year, and Mr. Mayur worked under my guidance, and he worked on halophilic archaea. He found that halophilic archaea isolated from hypersaline soil could significantly alleviate the salinity stress in wheat crop grown at very high salinity of 4 to 8 decisiemens, and then we carried out transcriptome analysis of that particular halophilic archaea, and we found significant some differentially upregulated genes which were related to transport and stress response and energy metabolism and detoxification. At lower salinity, detoxification genes were upregulated. He carried out he deciphered the secondary structure of differentially expressed genes and found that they were helping archaeal adaptation under high salinity condition, and also validated these genes by qPCR analysis. Ms. Lavanya used one bacteria, Bacillus, and she found that this bacteria significantly improved the growth under dual stress of water deficiency and nutrient limitation condition under in planter studies in the lab and in the pot trials. And she found that this strain was the main mechanism of stress. The elevation of the stress was due to the production of different volatile organic carbon, volatile organic compounds, and she finally found that this potential bacteria and its VOC is a valuable tool for sustainable mustard production in water limited environments. Mr. Dilbag improved the efficiency of Pusa decomposer, a popular product of IARI of our division, and he found some effective strains like Penicillium talloromyces penicillium and Trichoderma ricci, and using these 4 cultures, they developed a wettable powder formulation for this Pusa decomposer, and they found that this wettable powder, when applied for the degradation of paddy straw in the field, in situ degradation, they found that they significantly decreased the CN ratio and also decreased the hemicellulose-cellulose concentration of the paddy biomass. Mr. Anil Kumar under Dr. Sunil Pabbi worked on optimization process parameters including nutrient composition and cultivation temperature for enhanced biomass and lipid accumulation in Dunelella for the production of biodiesel. So they optimize the nutrient solubilization, nutrient concentration, and they use this technique, one variable at a time, OVAT technique, for optimization of the different nutrient conditions like specific nutrient concentration, sea salt, and temperature optimum. Using response surface methodology, they found that they revealed that sea salt stress in NPK fertilizer as a critical factor for biomass, lipid, and carotenoid production. Along with lipid accumulation, the culture Dunelella also accumulated carotenoids and polyhydroxybutyrate. And the techno-economic analysis indicated that optimized Dunelella cultivation, particularly when integrated into a biorefinery model for co-production of biodiesel, carotenoid, EPS, and PHB, demonstrate significant potential for economically viable biofuel production. Ms. Krutika Patil worked on co-digestion of paddy straw and wastewater grown microalga. They grew microalga using 100% wastewater, and they used this cultivated microalga for the decomposition of paddy straw and for enhanced methane production. And the digested slurry which they produced was able to promote the growth of rice crop in the field after by the application of digestate at 25% of the recommended fertilizer dose. The theme area, plant microbe interaction for crop growth promotion, Ms. Devashish Patak developed a synthetic microbial community formulation based on the core microbial community identified by using metagenome amplicon sequencing method. And this synthetic microbial community, when they inoculated this synthetic microbial community, when they modulate the rhizosphere of the wheat crop, they significantly improved the growth, and also they did the gene expression analysis and found upregulation of nutrient transporter genes in synthetic microbial community inoculated treatments. Mr. Vikas Sharma analyzed the nutrient mobilization potential of cyanobacteria and their biofilms in maize. What he did actually that was steady improvement in growth was observed in the biofilm based biofertilizer using cyanobacteria with an increase of 15 to 20% nitrogen fixation, IA, and exopolysaccharide production. He carried out field experiment for over 2 years and found that using B4 1 to 4 cyanobacterial consortium and Anabina trichoderma biofilm significantly increase the soil biological attributes such as soil microbial biomass, carbon, polysaccharide, and dehydrogenase activity. Both BF14 and Anabina trichoderma biofilm led to increased growth and vigor through enhanced activity of carbon nitrogen assimilation. And he selected two maize genotypes and studied interaction with cyanobacterial bioformulation under moisture stress. And they found that genotype G3 excels over its parent in soil biology, nutrient uptake, plant growth, and gene expression with bioinoculants. They also carried out validated and metabolism gene expression studies, and they found that application of this formulation increased the expression of GOGAT and GS pathway genes. Niveda RM, on other hand, under the same chairpersonship of Dr. Radha Prasanna, carried out development and evaluation of cyanobacterial formulation for nutri-fortification of vegetable crops. She used 4 different cultures, C11s, BF2, BF4, 2 cultures of Anabina BF4, and another one strain more is there. So she used these 4 cultures, and they carried out metabolome analysis of this, and they found that cyanobacterial strains exhibited strain-specific metabolite profile with a predominance of sugar amino acid or their conjugates valuable in plant growth promotion and stat adaptation. She carried out experiment under the spinach and tomato, and she found that soil drench application of cyanobacterial formulation at pre-flowering and fruiting stage significantly improved growth, productivity, and fruit quality in tomato. Seed coating and soil drenching with cyanobacterial mixed enhanced yield and quality traits like ascorbic acid, beta carotene, and leaf iron in spinach. Cyanobacteria as effective biopriming agent. Seed coating with cyanobacterial formulation, particularly Anabina laxa C11, Anabina laxa RPA and A, demonstrated significant improvement in germination disease, seedling growth, leaf enzymatic activity, micronutrient content even after 12 months of storage. So this study highlights the potential of cyanobacterial formulation as resource efficient biofertilizer for vegetable crops, demonstrating their ability to enhance plant growth, yield, and quality through nutrient fertigation and biopriming, contributing to sustainable agriculture and protected cultivation. And student published 17 research paper and 10 research paper in other than their own work. And these are some of the publications published by the students. And all the students are well placed in government jobs. Like some are getting in ARS, some are becoming scientists at DRDO and Haryana Pollution Control Board. One student, Anil Kumar, got selected. Then assistant professor at Galgotia University, assistant professor at MIT University, and 2 of the scientists are selected in ARS last year. And many students have got awards and honor. Most of them are best poster, either first rank or second rank. These are the awards. Then our faculty also, Dr. Lovelin Shukla got AMI Professor G.S. Rangaswamy Memorial Award. She is also elected treasurer for AMI. Then myself, I appointed IMC member, DBT nominee. Dr. Owen Tiwari got Agri Innovation Award for Center Research and Social Development. Meenakshi Govur became Associate Editor, World Journal of Microbiology. And some teaching, training, extension activities carried out our faculty, like Dr. Radha has delivered several keynote lectures, myself delivered and conducted several trainings. Dr. Tiwari.
Okay.
Gone.
Thank you.
Thank you.
Thank you, Rajiv. That was good.
Okay, thank you.
Now we move on to the last but one division, that is soil science and agriculture chemistry. Guys, we have a big list, so kindly summarize the crisply main points.
28 students have to present. So very good afternoon to all of you. Honorable Chairperson of this session, Dr. Venkateswaroli, and Dean and Joint Director Education, Dr. Anand Kumar, former Director, NIPB, PD, WTC, heads of the divisions, professors, distinguished scientists, colleagues, beloved students, friends. I welcome all of you to my presentation. So I am highly privileged and honored to present the significant educational achievements in the division of soil science and agricultural chemistry during the year 2024-2025. So mandate of this division to carry out research on the fundamental and applied aspects of the physical, chemical and biological properties of soil, to study the interaction of fertilizers and manures, then to carry out research on improvement and maintenance of soil fertility, study utilization of organic residues, carry out soil test correspondence correlation studies. Besides this division is also doing the teaching activities in postgraduate teaching and other human resource development activities, extension advisory to the farmers on soil test based fertilizer recommendations. Faculty and student strength, we have total faculty strength of 24, out of these principal scientists 11, senior scientist 6, scientist senior scale 6, 1 scientist, total 24. Currently 3 of our faculty are on deputation, and we have 2 faculty from IRI Assam and IRI Environmental Science 1 faculty. And student strength, so I have given the breakup of students in different years. So total MSc students it is 12 and PhD students 86. So total MSc plus PhD it is 98. So our students have done the research in the following key research areas. First one, theme area, synthesis and evaluation of novel fertilizer products, soil management strategies to enhance soil carbon sequestration, soil health and soil resilience, dynamics of nutrients and their interactions in soils, and remediation of polluted soil and water. So we have 12 students, MSc students. Out of these, 2 each student from IRI Assam and IRI Jharkhand, and PhD student 16. So total 28 students will be getting their degrees this 63rd annual convocation. And this students' research work addresses the thematic different sustainable development goals of United Nations, zero hunger, then no poverty and climate action, and then responsible consumption, water quality. So all these areas our students have worked. If you see these are the list of MSc students who will be getting their degrees, worked in different thematic areas. It's not moving. So theme area 1, particularly the production, development of novel fertilizer products. So Abhishek Pratap Singh Chauhan worked under Dr. K. Manjaya. So they developed the nano-based fertilizer, nano-clay polymer composite fertilizers. Mainly the approach was to replace the costly acrylic acid substituted with wheat flour by 15%, and they prepared the NCPVC, and then the FTIR spectra of the NCPVC was carried out, and it has been observed that different functional groups at different OM number it appeared, and these are the functional groups are the sites for making complex with the the nutrient element, particularly here the urea is loaded with the NCPVC products, and scanning electron microscopy clearly showed that urea loading is clearly seen in this picture, SEM images where the crystals of urea is seen here, and these products also the slow release characteristics also was studied, and it has been observed that particularly as compared to urea, NCPVC and as well as NCPVC with zinc phosphorus solubilizers are also used. So they found potent as your slow release characteristics for ammonia and nitrate. And then the yield, you can see that NCPVC with phosphate solubilizer and zinc solubilizer showed the highest grain yield, as well as your the apparent nitrogen recovery as well as grain yield. And another student worked on this, particularly the thematic area 2, that is carbon sequestration. Pradarshak Sen worked under Abid Day. So their objective to see the different types of residue which are used in the agriculture, and they actually studied the litter chemistry of different residues, rice, maize, cotton, with Pusa decomposer, and it has been observed that particularly use of Pusa decomposer actually increase the carbon mineralization rate, and among these different residues, cotton is very much recalcitrant in nature because it contains large amount of lignin and polyphenol and cellulose, homocellulose. So that's why they are very much resistant to decomposition, whereas maize residues with Pusa decomposer gets faster decomposition. And if you can see that when the temperature changes from 25 degree to 35 degree centigrade, so particularly here the rice residues which were found to be relatively recalcitrant, but these residues become more vulnerable to decomposition when the climate change, the rise in temperature happens. So that's why future scenario of climate change, so this type of information is required when we are going for residue management in future time course. And Q10 also calculated, and you can see that Q10 value of cotton was lowest, and followed by your rice having higher Q10 value. Because of the rise in temperature, the recalcitrant part of the rice residues become vulnerable to decomposition. So Pusa decomposer acquisition leads to decrease in priming. So priming means it is actually preserving the native soil carbon. So in turn protecting the inherent soil carbon, indicating towards soil carbon sequestration. Another student, Chandra Saha, worked with Dr. Abid Dey. Here actually a new concept, she worked on basalt rock mineral powder, which is marketed as Dharakolp in a Gujarat-based company, and this basalt rock is used as a carbon negative, carbon resilient agricultural practices. So here in this direction she worked, and she could see that, and they actually identified what is the maximum dose of the basalt rock which can be promoted for higher carbon capture from the atmosphere, and it has been observed that the first order differentiation of the second order polynomial clearly showed that 8 tons of residue, the rock mineral powder can be promoted, used at 25 degrees, but 35 degrees it can be increased to 25 tons per hectare. And carbon mineral, that is carbon capture, is 356 kg per hectare at 25 degree, and at 35 degrees it is 379 kg per hectare. But other co-benefits of this type of rock mineral powder is that it can act as a liming material. Calcium carbonate, pedrogenic calcium carbonate is produced, which can remediate the acid soils. So Shalini Kumari worked with Srila Das in the thematic area, your nutrient interactions, particularly third thematic area. And here actually the zero till and along with the nitrogen dose, they studied the effect of zero tillage and nitrogen dose and phosphorus dose on some macro aggregate and micro aggregate formations. And it can be observed that zero till with nitrogen dose of 125 kg nitrogen, 125% nitrogen dose showed higher aggregate formation as compared to conventional tillage. And but interesting thing is that when phosphatic fertilizer was used, so it actually increased the macro aggregate formation. But in the case of micro aggregates, so that smaller size aggregates, that actually behaved in a different opposite directions. And it can see that Shannon diversity index and Simpson diversity index, particularly the arbuscular mycorrhizal fungi population, they studied, and they could see that there is an increase in the population diversity, particularly in the zero till and nitrogen managed plots. And here they studied the arbuscular mycorrhizal fungi, how they are infecting into the roots of the plants. And Devmalla Sarkar, he worked with Dr. Devarup Das. They determine the minimum silicon dose for enhancing phosphorus availability. As you know that silicon, phosphorus, they are oxyanion. They actually compete each other for the adjustment sites, and it is already reported that silicate may decrease the phosphorus fixation into the soil. And they actually tried to identify at which time the maximum phosphorus availability can be observed through this study. And 20 soils they collected having different soil organic carbon content, and they are having different physiochemical properties. And silicon dose was applied 250 ppm. It can be observed that at the 4th day of the application of these, the maximum phosphorus availability was noticed. And on the basis of that, they also carried out to identify the maximum dose which can be used for this type of technology. And they classified these soils into 10 non-acidic alluvial soils, 4 acidic alluvial soils, 4 red lateritic soils, and 2 black soils. And they did the frequency analysis, and they determined the weighted average. And you can see from these the different types of classification, different maximum dose of silicon was noticed for different types of soils. It is not working. Some battery might be.
Manually करके देखो. Yeah, it's working.
बीच में नहीं कर रहा है ना. Sometimes it is not working. Previous slide. This is actually a server. By mistake, I have given Dev Mallo Sarkar. Server, SG server worked with Dev Mallo Sarkar. Actually, in the potassium assessment, available potassium in the soil, we used thermodynamic parameters like QI, which is used for measurement of the available potassium content in the soil. So this is a tedious method and time-consuming method. So how to circumvent this method and decrease the time for this purpose? So they actually developed this type of prediction equations for activity ratio of potassium, which is the intensity parameter. By using these equations, they actually predicted the activity ratio for the training and testing. So training purpose they use this, and testing purpose they use, and they could find that very good agreement with the predicted activity ratio with the measured. And then del K, that is your change in the potassium level in the soils during the adsorption-desorption process. And this actually activity ratio and del K could be estimated accurately using the two soil solution equilibrium ratio, 1:10 here, and here it is 1:80 at 0 ppm. And with ppm concentration, this 2-point approach can be used to estimate the QI parameters for soil K without compromising much accuracy. That is the major findings of this. And by using this activity ratio potassium, they also actually determine the change in your Gibbs partial molar energy, change in energy, change in energy in activity ratio, RTL and activity ratio of potassium. Del G they calculated, and they could come out that sodium tetraphenyl borate was a better indicator of plant available potassium, ammonium acetate or nitric acid extractable potassium. And potassium, sodium tetraphenyl borate and del G as a single point approach emerged as a useful predictor of soil K availability to the plants. So this is the major findings of this. And then another student, they used this, they worked on fungus inoculated rice straw biobait for enhancing phosphorus availability in soils. Atul Meena worked with Dr. Hindu Chopra. They actually prepared the biobait with the rice straw, race is the minimal medium, and fungus strain they use KSFC1. And they could use this biobait as for your solubilization of phosphorus, and they could see that from this incubation study at 90 days, particularly the tree treatment, which shows that biobait actually applied with PSF at the rate of 1.5% with the soil, found to be highest solubilizing capacity. And biobait they prepared, that is 1:4 weight by volume ratio. So that is the highest P solubilizing capacity. This is the major achievements, and in this work they also identified succinic acid as the major acid which is used which is produced by the fungus that is responsible for the solubilization of phosphorus in the soil. Tripti Pal, she is from IRI Assam campus and worked with Dr. A.K. Srivastav, and she actually worked in a Assam lemon in the village lemon orchard, where the village in the Lakhimpur district, and they actually because these are acid soils and very poor in calcium, magnesium content, and zinc deficiency, boron deficiency is a major factor for quality of the yield and production of the lemon. And from this study, it could be observed that T12, that is the NPK with calcium, magnesium, zinc and boron. So that showed the highest number of fruits and fruit weight as well as yield, and also fruit quality, particularly the diameter, length, peel thickness, total soluble solid, juice contents also improved by this. And they also identified the critical concentration, leaf concentration of nitrogen, phosphorus, potassium and zinc with the relative yield, and also the critical concentration available calcium, magnesium, DTP extractable zinc and boron they could identify. So this can be used as a guiding principle for managing this type of acid soils of Assam which are devoted for growing Assam lemon. Another student, Sneha, she also worked with Dr. Srivastav. They use this development of optimum soil fertility and leaf nutrient standards in relation to fruit yield of Assam and lemon. And they could use this plant available nutrient standards they developed for Assam lemon by using DRIS method. And then also soil fertility analysis based DRIS indices they calculated, and they could observe that these are the nutrients calcium, magnesium, zinc, phosphorus, nitrogen, potassium which are deficient, negative value, and iron, manganese, copper which are sufficient. And these are the 14 Assam lemon orchards she worked, and this type of information is very important for managing the acid soils of Assam growing for lemon cultivation. And then Ankit Kumar, particularly he worked on the theme area 4, particularly remediation of metalloid contaminated soils, and he actually developed the composite beads for the removal of chromium and crystal violet from the simulated effluents and its impact on mung bean crop. So here actually particularly the crystal violet is emanating mainly from the textile industry, and nickel chromium is from your electroplating industry major source. Being they are untreated, they are discharged, so they cause pollution in the environment. And they have also done the adsorption study of crystal violet and chromium. This is the before adsorption FTR spectra. You can see that FTR spectra, all these are actually disappeared because of the formation of complex with the chromium as well as crystal violet. That means there is a strong complex with this, and it can absorb these pollutants from the solution form, and it can arrest these metals and dye coming into the system. And here you can see the scanning electron microscope of the beads, where it is a rough surface which is potent for absorption of these pollutant from the soil. And when these remediated water was used for irrigating the crops, you can see the T7, particularly treated crystal violet, 1 milligram per liter, containing simulated effluent, and T2, T3 people are untreated. So these are untreated and it is a treated, so there is a significant increase in the yield. But when there is untreated is used, there is a decrease in the yield. That means the contaminant is coming and it is causing, decreasing the yield of the crop. And these are the list of PhD degrees recipients working in different thematic areas. Total 16 students. Here theme area 1, particularly your novel fertilizer products production. Premlata Meena worked with Dr. K.M. Manjaya, and here actually in NCPC mainly the cost of acrylamide is very high, acrylic acid. So how it can be substituted with your other biopolymer like maize flour, so she used maize flour at 20% substitution, and you can see that here they used for in different crops like maize and wheat, and it can be observed that particularly she actually uploaded the your DCD as the your nitrification inhibitor. So she used along with urea, and it can be seen that particularly that treatment showed the highest grain yield in case of maize as well as wheat, and apparent nitrogen recovery also was highest there. And another thing of this experiment was that these NCPVC based fertilizers can reduce nitrous oxide emissions by 28% and 30% in maize and wheat respectively. And here also another student worked with Dr. Manjaya in the same slow release fertilizer, novel fertilizer products, particularly NCPC they prepared by your acrylic acid, acrylamide and ammonium persulfate bentonite. And here also he did with 20% substitution of acrylic acid with wheat and maize flour. And you can see that particularly your XRD analysis clearly showed that this 2 theta 6.4, that is 13.8 degree angstrom, that indicates the d-spacing of the montmorillonite present in the bentonite mineral, and here it is the wheat floor and maize floor. But when there is a NCPC for an NCPC wheat and NCPC maize production was there, these fertilizers are produced. So then these are the disappearance of the peak it was observed. So that means that incorporation happens with the polymer. So better grafting that took place. And FTIR spectra also you can see the band at 14 and 40 centimeter per centimeter. The skeletal C double bond O stretching aromatic suggests the presence of wheat and maize flour in the NCPVC based fertilizers, and 1028 per centimeter presence of bentonite. So there is proper grafting of the bentonite and the polymer that happened with these products. And then they also did field experiment where the nitrous oxide emissions they studied as compared to urea, NCPC, NCPVC, they decreased the nitrous oxide emission as well as ammonia volatilization. And from this study, apparent nitrogen recovery also studies, and particularly the NCPC, NCPVC, maize and wheat, so it increased the apparent nitrogen recovery as well. And this is the thematic area 2, carbon sequestration. Angshuman Das, he worked with Ranjan Bhattacharya, particularly zero-till direct seeded rice, and then rice residue, then zero-till wheat residue, zero-till mung bean. So this type of combination proved beneficial for changes in carbon addition over time. This is the highest carbon addition that happens in these two treatment, and carbon stabilization, so that happens also highest in this. And from this it can be observed that about 6.7% of the added carbon under transplanted rice and conventional wheat plots stabilized inside the microaggregates from the intra-aggregate microaggregates which is present within the macro larger side aggregates where the carbon is stabilized. So this is another important study. And Jyotirmoy Sahu worked on impact of different residue management practices on the dynamics of your carbon in soil. Here this is a long term experiment, 10 years impact, where the residues incorporation, biochar, and then residue burning and residue complete removal that were studied in a long term study. It can be observed that particularly the treatment which has been receiving biochar with NPK showed the highest carbon sequestration rate as well as amount of carbon sequestered, as followed by crop residue incorporation. So this another student worked with me, Devali Rai. So she actually worked on development of novel soil organic carbon based protocols for soil quality assessment. You know that soil quality assessment is a very tedious process and it requires large number of indicators, 22 indicators it requires. So how to substitute that indicators with surrogate, SOC based surrogate indicators, because soil organic carbon is considered as the one of the important soil health indicators. So by this approach, she actually in a conventional, conceptual framework based soil quality assessment. So these are the surrogate indicators, the SOC based surrogate indicators she identified, and then this protocol used for soil quality assessment. So here in this long term experiments we studied, the different residue management studied, we can observe that particularly the treatment which has been receiving the residue incorporation showed the highest soil quality, and these are the SOC based soil quality indicators. And the SOC indicators which came most important, these K1A4 oxidizable carbon, DOC, TOC and beta glucose, these are the 4 important SOC surrogate indicators which can be employed for assessment of soil quality instead of there were a large number of chemical, physical and biological parameters. And she also validated this, this one with the other 4 type of soils, vertisol, inceptisol, alphisol and mollisol, and there is a good agreement between the SOC based approach with CF based approach. And also she developed, she developed the DSS also, decision support system, computer model she developed, computer software, and this is for under copyright. And Sunita Jadhav worked on the theme carbon sequestration. She worked on different plantations in the northeastern part of India, then Tripura, bamboo 5 years, tea 30 years. So major findings of this study that particularly the tea plantations for 30 years. So that produced the highest, showed the highest TOC stock, and also that your your different depths also that that also was higher in particularly tea plantations. And another theme area 3, she worked, Dr. Riyaz Rahman worked with Sunanda Biswas, and they calculated the carbon sequestration potential of mollisol, then insectisol, vatisol and alfisol, and they could observe that particularly the treatments which had been receiving chemical fertilizer with organic source, that they have higher potential for and then Ravindra recovered these nutrient interactions. So particularly potassium in conservation agriculture management, very few studies are there, particularly zero tillage permanent bed and the zero till flat, conventional till flat, where the zero till permanent bed showed the higher water soluble potassium, exchangeable potassium, and among your cropping system, particularly your all the systems, particularly two system maize, wheat, mung bean and maize, chickpea, sesbania showed higher potassium availability in the soil. And King-Shuk Modoc also worked on these nutrient interactions, particularly the vertisols and insectisols. She worked particularly in the long-term experiments where 10 years impact was there, particularly the treatments which had been receiving NPK with FIM, where the fixation of potassium was less because adsorption of potassium lowest in this treatment, and desorption also it was highest in this treatment. So this indicates that particularly the long-term application of organic manure along with fertilizers, so that actually prevents the potassium, your phosphorus fixation in the soil, so that can modify the fertilizer application rate and it can save the costly phosphatic fertilizers. And Moumita Ash worked with Dr. M.C. Meena on the potassium stratification, particularly she worked in a conservation agriculture practices, 7 years impact. It can be observed that moderate residue distribution of combined with bioinoculants and have recommended the potassium offers an optimum balance between productivity and sustainability. And you can see the activity ratio of potassium was higher, and there is a stratification of potassium in the particularly in the 0 to 5 centimeter, there is a higher potassium activity ratio as well as the potassium buffering capacity that decrease if you go from your residue to higher residue treatment, because these two parameters they work in opposite directions, potassium buffering capacity and activity ratio of potassium. So main thing of this research is that in conservation agriculture, crop residue detection has lot of implications on potassium availability in the soils. And Pooja Tamuk worked with me, and particularly she worked on the iron deficiency tolerance.
Sorry to interrupt. Can you just go
so she actually 57.
Since time is already almost over,
Just I will tell that she identified sensitive iron sensitive variety as well as tolerant variety. From this study, it can be observed that particularly Danteswari and Sona Masuri sensitive and Danteswari. So this type of information, iron deficiency tolerance behavior of different cultivars will be helpful for the breeders to breed particularly your iron your tolerant variety for meeting the iron deficiency situation and also the nutrition.
Dr. Purkastha,
here,
because we have another session also, so kindly be very concise here.
No, I think you go to this, anyway we have read these abstracts, come to the conclusions and publications and individual we have already read it.
Okay. So here Basid Raja, she worked with Dr. S.P. Datta. They prepared the nano zero valent iron and particular bentonite based product for removal of arsenic from the soil, and they could also see that particularly a basic slag can be used as a potent amendment for arresting the arsenic from soil to plants. And Kavita worked with Dr. Nayan Ahmed, particularly this from this slide I can say that if you increase the organic matter level of the soil, particularly the humic acid fractions, so that can stabilize the heavy metals transfer from soil to the plants. And Gupta, Saurabh Subra Gupta worked with Dr. Meena. So he worked on molybdenum. Particularly, he used different extractants, and out of this, anion exchange was most effective. But it is
kindly come only on the outcomes. That's all. Let's not explain the methodologies.
So just I will take 1 minute. Yeah. Publications, we have 36 publications out of from the students and faculty 36, total 72. And these are the different awards and honors received by our students. Faculty, this year Dr. Mandal, our HOD, got inducted as fellow nurse. And these are the training programs attended by our students. And these are the different activities our students are involved in: World Soil Day program, field day, educational tour, and then workshop, and the foreign delegates. So thank you very much. Because I had to present.
You, but you have a very large number of students.
Sir, 28 students I have.
Yeah,
so it is very difficult to present by 20 minutes even.
Given the context of this meeting, we have to accordingly plan. So last, madam, you have only 5-6 minutes. You have only two students, I think. Saw your list. You know, water is very important, but 3 students. 2 MSc, 1 PhD.
So 9 students.
Okay, I thought she will present only this. 9. Oh.
Why then 9 are not given here?
Why it is not given?
No, मैं इसमें कर देता हूँ.
Yeah, only 3. Okay, anyway, Sushma, be considerate to the next session. Try to be.
So now that every time we have a scope of improvisation, so here also another thing has come up. Next time onwards, whosoever are going to submit their complete their viva and all after the deadline, their work will also not be presented, and simply that will be shown as these are furthermore theses which have been done, because otherwise it's a very, very humongous task for our very worthy and learned professors to complete, and also for the chairs also to comprehend and to get an idea. So let's all honor that. I would request the heads and the professors to kindly cooperate in that.
Good afternoon, one and all. I am going to present the significant achievements during 2024-25 of water science and technology. We are having 12 faculty and 33 students. Out of this, this time 2 MSc students and 7 PhD students are going to take the degree. So I will be very brief because time is short. So they have worked in 3 research themes: water availability, conservation management, and measurement and modeling. Next is marginal quality water use in agriculture and efficient irrigation water technology for enhancing water and crop productivity. So coming to the research theme area, under this 3 PhD students, they are going to take the degree. Raghav Maria, under my guidance, he has developed an automatic runoff and sediment measurement device and evaluation in horticulture system. As you know, there is very much important for runoff and soil loss measurement, but it is at this time this is manual and this is very tedious. That's why he has developed this automatic runoff sediment measurement devices. He has followed all the procedures for performance evaluation of different sensors, and through Arduino and the best sensor he has integrated and developed this automatic runoff and soil loss measurement devices. With this, all the procedures he has done. So, and the automatic this device is giving the R square of 0.99, and it is working very well, and the cost is only 18,800. The next is a performance evaluation of simulation of subsurface dam by Sangeeta under my guidance. She has studied the performance about different subsurface dam in Himalayan region, which is very important due to the boulders in streams and rivers to construct the surface dam. So that's why she has conducted the experiment as well as field to lab to field. So all these things she has done, and after this she has taken 432 treatments with different construction materials, different height, different this discharge, different slopes. It's a total 432 treatments in lab also and in sandbox model. And it was found that the concrete trapezoidal both side slope with the geotextile structure is the best, and the cost is around 4 lakhs. When in the field, the present situation is 11 lakhs. Then next is modeling of water resource availability and demand for enhancing crop water productivity in Ramanga River Basin, that is by Hari Krishna. He has used the SWOT model and model all the water balance components and also the optimal water allocation plan. So the SWOT model is performing very well, and with R square more than 0.9, and also the NSC efficiency, it is more than 0.8. And next is marginal quality water use in agriculture. One MSc student, that is Ankit Pratap Singh, under the guidance of Dr. D.S. Gurjar, he has done the standards video sensitivity for soil moisture sensor guided wastewater use in gladiolus. They have taken different treatments with different moisture depletion, and it was found that 50% made with the system using the wastewater is the best. Then efficient irrigation technique for enhancing water and crop productivity. Under this, the Mohammed Ekhlaq is the MSc student of Dr. V.K. Prajapati. He has compared the crop evapotranspiration and soil moisture depletion based irrigation scheduling. They have taken different soil moisture depletion methods as well as the evapotranspiration. That is that different SMD and ET based, and it was found that either 20% SMD and 100% ETs can be used. The next is Siddhartha Gaudam, as we are talking about organic fertigation, organic farming. So how we can use the bioslurry, that one student in last year, she has taken the gold medal for this work. She has developed the filter system at Water Technology Center, and Siddhartha has done the extension of that, how to utilize more the bioslurry to reduce the clogging. So he has used the Pusa decomposer before filtration. Then after filtration, he has used the biofertilizer, and he has taken different treatments in 2 crops, cauliflower and radish. Then it was found that 7.5 ml, 100 ml per Pusa decomposer can be used for 21 days incubation, and 80% it is with filtered bioslurry and biofertilizer nutrition is the best. Then next is Saujanya. She has done the in drip system, the tuberose, under the guidance of Dr. D.S. Gurjar, and she has studied the different pan evaporation method for irrigation scheduling and different treatments with the different deficit method. And it was found that raised bed and 100% pan evaporation, 100% RDF is the best. And the next is Kishore, and he has studied what Dean Madam has developed, the Pusa hydrogel and also SPG 1118. And there are 2 things, root dipping as well as soil application. He has compared in drip irrigation system with in red cabbage, and it was found that SPG 1118 is the best with root dipping to be used, which will provide the net benefit more under drip irrigation. Now coming to the another student, Ankit Dahiya, what we are talking about, whether we will go for soil moisture depletion method or crop water stress index based. So he has compared under different nitrogen and also different irrigation method under Dr. Manoj Khanna, and he found that it at 50% SMD soil moisture depletion, and it is the best method with 150 kg nitrogen. Now come to the student replacement. Ankit Daya and Gokulraj, they have already posted, and Dr. Smith and Dr. Rasmi Javjana, other students, they are continuing their PhD. Research publication 22 with the training manual 2, book 2, conference 10, and other patents nil, but technology certification 2 and popular article 10. And these are the student publication, just only from their work, not from other work, during 24-25. And our students have attended different training programs. And educational trip to Uttarakhand for 5 days. And last year, Rasmi Yadav has received the IRA merit medal during 62nd Convocation, and Vishnu Prasad received third prize in speech competition during IRA Foundation Day, and all other students, they have participated. And during our World Water Day celebration also, these two students, Sairam and Kirtana, they have bagged the prize in quiz competition. So these are the different activities that has been conducted in WTC. Once our project director also has taken keen interest for the participation of the students, and as he has due to his supervision, skit was prepared, and that has been shown during the IRA Foundation Day last year. And these are the other activities, that is students, director sir was interested that students should be included and they should take more participation in Swasth Bharat mission. That was also we have conducted, and students participated in different conference to organize. Honors awards, Dr. P.S. Brahmanand, our project director, has received Eminent Scientist Award, and our Water Technology Center has received the first prize for a successful implementation of a Rajbhasha Puraskar from IRI, and Dr. B.K. Prajapati is the nodal officer. And there are so many in core committee, Dr. P.S. Brahmanand Sahab is there. And then Dr. Ravinder Kaur also is actively contributing the different SOPs and different standardization of sewage water. And she is also involved in different members, and I myself also bagged commendation medal from Indian Society of Agriculture Engineers. And then Mona Lisa also received different awards, and these are the different activities from WTC. And the first time we have done the Technology Day come industry meet, and also in different national workshop, Teachers Day, and other things that has been organized. And not only our students are going, other students are coming for educational trip. And this is very important. Our Jalopchar technology developed by Dr. Ravinder Kaur has also been commissioned, inaugurated by Honorable Governor of Rajasthan, and that has been placed at Jobnar, 1 lakh liter capacity. And next is, it has been commissioned at Umiam, Meghalaya, 0.5 lakh hectare. So that has been also connected to drip system, and one is awaited. So with this, thank you very much, all faculty, students, and Dean, Director, Administration, and Chairman Committee. Thank you very much.
So thank you, Sushma. You have been really very brief and very pointed.
Thank you.
So with that, we come to the end of this session, and I would have wished to have some input from the audience, because there is a lot of wisdom in the house, but given the time constraint, I think we have really no option but for me to summarize in 5-10 minutes what are my impressions in the afternoon session. First of all, I would like to congratulate whoever has put up this entire volume, to bring all this together into one place. And obviously, in an NRM kind of subject where from engineering to microbiology to, water, it's a very diverse subject, and obviously it's not that easy, and there will be always some overlapping the presentations, and there have been some overlappings. But leave that aside, I would only like to, congratulate and appreciate the quality of work, that is being done, and the level of intellectual input that is going into the planning and implementation of this work. Definitely, I had a chance to visit many universities and chair many meetings, but this is my first time to have an opportunity to chair something in IRI. So definitely the reputation of IRI, I think I now see today that why it is so reputed. Next point I thought is that next time when you make this session, have some uniform format, not that, especially the individual results of experiments, like bar charts, CD values, they are not required here. I think what we require is a student name and then what thesis title he has, and what are the 2 or 3 outcomes. That's all. Remaining, I think there are other opportunities for you to really present in depth, maybe division level meetings or your other reviews, but not here. I think that is my understanding. The Dean may also have her own opinion, but that is my understanding. Second is that 4 or 5 key recommendations from my side I thought will be useful. Number 1 is that there is a lot of good output that is coming out from the students' work. Now, I don't know historically in the last 60 years, how much of that student work has really gone into commercial use, either by a farmer or by a company or by an industry, whatever it is. So for that, I find some gaps, in this. For example, if an MSc student comes out with something very good, I really found the student work in agricultural engineering, the person who did that, canopy reflectance-based variable rate application of fertilizer. It's a very good work. In fact, it is a very much commendable work, but point is, if the student after MSc gets a job and goes for outside, leaves this place, and then who will continue that work? One way is that work can be given to a PhD student in the next year by the same guide. That is one way. So otherwise we have to really see because the key point is the continuity of the work, to take it to the logical conclusion so that a product ultimately comes out from IRI. Paper is okay, but paper is not a real product, it's definitely a good product for an academic angle, but for an angle, country's benefit point of view, it is the product which should really come out. And that can happen only if there is a continuity of work. So one way is this MSc going into PhD, and if it is a PhD work which has come out with something good, then next thing can be, in other models what we have in our country is the incubation. I know there are many institutes, in CSR institutes there are few scientists, 40, 30 scientists, but there is a very strong incubation system. But in IRI, I do not know there is an incubation center or not, otherwise it is urgently needed. In fact, that is the first thing that is required now. Whatever budget you require, I think you must make a case to the ministry, to DST or Anusandhan Research Foundation of the Government of India. Because in IRI, if you have 100 ideas or 100 semi-cooked products that are coming out every year, out of those at least 10 to 12 should really go into the market, either to an industry or to the fertilizer company, whatever it is. So that, I think, the core point you must take from today's presentation from my view. So whatever is required for that to happen, you must really discuss with your director, with the DG. Had Dr. Pathak been here, I would have talked to him, but now we have no DG. But whenever we have a chance to talk to DG, I think we will certainly push this point. Second point is that, at least I find 2 or 3 important national issues which can be addressed through this as an institute, For example, one is being addressed already, is the enhancing fertilizer use efficiency from current 45 or 40, whatever. Even if you increase by 5% urea use efficiency, you will save 20,000 crores of rupees for the country, and that is the budget of IRA for 20 years, or maybe for this kind of research. So just imagine. So the kind of impact that will have an issue which you can address at national level. So that, I think, is being addressed by soil science people and also engineering people. But you have to really focus on that. That, not just leaving after 1 or 2 students work MSc, PhD and then because many times what happens when this professor also retires, then the next professor may not have interest on this. So then you will be driven by the professor's priority rather than the country's priority. That should not happen, at least from an institute like IRA. So the country's priority should keep on reminding you, keep on driving your research agenda, irrespective of the professor's interest or the student's interest. And that can happen only if the leader is really aware of the urgency of the country's issues and then keep on driving the research agenda to meet that goal. That is one thing. Second is, of course, lot of entrepreneurs are coming in the country, who are very keen to use the IT, the sensors, the remote sensing technologies, etc., all in agriculture. there are 100, 150 entrepreneurs, startups who are doing very well. Some are not doing so well, but some are doing very well. And the kind of knowledge that we have seen today, that really can go to the startups. And for that to happen, merely this presentation won't help. And I feel that next year onwards, you must invite some entrepreneurs also to this meeting, to this presentation. After this presentation, if not this, if you don't have any provision, but at least to organize one a linkage or entrepreneurship linkage day wherein you present whatever output you have and let them pick up, whatever. And the best output can come only by licensing, by having some MOUs and, this kind of interaction with the industry. Not just by publishing and then bringing out volumes like this. No. So that is another, but these things are easy to say than done. But I think we must start thinking in that direction. At least if you now think in the 5-10 years, we will have some impact done at the country level. Fourth point is, whatever your fertilizer formulations or biofertilizers, etc., whatever we are doing, focus on oil seeds because that is the gap we have in our country. Oil seeds production, even if you increase by 10%, you will again make a very big contribution to the national, foreign exchange spending. So focus on any PhD student who comes to you, first priority you give to oil seed and pulses. Whatever problem, it is a agronomy or it is a biofertilizer or it is a pest control, whatever it is, because that is which is what is hitting the country. So like that, I think if you can plan the dean level or director level, keeping the national interest in mind and keeping the need for taking every work into a logical conclusion, into a product, into a licensable product. So many times we may not be able to ourselves, bring out a final product, but incubate it or license it to a person who can do it. And there are a lot of people, a lot of venture capital in our country, a lot of people who can do this. But we have to have that sensitivity, that kind of thought process in our mind so that we ultimately give some good thing to the country. So this is, of course, a big agenda before you, but I thought this is very important to highlight having come here. Thank you very much.
Sir, thank you. On behalf of the faculty and students, sir, may I kindly respond to some of the points sure. And so as to showcase the opportunities and the systems in place at IRA in that direction. Sir, as we discussed during our in the afternoon when we had met during lunch. So, sir, the students are essentially working as the instruments of operation in the research projects of the faculty, and the fact once the student moves over, he or she goes over out out of the system, then automatically that becomes a part and parcel of the faculty's ongoing research, which is then given to either the next student, or then it is then upscaled in the form of the research grants or the in-house projects. This is one thing, sir. Second is, sir, we have a well laid out system of the incubation. So we have zonal technology management and business planning development units, sir. And in order to, so far, the ecosystem was so that, sir, it was only, it had a limited scope of benefiting only the graduates and the youth from outside the IRI system. Students were actually not the beneficiaries out of that. So therefore, sir, we have, Academic Council has approved now a system which is very robust. It has been implemented. Now it is the proactiveness of the students and the faculty. I have been repeatedly saying that they have to harness. That system is, sir, that is student to startup scheme. This is one. In that, the students are made aware about the innovation basket in their own labs or across the labs of the entire IRI, from which they can take up the innovation as they want and then work up in line after due mentoring to become, first of all, the pre-incubatees, incubatees, and then as the entrepreneurs. So this system is there, sir. Now it's 1.5 years we have started, we are sensitizing. Another thing is, sir, as regards the IP part of it, so sir, normally in our case in the IRA system, we work on either the bench scale know-hows or up to a certain limit up to that. And of course, now we have that IRA industry fellowship scheme also, wherein I have been most humbly, but I have been repeatedly sensitizing our faculty to get in touch with the industry and know that what is the problem of relevance which the industry will like to then take up. So that once the student takes up a problem which is of industry relevance and of course of the impactful relevance, then industry will support that. So these are some of the things, sir, and definitely, sir, your words of wisdom, we are going to record and put it up to the director for further necessary action. Thank you, sir.
Thank you, sir. It's now time to felicitate the chairperson, ma'am. I also request Dr. Brahmanand, sir.
So thank you everyone. As the co-convener of the session, I would like to thank everyone present here, especially Dr. B. Venkateshwarlu for his presence and gracing the occasion for 2-hour session. Sir, the Natural Resource Management School is the biggest one and we are giving 108 degrees in this convocation in 7 divisions, sir. So that is why it took a lot of time. Thank you for your presence, sir, and your words of appreciation to the scientists, to the students, and also your advisor. So, and thank you everyone, all the professors, students of the sister institutes, IRI Outreach Campus, hubs, everyone who have contributed to the successful completion of this session. Thank you everyone. And we will be meeting after 15 minutes because we are already late. After 15 minutes, we'll start the next session on School of Basic Sciences. We already have the presence of the chairperson of that session. So please be very punctual and come within 15 minutes. Thank you, everyone.
We will start at sharp 5:25. Kindly try to make back.
Good evening to all. So we are about to begin the last session of the day. Myself, Dr. Monica Joshi, professor in the discipline of Seed Science and Technology and convener for 2 days professor presentation, along with my co-convener, Dr. Shruti Sethi, principal scientist, Division of Food Science and Post-Harvest Technology, heartily welcome all the esteemed dignitaries, faculties and students to the 4th and the last session of today. The chairperson for today's 4th session on basic sciences, Dr. P. Ananda Kumar, former director, ICAR NIPB New Delhi, is amongst us today. It's an honor for us to have you here, sir. So may I kindly request Dr. Ananda Kumar and Dean Ma'am Dr. Anupama Singh to kindly grace the dais, along with the co-convener Dr. Shruti Sethi. Thank you, sir. Thank you, ma'am. A round of applause, please. May I now request Dean ma'am to kindly welcome the chairperson. I now request Dr. Anupama Singh to kindly deliver the welcome address and introduce the chairperson to the August gathering.
You have not given me. Honorable Chair of the last session of the day, Basic Sciences, School of Basic Sciences, worthy Dr. P. Anand Kumarji, very learned HODs and professors of the School of Basic Sciences. It's indeed a privilege and kind of nostalgic moment to be here with Dr. Anand Kumar, sir. Sir, when I used to be there as the scientist in the Lal Bahadur Shastri building, it was the time when your persona used in that particular building used to be something like a banyan tree. And always I used to look forward for the chance to when you used to pass through the corridors. I used to, greet you with a hope that sir will look at me and then greet, and always he greeted with that charm and that benevolence. So that was the charm, and that is still there today. And my dear friends, just like there is that quote कि वृक्ष जितना फल से लदा हुआ होता है उतना ही ज़्यादा झुक के रहता है, ठीक उसी तरह से आनंद कुमार जी को मैं हमेशा उसी भावना से मैंने सर को देखा है, और सर ने हमेशा आज भी just like that only. So these are my personal feelings for Dr. Anand Kumar sir, which are not a part of my formal address. So thank you very much, sir, for being here. So, dear audience, I welcome you all in the 4th session of this professor's presentation. Of course, again, just like I said, from the core of my heart, good words for Dr. Anand Kumar. You are aware that I am deeply disappointed by the very, very thin attendance. Again, it's our function, it's our movement, but I don't know where I have faltered or where the team has faltered, that we are not able to sensitize, or we are not able to create that feeling for the people to prioritize, and the students to prioritize their other things and come up and gather and then applaud and enjoy. So I'm really heavy-hearted on that part. Again, this is not a part of my formal address, but since this is the last part of the 3rd day, I thought to indicate my feelings rather than saying on the professors group key, okay, tell the all, otherwise their fellowship will be deducted or attendance will be reported to the competent authority. There is no point. So with that, I welcome you all. You all are aware that today we have Dr. Anand Kumarji, and in this presentation, in this particular session, sir, there are 51 theses comprising of 36 MSc and 15 PhD students. And there are three core disciplines: biochemistry, molecular biology and biotechnology, and plant physiology. So on behalf of now the director, I take this opportunity to introduce the chair of the day to the audience, Dr. P. Anand Kumarji, former director NIPB, served as the director of National Institute of Plant Biotechnology, and also director of Institute of Biotechnology, Acharya N.G. Ranga Agricultural University, Hyderabad, and director of the Indian Institute of Rice Research, Hyderabad. Dr. Anand Kumarji is a botanist who specialized in plant physiology, molecular biology, and biotechnology. He obtained his MSc degree in botany from Sri Venkateshwara University, Tirupati, and joined as a scientist in the Agricultural Research Service of Indian Council of Agricultural Research in 1978, when we were all, either many of us were not even born or were in our early childhood years. After working as Alexander Humboldt Fellow in University of Hanover, he moved to Biotechnology Center, ICAR, in 1993, specialized in the area of transgenic development for insect resistance, utilizing the genes encoding insecticidal proteins of Bt. Dr. Kumar developed fruit borer resistant brinjal and tomato, which were licensed to private companies. Sir, it was, I think, the time in 2006 around or 2005 that this was happening, and Dr. B.S. Parmar used to be the JDR. Yeah. So that was the time. And pod borer resistant pigeon pea expressing 2 codon modified Bt genesis is currently undergoing field tests. So even right now, he is very active. Dr. Kumar has published over 150 research articles, books, and book chapters, and he has 3 patents on codon modified and chimeric Bt genes. And he is recipient of many prestigious awards, notably Mahindra Krishi Smriti Award of 2011, Best Institution Award of ICAR 2010, and Recognition Award, National Academy of Agricultural Sciences 2005, National Bioscience Award, Department of Biotechnology, Vasvik Award for Agriculture and Industry, and he is Fellow of National Academy of Sciences India and NAS, and also Humboldt Fellow, besides many Young Scientist Awards to his credit. He is serving as editor, GM Crops and Food, USA, and secretary in Society for Plant Biochemistry and Biotechnology, India. Thank you very much, sir, for gracing this occasion. And now floor is all yours, Dr. Monica.
Thank you so much, ma'am. So as convener of the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentations. For session 4, as already briefed by Dean ma'am, we'll be having 3 presentations from the professors of the following discipline in the following order: biochemistry, to be followed by molecular biology and biotechnology, and plant physiology. For all the 3 disciplines, respective time allocation is based on the number of students receiving degree, and the professors will highlight the research outcomes of both MSc and PhD students based on the identified theme area of respective disciplines. For the information of the chair and the House, the respective time allocation has already been communicated to all the professors, and it's my earnest request to kindly wrap up in the given allotted time. For the ease of the presenter and as an indication of wrapping up the presentation, yellow light will blink 2 minutes prior to the allocated time, and a red light will blink at the end of the time. So with this, I now hand over the session to respected chairperson for conducting the further proceedings of the session. Thank you so much.
Thank you, Dr. Anupama. Of course, at the outset, I should thank Dr. Srinivas Rao, Director of IARI, for giving me this opportunity. And I also thank Dr. Monica and then Dr. Shruti. And good evening to all of my erstwhile colleagues and friends and whatever. So we are going to start this session of this day, the last session of this day, comprising presentations from physiology, biochemistry, and biotechnology. By habit, I started physiology, biochemistry, and biotechnology because I started as a botanist or plant physiologist. Then I have done biochemistry, and then defected to plant molecular biology and biotechnology. But the real sequence today is biochemistry, molecular biology and biotechnology, and plant physiology. And in my view, all three are an integral whole. There is absolutely no distinction between these disciplines in the current 21st century. Maybe in 19th century or 20th century, they were different. So let us consider this whole session as one. And without losing any time, let us first listen to the professor, molecular biology and biochemistry. Biochemistry, I welcome the professor of biochemistry to make his presentation. And as Dr. Monica has already mentioned, please stick to the time and the guidelines for the presentation. Thank you.
Honorable Chair of the session, Joint Director Research Education and the Dean IRI, the convener and co-convener, and Director NIPB, Head of the Departments of the Biochemistry Physiology, faculty members and my dear students. So it's my proud privilege to be presenting the significant educational achievement of the discipline of biochemistry. So before I present the significant educational achievement of the biochemistry, I would like to give brief stats about the biochemistry discipline. So as you can see that we are total 12 faculty members. So out of the, as far as the students they are concerned, we have 4 MSc and 40 PhD students at present. The total courses which are offered, 16 courses they are being offered. And this time the degree recipients, they are 9 from MSc and 2 from the PhD. So here I am presenting the list of the students who will be receiving their degree in the forthcoming convocation. So we have 9 MSc students who will be receiving the degree, and these are the titles of the thesis on which they have worked. And all the titles, they are addressing the various socially relevant problems of the nation. And as far as the PhD students they are concerned, there are 2 degree recipients, Ms. Meenakshi Datta and Mr. Nandal Meena. And theme areas of the students' research. So these can be basically divided into 3 theme areas. The first one is development of the nutritionally enriched food products for improved human health and the well-being. And the second one, it is the developing industry relevant and the commercially important biochemical methods and the tool. And third one is the improving yield and the abiotic stress tolerant of the crop plants. So in the first theme area, we have 1 PhD and 6 MSc students. In the second, 1 MSc student. In the third one, we have 1 PhD and the 2 MSc students. And as you can see here, that the traits which are being addressed, it is the mainly the millet protein isolates, then you have pulse protein isolates, and then you have the quality pigeon pea proteins. And in the second theme, we have the development of the glucose nanosensors. In the third theme, we have the seed priming for mitigating the drought stress, and then the melatonin treatment for enhancing the rhizosphere soil microbiome, and then seed quality under the drought stress.
So coming to the first theme area, that is development of nutritionally enriched food products for improved human health and the well-being. So as I have listed before that, in the first one, the millet protein isolate blends, they have been developed, and this is the extraction protocol which they have developed. And then the processing methods, they have tried 3 different processing methods, and out of these 3 processing methods, the infrared processing method, it has been found to be very, very effective. And then what they did was that they took the millet proteins from different sources, and then they developed the blend in the ratio of 3:3:2:2. And what they found was this blend of the millet protein isolates, they have the better protein digestibility, solubility, and the higher content of the essential amino acids, and then improved functional properties. They were also noticed, as you can see from the emulsion capacity of these blends. And the second student, Mr. Mansoor Shaji, he's worked on the pulse protein isolates, and these are the objectives of the study which he conducted. So again, this study, it was very similar to the millet protein isolates, but here the pulse proteins, they were being used, and also they developed the extraction for the protein isolates, and then the processing treatment. In this case, they found that the hydrothermal treatment, it was found to be very effective. And then they developed the pulse protein blends using the chickpea, green pea, soybean pea, and the lentil. And another good observation which they noticed was that the storage of these treated blends, it was found to be having the very high antioxidant activity, and then the less comprehensive peroxide value was there, which indicated that the enhanced shelf life at the ambient conditions. And the third thesis in this area, it was by Ms. Meenakshi Datta. She presented her work in the ICAR medal presentations yesterday. And these are the objectives of this study. So again, the emphasis, it was to have the pigeon pea proteins with higher quality in terms of the essential amino acid as well as for the digestibility it is concerned. And then they tried different processing methods to enhance the digestibility of the proteins and to have the higher quality of the proteins. And they determined the protein profiling using the SDS page, and what they could notice was there are certain anti-nutritional factors which were absent, which indicated that these processing treatments, they were quite effective. And then they found that the bioavailability of these proteins in terms of the amino acids, especially particularly this neutral and the polar amino acid, it was also enhanced. And there was an enhancement in the levels of the beta turns, so which is an indicator of the enhanced digestibility as well as the solubility of the proteins. And the next student worked on the carotenoid enrichment of the chickpea sprouts. So as you know, the carotenoid, it is a very, very useful bioactive compound which is required for the human health. And they tried to develop the products which is having the higher levels of the carotenoids, especially the chickpea sprouts they used, and they did the carotenoid profiling and estimated the total phenol content. There was an increase of 4.1-fold as far as the total carotenoid content was concerned, and 4.4-fold increase in the total phenolic content. And apart from these carotenoids and the phenolics, there was also enhancement in the levels of the antioxidant activity, iron, zinc, phytic acid was reduced, and then amino acid content, it was also enhanced. And another thing which they noticed was these products, they were having the very high LDL oxidation inhibition activity and also the anti-diabetic activity. So next student worked on the micronutrient and vitamin enriched nutrimixes. So they used different sources of the protein. The germinated soybean powder, it was used, then hydrothermal treated pearl millet powder was used, and the processed carrot powder was used, and they mixed these 3 powders in a combination of 3:1:4, and then based on the solubility, dispersibility, and the water absorption capacity, so they decided about which combination is working best. And then the foam mat dried sample, it exhibited enhancement in the nutritional levels. And next student, Bidut Prava Nayak, she worked under the guidance of Dr. Archana Singh, and these are the objectives of the studies. And she worked on the mushroom, especially the shiitake mushroom, and she identified a shiitake strain, UN2, after screening the 26 strains of the shiitake, and this strain was found to be having the high levels of the retadenine, because this retadenine, it is a very important anti-hyperlipidemic compound, and very high levels of this compound it was found in the shiitake strain. And when they determined the beta-carotene bleaching activity and the DPPH radical scavenging activity, it was also found to be very high, so which could be the mechanism for the anti-hyperlipidemic activity of the this particular compound. And another thing which they noticed was that it is having the binding site for the HMG-CoA reductase as well as for the lipase enzyme. So that could be the reason for the reducing the levels of the cholesterol. So next student is from IIB Ranchi, and these are the objectives of the study, and he worked on the another bioactive, that is the DOPA, which is synthesized from the tyrosine. And they determined the level of the DOPA in the faba bean, and what they could find was that the level of the DOPA in the mature seed, it was less, while in the young leaves it was very high. So that means as the development goes, the metabolites, they are diverted towards the another secondary metabolites, and the synthesis of the DOPA, it is reduced. So that could be the reason for the lower level of the DOPA in the mature leaves. And another student, Mohammed Salman, who worked in the second theme area, that is the development of the biochemical tools and methods. And he presented his work yesterday in the IRI Merit Medal presentation, and he was awarded with the IRI Merit Medal. And these are the objectives of his study. And the problem which he addressed was, because still now what a problem which we are facing is there is no correlation between the predictive glycemic index and the inherent glycemic potential. And because we don't have at present the methods available for the precise assessment of the glycemic index, and he used the technique of the machine learning and combined it with the nanosensors and then the in vitro methods, and he could find a method which can give the perfect correlation between the PGI and the IGP. And this method, it was found to be having the very high specificity. There was no interference from the monosaccharides, and then he established the correlation between the IGP and PGI as well. So next student, Shakti Tangle, he again worked under the guidance of Dr. Archana Singh, and he worked on the seed priming aspects for mitigating the drought stress. And he worked on two genotypes of rice, Dagina 22 and Pusa Sugan 5. And as you can see here, that this in the first generation, they did the priming with the methyl jasmonate and then the ferrous sulfate and the zinc sulfate. And in second generation, when they analyzed the plants, they could find that the plants, they were able to cope up the drought stress as they were having the higher antioxidant potential. And the other biochemical parameters, they also favored to combat the stress. The next student, Arun Kumar, he worked under the guidance of Dr. Arna Tyagi ma'am, and he worked on the melatonin treatment for enhancing the rhizosphere soil microbiome. And what he noticed was that after the treatment with the melatonin, so there is a better communication between the microbes and the signals which are coming from the roots in the form of the root exudates. And that could be the possibility for the enrichment of the soil rhizospheres with the nitrogen fixing bacteria and these phosphate solubilizing bacteria. And that in turn helped in the enhancement in the yield. The next student worked on again the rice plant improving yield and abiotic stress tolerance of the crop plants using, and what he observed was that seed quality, it was compromised during the drought stress, but what are the mechanism underlying these changes which are happening under the drought stress, he wanted to decipher that, and with, for deciphering this aspect, he carried out the proteomic studies and the metabolite profiling, and from the results of the proteomics and the metabolite profiling, he could find that metabolites of the bran are involved in the flavonoid biosynthesis, unsaturated fatty acid biosynthesis, and metabolites of the grain are involved in the starch and the sucrose metabolism. And further, he noticed was the N22 exhibited upregulation of the differentially expressed protein associated with the glycolysis, starch, sucrose metabolism, and the defense, especially the gene encoding for the sucrose transporters, it was upregulated. So this was the conclusion of his study. The higher content of all the macronutrients in bran, it was observed decrease in the interactions between the starch and lipid proteins, because bran, it is a rich source of the lipids. So he found that in the bran there are interactions between the starch and the lipid that could be responsible for the lower digestibility of the starch. So overall, from the discipline of biochemistry, faculty members and the students, they published 85 research papers, and out of the 85, the 29 are having NAS rating more than 10, and 30 having NAS rating between 8 and 10, and 20 having NAS rating between 6 to 8, and only 6 having NAS rating less than 6. So this is the placement record of the students who received the degree in the last year. So most of the students, they have either joined as the assistant professor or the agriculture research scientist, and even there are students who are having 2 jobs at this particular moment. So they are already working as assistant professor, but they have also qualified for the agriculture research services 2024. And these are some of the honors and awards conferred upon the faculty members. One patent has also been granted to Dr. Vinuta Chi. And these are the awards and the honors bestowed on the students. Mainly the best poster award or the best oral presentation award. And some of the students, they also got the opportunity to have the exposure to the foreign labs under the auspices of the National Agriculture Higher Education Project. And then our department, it is also a center for advanced faculty training, so we also conducted one training program. One training program, it was conducted under the ST Karyashala program and Skillify under the SPBB. And then we also organized one international conference in collaboration with the Society for Plant Biochemistry and Biotechnology. So with this, I pay my sincere gratitude to Director IRI, Dean and Joint Director Education, Joint Director Research Extension, Associate Deans, Coordinator School of Basic Sciences, Head Division of Biochemistry, Professors of the all the basic sciences disciplines, funding agency, and all the students and the faculty whose work I present now. Thank you so much.
Thank you, Dr. Anil, for a very lucid presentation. And I also appreciate your time management. Thank you. So next I invite the Professor of Molecular Biology and Biotechnology, Dr. Jasdeep Padaria. Jasdeep Chatrat Padaria. She was Jasdeep Chatrat when I joined the center, you know. Later she has become Padaria. You all know that.
Honorable Chair of this session today, Dr. Anand Kumar, and I would like to say our very own Dr. Anand Kumar, respected Director NIPB, my dear colleagues and dear students. I see a number of them have come from our center. So I will be presenting significant educational achievements in molecular biology and biotechnology. I will be representing 3 centers. One is IRI at NIPB, then this Ranchi hub of IRI, which is at IIAB, and Raipur hub of IRI, which is at NIBSM. So next. The current status of the student and faculty, it is slightly on a very heavy load on the faculty. We have UG, BTech Biotechnology, and 43 courses are being taken care of right now. PG, we have 28 courses, and as you can see, the faculty in all the centers combined is 42 plus 3, which is a very bad ratio. But anyway, we are trying to do our best. The degree recipient this year, we have total all centers together, MSc 19, PhD 9, a total of 28. So I will be presenting the research achievements of 28 students. Next, please. These are the list of students who have done. Over here, just one attention I want when I have written there 3F plus 6M. I have just tried to female and male. Somehow, this year, the number of females is on the lower side as compared to males. Males are on a higher side. Though no particular correlation is there, but just a point to be made. Similarly, at Ranchi also we have 2 females plus 3 males, and Ranchi and Raipur are having only MSc. PhD students are from NIPB. In case of PhD also, we have 4 females and 5 males, almost equal. Now, as far as the theme areas is concerned, the theme areas overlap a lot, but I have tried to divide them on the basis of crop. First is search and deployment of genes for stress tolerance and yield traits in rice. Second is molecular mechanism for stress tolerance and nitrogen uptake in wheat. Third, developing biotic stress resistance in mustard. Stress tolerance and quality traits improvement in pulses. Pulses I have combined together chickpea, pigeon pea, mung bean and winged bean. Stress tolerance, genetic diversity and post-harvest quality in vegetables and fruits, where it is potato, okra and strawberry. Then finally, it is exploration of microbial genes for crop protection and productivity enhancement, which is the BTG. Bacillus thuringiensis has been taken up to identify a number of microbial genes. So the first area we have abiotic stress tolerance, drought and heat, biotic stress, rice blast, bacterial blight, and third area is grain number. These are the sub areas under the main theme area. The first student in this area is Dr. Babli Kumari. She has worked with Dr. Amita, and her study was concentrating on identification and validation of robust candidate genes identified from the major heat tolerance QTLs. These 3 QTLs were taken, and they tried to identify the genes. एक second, pointer. And these genes are 32 QTL specific genes were, they showed SNPs in genic regions, and these were functionally validated, of which 16 were differentially expressed. One of the most promising gene was taken up for cloning and functional validation, and it had shown good response. It showed that in IR64 the performance was good, but Nagina 22 was a better performer for this gene. The second student was Dipesh Kumar, identification, characterization and functional validation of drought responsive microRNA in rice. So he first carried out, sir, just 1 second, I will point there. So they had, in silico studies were carried out, and 19 target genes were for 1 microRNA were identified in the 3 major QTLs for drought tolerance. Similarly, the global database was searched, and a number of DEMs were identified, of which 2 microRNA mRNA modules were identified and were being studied further. Differential expression studies were carried out, and then functional characterization of 1 very highly performing microRNA 3979 was taken up, and it was shown to perform good, and molecular characterization of the transgenic lines are being done. They have successfully shown that this is functionally validated. Now we move to biotic stress in rice. This is a MSc student, Pooja, who has worked with Dr. Amol Kumar Solanke, and they were targeting to screen multi-gene CRISPR-Cas based mutant lines for the presence or absence of tDNA. And second objective was to identify single, double and triple mutants for 3 main genes. And the study showed that trans-free using Cas-PCR, they could identify that genome edited lines were blast resistance as compared to the control TP309. All 3 genes are there in one genome edited line. In an era of genome editing, we have moved further, and over here we are taking care of rice with respect to bacterial blight disease. Over here, Dr. Ritu Rai with her student Anurag worked with her, and they have been able to identify 5 different variants of TAL7 which were custom designed and generated for complementation into a non-virulent zoo strain for studying the significance of repeat backbone variation in virulence of TAL effector. This study would be used for genome editing of rice for this generating resistance for bacterial blight. This, the last is in this rice is the one QTL GN 4.1 was studied, and it was narrowed down from 385 kilobase pair to a 70 kb region, which was present in all the homozygous recombinants. The study was mainly targeting to study the promoter also of this QTL, and this particular gene which was identified in this QTL, and a CRISPR-Cas construct for knocking out the particular gene was designed and developed. Knocking out of this gene would result in conclusive evidence of its role in high grain number per panicle phenotype. Next. Now we come to the wheat, and in wheat, all the theme area we have abiotic stress, heat and salt, biotic stress, pink stem borer, and nitrogen uptake. So this is how I have divided. First we come to, we just saw that Dr. Dahuja was talking of melatonin. Over here also seed priming with melatonin has been done by Shazia Sultana under the guidance of Shamishta Balthakur, and they have found that this has resulted in differential expression of genes in prime plants during high temperature stress at generative stages, and they have also identified some genes which are differentially expressed. Simultaneously, they have also carried out transcriptome data analysis, and they have found that the priming has resulted in the the plants performing better under heat stress as compared to control. This Ahmed, he was a foreign student from Ethiopia. He worked with Dr. Kanika, and the target was to quantitative trait loci to identify related to salt tolerance at the vegetative and reproductive stages in heat in inbred lines of the population between 2 parents, HT2851 and Khurchia 65. Khurchia 65 is known to be salt tolerant. He had carried out screening and phenotyping of the population, and he could find 30 SNP markers which were significantly associated with QTLs related to salt tolerance trait, and a number of genes, mainly 3 genes, were significantly upregulated in the leaf and root tissues of Khurchia 65 under salt stress tolerance. This work will be now taken up further to further validate these genes which were differentially expressed. This research is by an MSc student, Hari Chandana. She worked with Dr. Subodh Sinha, and they had the cloning and sequence characterization of gene NRT 2.5 of red wheat from and further characterize the upstream region. Actually, they have taken the genotype which is nitrogen uptake efficient and found 3 alleles of this gene which were differentially expressed for drought and tissue-specific expression, and the genes were also, they were cloned, and they have found that some of the stable lines carrying all the 3 receptor constructs are there at T1 stage. Further characterization will be done for these transgenics obtained. This work is from NI Biotic Stress Management Institute at Raipur, and over here they have done studies on the effect of silicon amendment on pink stem borer of wheat under changing climate scenario. They found that the presence of silicon combined with elevated temperature resulted in reduced larval survival and development, and silicon supplementation increased activity of defensive enzymes, which differential expression studies were done, and these biochemical changes indicate that silicon enhances plant defense mechanism against pest infestation and environment stresses. In an era of genome editing, it is more important to have a negative regulator, for which the student Akash Nigam, he worked with me, and we tried to clone and sequence the identified negative regulator for heat stress tolerance. A number of genes from the available DNA sequencing transcriptome data were taken up, differential expression studies carried out. Like, it was found that the gene was down-regulated in Raj, which is a tolerant genotype, and up-regulated in HD2967, which is a sensitive genotype. So it can be a probable negative regulator. The gene has been cloned, and now this is being functionally validated by the next student in wheat. The third area is mustard, and in this we have biotic stress, aphids, and sclerotinia stem rot. So these two, we have one PhD and one MSc student from this in this theme area. Asha Kiran has worked with Dr. R.C. Bhattacharya, and she has found that in terms of tissue-specific expression of the effector encoding genes are much higher in head segment, which include salivary glands. Gene expression of these selected effectors are host-inducible and putatively signify involvement of the effectors in host-insect interaction. Activation of insect effectors, host-specific, and presumably determines outcome of the interaction. So this study is based on effector molecules. Then in bioassay studies, they found that one of the effector, E3, showed suppression of different jasmonic acid, salicylic acid, and peroxidase and downstream defense genes at a dose of, the dose was also standardized, 20 to 40 nanogram. LE3 can be used for identifying host susceptibility factors or other genes related to this work. This is a MSc student, Shreyas D, and he has worked to analyze the transcriptomic data available and identify the susceptibility gene for stem rot disease in brassica, and he cloned the susceptibility gene and designed and developed CRISPR-Cas construct for editing. Now, the theme areas is stress tolerance and quality trait improvement in pulses. We are taking care of the students who are getting degree will have done research in 4 major pulses: chickpea, pigeon pea, mung bean, and winged bean. And different traits have been taken up. For example, in chickpea, it is seed protein content, fusarium wilt, and growth promoter and fertilizer. In pigeon pea, it is non-synchronous flowering, methylation, demethylation. Mung bean, yellow mosaic Indian virus. In winged bean, nodulation and anthocyanin content. Now, PhD student Gopal, he worked with Dr. Pradeep Kumar Jain, and the study was involved to identify and characterize genes and microRNA and their corresponding target genes associated with seed protein content in two genotypes with contrasting seed protein content. The two genotypes taken were FG212 and ICC8397. ICC8397 is high protein genotype, and they found that amino acid transporter genes were on a high expression in low genotype of chickpea, and it was suggested that this may be because increased amino acid transport may lead to enhanced redistribution rather than retention within the seeds of the potentially limiting seed protein content. In continuation of this work, he also did identification of the circular RNA and their targets associated with seed protein content, and it was found that first they identified few target RNAs, around 7 were identified, of which 3 were differentially expressed, and they found differential expression, which now they will be cloning and trying to go ahead to further analyze this. The circular RNA 3 was the only circular RNA that was encoded from storage protein specific to legumin gene. This work is again from IIAB. Subramaniam, he has worked on molecular response of bacterial endophytes mediated defense against the Fusarium wilt in chickpea. And over here what he has done was 94 bacterial endophytes were isolated, and they were studied for their different 16S. They were identified and characterized. 16S ribosomal DNA sequence was done to identify the bacterial, and further they found out that treatment of these endophytes led to the plants becoming tolerant to Fusarium wilt. And molecular studies show that there was enhancement of a number of enzymes as well as total phenol content. Besides that, they also carried out differential expression of few genes, and they would try to correlate it with the endophyte having response on the wilt in chickpea. Amisha is from IIAB, and she has worked with Dr. Sarkar, and she has tried to get a molecular insight into the fungicidal action of green-silver nanoparticles against the chickpea wilt caused by Fusarium oxysporum. And her studies showed that at the molecular level of 300 ppm, this nanoparticle developed led to upregulation of stress and defense-related genes. So this study has to be carried out further to come to a conclusive result. But genome differential expression studies have given them a lot of leads, which they will be taking further. This is understanding the nanosilica-induced molecular regulation in chickpea. This was again done with Dr. Sarkar by Tarun Kumar, and they have tried to first synthesize and characterize greenly encapsulated silica nanoparticles, and then deliver the nanosilica and try to see its effect on morphology and physiology in chickpea, and also did some differential expression studies to hit upon the gene which may have a clear-cut role in this. So, this is related to pigeon pea, and this work is done by Sogoto Bhattacharya with Dr. Debashish Patnaik, and his study was functional characterization of homologs of tomato self-pruning gene for the role in flowering in pigeon pea. What he did was he tried to study the expression pattern of this SP5 gene homologs in different tissues and developmental stages of pigeon pea, and then functionally validate the gene, the ortholog of SP5 gene. And he found that upon
identified 412 floral orthologs in pigeon pea, and of them he could hit upon 12 key floral orthologs which he took for differential expression, and then the identified ortholog of SP5G was functionally validated by two approaches. One was constitutive expression in tomato and the other was silencing in pigeon pea. In case of tomato, they were able to get very good transgenic lines which were also screened, and they found that it was performing well, and it confirmed the role of this gene in delaying flowering and promoting vegetative growth through constitutive expression in transgenic tomato. Priyanka Kumari also worked with Dr. Debashish Patnaik, and she carried out studies on molecular characterization of methylation demethylation dynamics in pigeon pea, and she tried to identify using genome-wide identification technique genes which were methyl transferase as well as demethyl transferase. A number of genes were identified which were located on different chromosomes, and they were differentially expressed. And they found that these genes were showing differential expression with respect to their performance in different genotypes, different tissues. Then a human FTO gene was synthesized in the lab and codon modified for plant gene transformation and binary vector construct was made, and this they did transformation in pigeon pea and potato using this gene, and they found that in case of potato there were around 10 lines of transgenic plants, and they found that they were performing well, and the transgenic plants were bigger as compared to control, and also the number of microtubers were much more in transgenic plants. In case of pigeon pea, also transformed lines show more growth during balancing stage, and as compared to control plants, the plants are in the field and their effect seems to be pronounced. Now we move to Moong bee. This work is done at Raipur by Dr. Sivalingam and by Rashmi Kumari under the guidance of Sivalingam. And what they did was they tried to find the key host factors involved in pathogenesis of Moong bean yellow mosaic virus, and they found that there were different genes which were upregulated during early infection and a few at late infection. They cloned 2 of these genes and they tried to see the homology of these genes. ERF showed homology beyond the leguminous family, while LHY showed strong similarity with leguminous species. This work is in IIAB by Jamshida, and she worked with Dr. Kishore Krubhavan, and this is on winged bean. And what they did was they had to identify genes responsible for anthocyanin content in winged bean. So they tried to identify a few genes, did expression analysis, and hit upon the genes which were the potential candidates responsible for anthocyanin content in Moong bean. In continuation, in Moong bean, this was to identify expression analysis of genes responsible for nodulation in winged bean, and they found that first they did isosequencing, hit upon a few genes, 14 genes were identified, and they tried to see the expression of these genes in different legumes. And real-time analysis showed that 9 were upregulated in winged bean, 3 in faba bean, and 5 in chickpea. This is again MSc student, and he is from Raipur, and they found that this is Devjan Basu Roy, and he worked with Mallikarjuna Jeet, and over here it is slightly different. Now we are talking of ochra germplasm was screened for ochra yellow mosaic virus, with respect to the transmitter also, whitefly, and they did artificial screening of okra germplasm for resistance to yellow mosaic virus and characterized the germplasm, and they found one accession which was very highly resistant to the yellow mosaic virus, and it had high trichome density, and this germplasm also disease-free condition as well as very low number of whitefly populations were infesting on this germplasm. So they try to identify the key factors which contribute to this resistance. Now we move to the fifth, which is stress tolerance, genetic diversity and post-harvest quality in vegetables and fruits. Okra we have already talked about. I think there has been some problem in these slides. So I'll talk about potato now, cold-induced sweetening. This work has been done by Krishnan and Paul under the guidance of Devashish Patnaik, and they found that they were targeting two genes, UDP, glucose pyrophosphorase, and vacuolar acid invertase genes, and they could develop hairpin RNA gene construct and binary vector cassettes were spread, and the transgenics developed. And like you can see over here, after 30 days of post-cold storage, the control chips had turned brown, while the transgenic lines are still yellow. Further, this is Dr. Krishnanan also developed a novel recombinant CRISPR-Cas vector. He codon modified it so that it can be used in a number of plant systems, and in two systems they have validated it in tobacco and potato, and this construct is totally patent free and can be taken up once the patent is granted, it can be used extensively in the ICAR system as we are moving from transgenics to genome editing. This is studies on genetic and population dynamics in okra in Chhattisgarh, and over here they found that there were 7 distinct halophytes of leafhopper with 2 distinct clades, and they tried to just study the different genetic diversity of these, and their results show that this leafhopper population is under pressure and going through continuous genetic changes, and it may lead to development of biotypes in future. Results can support in formulating transgenic strategies for control of leafhopper in okra. Studies on genetic diversity, and again this study so this over here there's a problem. I will just read the objectives. I think from system to system there has been. This study is based on regeneration protocol in strawberry. This is from IIAB, and Ankit Patel has done the standardization protocol, and he has found different explants were taken, and he has got some very good results, and maximum regeneration percentage was obtained from runner shoot tip explants. And they also felt that there was no need of pre-hardening in case of strawberry. The student has ultimately also landed up in a startup where he is taking care of tissue culture of banana. Now we come to Bacillus thuringiensis. Over here this student had presented for MSc gold medal, and he had tried to screen Bacillus thuringiensis isolates for their antifungal activity against Fusarium oxysporum and Fusarium graminearum, and cloning of the gene was done. So different genes were identified, and their phylogenetic studies were done. There were a number of submissions to NCBI, 16S, as well as 3 chitinase genes were submitted to NCBI. This is another student who has worked with Dr. Sarabjit, and he is Jagbandhu Khan, and he also did, but with PT isolates. The aspect over here was entomocidal genes and evaluation of insecticidal efficacy of these isolates towards lepidopteran pests. They also tried to see some growth promoting response from these isolates, and their studies identified Bt isolates with dual insecticidal and fast growth promoting potential, and they can offer promising application in agriculture. As far as research publication from the discipline is concerned, this is from NIPB. We have 107 publications and trainings, practical manuals, book chapters, books edited, review articles with a good NAS rating. Student publication is also good. We have around 48 publications from students who are getting degree this time, and it is the NAS rating is also reflected over here. We have good NAS rating publications. Now, student placement. The student placement scenario with respect to MBB is not very bright. As I have already communicated to you also, sir, I have communicated to madam also, that this has to be taken care by the people at the top, how we handle this. Anyway, still for PhD, 4 students have joined research and training, 3 are targeting postdoc, 1 has joined as patent officer, GOI, and the foreign student has gone back to his country. In case of MSc, we are slightly better because most of our students, except 2 from NIPB, are pursuing PhD at different level, different universities, and some are preparing for the competitive exams. Just 2 minutes, I'll finish. These are honors and awards of the students. Most of the students are able to crack DBT, JRF, CSIR, NET, UGC, NET. So which clearly shows that they are bright students, but with no seats in ARS, I think they are slightly getting demotivated. And this is BTech students are really good. They are active and they participate in different activities. We have a patent from one student's research work, 2 patents. We have a web page developed by one student. We have an app developed by a student where you can search for all the genes related to chickpea. Honors and awards in the faculty. These are few which I have mentioned here. And there are also foreign visits by our faculty. Then besides teaching, we have other activities. We have different occasions. We had one session where Dean Madam had really supported us with the net facility, and we had Khurana scholars to motivate the students to apply for such things. Regular orientation program, workshops as and when asked by PG school, we nominate students. This is different trainings, SPP sponsored, SERP sponsored, school students visit on a regular basis, NAHEP sponsored training, which are taking place at the center. And National Science Day, our students had participated with IRI. Besides that, we also have some fun, Holi celebration, freshers welcome, felicitation of degree recipients, and student farewell to the ex-Board of Studies member, and welcome to the new one. So I acknowledged Director ICAR, IRI, Director NIPB, Dean and Joint Director Education IRI, Faculty MBB of all the 3 centers, students, administrative staff of TGS, all supporting staff, professor section, MBB, and whosoever has contributed in some way or the other towards this presentation. But this is not working, sir. This has to be taken care of. My time exceeded because of this. You need to come. No, I didn't say that just to get us.
No, but yeah, conveners had to take this, care of it. And tomorrow, let's be more careful. Okay? Never mind.
Thank you, Dr. Jasdeep, for extensive coverage of your student research, Now I invite a professor of plant physiology to make the presentation of the students' research in plant physiology.
Before the next, sir, may I kindly submit one thing? Because otherwise I may forget, although I have passed on the message. I request all the professors who may have also joined online. This year I have planned that kindly pass on the details of the placement profiles of the students, placement details and the statistics of the students in year 2024. And which will be displayed for the entire graduate school as a hoarding and as a big poster on the convocation day. So kindly do this and by tomorrow, so that Dr. M.R. Khan will be making this entire exercise. So kindly do that. Thank you.
Can I start, sir? A very good evening to one and all present here. Respected Chairman, Professor Anand Kumar, Director NIPB, Dr. Bhattacharya, Joint Director Education, Heads of the Division, Convener and Co-Convener, all the faculty members present here, and my dear students. I'll be presenting the significant educational achievements of Division of Plant Physiology, and I feel honored to present these achievements in front of my teacher, Dr. Anand Kumar, because when we met at the start of the session, he said, Anjali, I thought you have retired. So, sir, I'm still here for a few more years. So, the faculty position at Division of Plant Physiology is that at IRI we have 16 faculty members, and we are fortunate enough to have 2 adjunct professors who are very distinguished scientists in their own field, and whose teaching and mentorship is helping all our students to excel in their academics as well as research. So in total, we have 14 MSc students and 41 PhD students. Some have taken temporary relief. And in 63rd convocation, we have 8 MSc and 4 PhD students who will be awarded degree this year. As far as the theme areas are concerned, I'll be presenting the work of the students on 2 basic theme areas, abiotic stress tolerance and plant ecophysiology. Under the abiotic stress tolerance, I'll be covering nutrient stress, high temperature stress, drought stress, combinatorial stress, and under ecophysiology, 1 MSc thesis on photothermosensitivity. So this is the list of the students who have worked in different areas with the title of their thesis given. 8 students are, as I said, receiving degree this year. So I begin with Bailang Chain, who worked under Dr. Renu Pandey's she was the chairperson, Dr. Renu Pandey. So as we know that the northeast soils are acidic in nature, and because of that acidity, there's iron toxicity and low availability of phosphorus in these soils. So Bayer, she screened 82 NER rice genotypes for iron toxicity as well as low phosphorus content, and she identified few genotypes which could serve donor and which were efficient ones. And these efficient ones, they had high ferritin content. I mean, they could store ferritin in their vacuoles, and also they had showed increased expression of high affinity phosphate transporters in the roots. Then Harita SI, she carried out bioinformatic analysis of amino acid permease family genes. Amino acid permeases are involved in the uptake and transport of amino acids from the soil as well as the source tissues. And she also carried out the in silico expression of these genes in response to low nitrogen, and also developed a CRISPR-Cas editing construct of few genes of ARP family. So she found that ARP3, ARP5, and ARP11 haplotypes she could identify that from the database, and she found that ARP1 and ARP16 had high expression in the leaves. Also, she could make the construct of 3, 5, and 11 and could successfully clone it. Srihari V. Santosh, he screened or he phenotyped 198 rills of the population derived from HT3086 and HI 1500, and he also found donors which were tolerant to drought stress, and he found that few traits like colluctile length, shoot-to-root ratio, seed weight, and harvest index were the key traits which could be used for the screening purpose. Then Thok Chom, under my guidance, she carried out physiological and biochemical basis of amelioration of heat stress through nitrogen management and wheat. She conducted her study under 2 high night temperatures and 1 high day temperature, in which she sprayed urea, 2% urea, at post-anthesis stage, and she found that foliar nitrogen spray helped in maintenance of yield under both the high night temperature, but it did not work under high day temperatures. And carbon to nitrogen ratio was significantly low under high day temperature conditions. Then Unti, she screened around 160 Brassica gensea genotypes and identified donors for drought tolerance, and based on few indices like MGIDI and FAI and SH index. Then Niteti Siddharth Kumar, under the guidance of Ruchi Bansal, he also phenotyped a diverse panel of 243 lentil genotypes for seedling stage drought tolerance, and he also identified a few donors and 64 stable MTAs in control and 72 in drought condition through the GWAS. And then the gene annotations of these stable MTAs led to the identification of few genes in this study. This study was conducted, these 2 studies were in fact conducted at Niazam, Baramati by 2 students who were guided by Dr. Gurumurthy. And Kritika brilliantly presented her research work for the gold medal presentation yesterday, in which she did a characterization of common bean genotypes in reproductive stage, again under a combinatorial stress of drought and heat condition. Then she identified these few genotypes which were tolerant to drought as well as heat stress. And like my previous presenters, they have talked about melatonin, and Sir said we are a one group. All 3 divisions, they are one group, and we also had a study on melatonin spray, where melatonin improved the yield under stress condition, heat stress condition. Then he identified 2 spectral indices which improved RWC prediction accuracy. Then Apurva, also under the ecophysiology theme area, he took up a study in 3 seasons, Kharif, Rabi, and summer season. And in the chickpea genotypes, 8 genotypes, he evaluated their photothermo sensitivity and identified these 2 genotypes which were highly photoinsensitive and could be taken across all the seasons. For the PhD degree recipients, we have 4 degree recipients this year, and most of them are working in the area, have worked in the area of abiotic stress tolerance. Vijay R., he studied the phosphorus use efficiency in bread wheat genotypes with the rationale that phosphorus availability is a major challenge for sustainable wheat production. So he phenotyped under 2 experimental conditions, a set of 304 diverse wheat genotypes. The conditions were hydroponics as well as soil. So from the hydroponic study, he found 77 stable MTAs for phosphorus use efficiency traits, and also identified few putative candidate genes which were related to root system architecture, phosphorate uptake, transport, and utilization. And in the soil experiment, he identified 12 low phosphorus stress tolerant genotypes which exhibited lesser reduction in the photosynthetic rate, stomatal conductance, and yield. Jiva's analysis led to 3 important major MTAs, which were identified for, again, photosynthesis, yield, and other indices. Now I'll be presenting the work of Sukumar Taria, who was awarded gold medal yesterday, and this elegant study was on stem reserve mobilization and stay green traits for each stability in, again, combined heat and drought stress. So the functional markers, molecular markers of stay green and SRM are not available, and also both of these traits contribute quite a lot under stress condition to the reduced yields. So he phenotyped 220 wheat rills of the population derived from 3086 and 1500, HI 1500, and also phenotyped these rills for SRM. Then the results showed that there were 44 QTS which represented the stem reserve mobilization. These are the 2 donors which he identified from his studies, which were superior based on the various indices he used. For stay green and SRM taken together, there were 14 QTLs which were identified. 11 were mapped for SPAD value, 2 for leaf senescence rate and stem reserve efficiency, 1 for stem reserve efficiency. And he concluded from his study that stem reserve mobilization works better under drought rather than heat. And the gene prediction from these QTLs led to the identification of these genes, which are highlighted in red, and they contributed to mobilization of carbon reserve under stress condition and also to the regulation of leaf senescence. Devika worked with me and she worked on high night temperature. Again, she used nitrogen application as a mitigation strategy. She carried out a field study for 2 years to identify if varying the nitrogen or rescheduling the doses had any effect on the rice genotypes. She took short duration and long duration genotypes. Under the short duration genotypes, she found that the medium nitrogen dose with equal split nitrogen application could give similar or higher yield. And under long duration rice genotypes, higher application at heading showed at par value with the control condition. So out of these, she selected 2 short duration genotypes which were contrasting in nature, Vandana and Nagina, and conducted the experiment under high night temperature chambers with an interaction with the nitrogen application. And her results showed that the net rate of photosynthesis improved with split application of nitrogen, and the real reduction in Vandana was correlated to the number of grains as well as test weights. She did biochemical analysis of few nitrogen metabolism enzymes and also the starch metabolism enzyme, the results of which are listed out here. And this is the last PhD candidate who in this series, Adip Das, who worked on phenotyping and identification of superior donors for high temperature tolerance in rice. He started his work under the guidance of Dr. Madan Pal, but after his superannuation, he continued his work under the chairmanship of Dr. Vishwanathan. So he's fortunate to have 2 distinguished plant physiologists as his guide. So his rationale for his study was that the reduction in yield due to high temperature in rice is due to higher spikelet fertility, and he wanted to identify some efficient genotypes and introgress and genes, and later on introgress these candidate genes into the cultivated varieties. The work is carried on after his submission of his thesis. So the results of his work showed that high temperature stress significantly reduced spikelet fertility and panicle weight by over 50%, and he found 2 accessions which had higher spikelet fertility under the heat stress conditions. A total of 17 QTLs were found associated with spikelet fertility, 19 for panicle weight and high temperature stress conditions, and he identified a few candidate genes and saw their and observed their relative expression under the heat stress condition, which varied with the high temperature stress. Then he also carried haplotype analysis of triose phosphate transporter gene, and he found that out of 3 SNPs, 2 were non-synonymous, and 3 haplotypes for these 2 SNPs were found in all the accessions. The publication of plant physiology for this year, we have 53 publications, 14 from the year is 2024. So 14 from the student side, rest there were 4 training manuals, 20 conference papers, 17 book chapters, and popular articles. All these research articles have found place in very prestigious journals with high impact factor. These are the few publications of the students who have passed out last year and also who will be receiving degree this year. We have a good figure to show for the awards and recognition of the plant physiology students. One of our bright students, Dr. Sandeep Adri, he was awarded with IRA gold medal last year. He worked under the chairpersonship of Dr. Lakshmi Sathi. Then all the 5 seats of ARS were secured by our students. And Sukumar Taria, who received yesterday's gold medal, he was also awarded Young Scientist Award. Most of our students qualify ICAR, SRF, CSIR, JRF, and many have been awarded in the symposiums also, national conferences also. And Sri Hari was also awarded Best Thesis from this foundation. And I feel proud to mention before the audience, august audience, that our students have also excelled in sports activities and also other curricular activities. One of our students, Navodaya, she was a winner in most of the sports events which were conducted last year. And Hari Madhav, he was the winner of the IRI Alumni Logo Competition. And this logo will be carried forward for times to come. So this is an important contribution from a student of plant physiology. Awards of the faculty, as far as it goes, we always take pride in presenting the laurels that are awarded to our very distinguished faculty, Dr. Vishwanathan. And he also got NAS Recognition Award this year and is also ranked amongst the top 2% scientists of the world. And Dr. Archana was also was awarded the Best Poster Award this year. Dr. Redu Pandey went on a very prestigious OVDF fellowship, faculty visit to Purdue University. And these are a few pictures of the lectures and the lectures by the adjunct professors, and the interactions were very friendly also, and getting even a small flower from Dr. Khunuja means a lot. So I captured these photographs and I'm presenting it here. And also Dr. Kadambodh Siddiq, whenever he visits India, he makes it sure that he makes maximum presentations for the students, for the benefit of the students here. So they are also benefited by his presence in the division. So these are glimpses of some of the activities that were held in the division, the study tour, and thanks to Dean Ma'am for giving us a generous amount for this study tour. The students were really benefited visiting a horticulture institute, because most of us are not working on any horticultural crops. Then workshop by ICAR alumni. We also held the first alumni week come 50th Foundation Day of Plant Physiology, where the students actively participated, and we share lighter moments with the students by holding many of the festivals together and Teachers' Day celebration and other games and sports. Our students also rule the stage. They make their presence felt in all the IRI functions. And as you can see, I think they'll be performing in the Convocation Cultural Evening also. So they'll be seen on all the function, whether it is Paras, Convocation Day, First Alumni Meet, Soil Conference, National Symposium. So you'll see them everywhere performing. So these cultural activities are also included in the growth of the students. Then these are the few trainings which were held in the division. In the end, I'd like to acknowledge all the support that is given by Vice Chancellor and Director IRI, Dean and Joint Director Education, Joint Director Research, Head Division of Plant Physiology, all the staff and students of Plant Physiology and the funding agencies who have supported their research. Thank you one and all.
Thank you, Dr. Anjali, for a very nice presentation. So, I would like to make a few remarks as the chairperson of this session. At the outset, I would like to profusely congratulate, compliment, and appreciate the work of scores of students and respective supervisors, these three divisions of the biochemistry, molecular biology and biotechnology, and plant physiology. As you also might have noticed, the research, whether it is done by students or scientists, by themselves with a battery of project staff, has all come around the central theme of molecular biology and biotechnology. As I mentioned earlier, it is an integral whole now. So whether it is, let's say, dissection of physiological traits like drought tolerance or heat tolerance or protein quality or whatever, in physiology or biochemistry and biotechnology, it is all now got integrated automatically. And another very important point I would like to make is, fortunately, there is absolutely negligible duplication among all these three disciplines or divisions. Although the science and technology are the same. So that is one thing which is very, very commendable. And when you really see the research division-wise, the students' research, for instance, if you take biochemistry, there are some very exciting research problems given to PhD students in particular reference. And some of these very exciting things are, the protein quality in pulses. And the other thing is the glycemic response and how you can monitor or measure this glycemic response by making certain biosensors. It's a very exciting thing. And same way, the involvement of melatonin in the rhizosphere microbiome and the association with the nodulation or expression of nodulation genes is very, very exciting. And the track record of the students in biochemistry is also very appreciable. 85 publications in total for students and faculty combined is really good. That brings me around to my premise I made sometime in 2007 or 2006 in NRC Plant Biotechnology, which is now National Institute of Plant Biotechnology. The premise is based on 4 Ps, and all my young colleagues and students, etc. Remember that. That is, these 4 Ps are publications, products, patents, and processes. That is what is expected of any scientist for that matter. It's not simply biology or physics or chemistry or whatever. So as far as we are concerned, physiology, biochemistry and biotechnology, the students and the scientists should excel either in one or more of these 4 Ps. It's not necessary or it is not mandatory that everybody makes a product or everybody makes a patent or everybody makes a process. But you take up 1 or 2 and you excel in that, and that's what I found very happily, very pleasantly. That's what I found in today's presentations. I have seen quite a few patent applications, very good record of publications, that too in high impact factor journals, as reflected by impact factor or NAS rating, and also certain products that are in the pipeline. So that's really good. It's really admirable. And one particular thing I just wanted to point out to Vesavi biochemistry is probably the students are not given topics related to anti-nutritional factors in pulses. Protein quality, I have seen quite a few projects are there for students, but the anti-nutritional factors which are very, very important in pulses like chickpea, pigeon pea, soybean are missing. Maybe the scientists are doing that with their project staff. I am not aware of that. Similarly, phytic acid. Phytic acid is a very, very important anti-nutritional thing. And some of the previously observed research on lipoxygenase in soybean, I haven't seen that, lipoxygenase related research. Maybe, as I mentioned, it is not given to the students, but scientists are doing it themselves in their projects, respective projects. When it comes to molecular biology and biotechnology, I can see there is tremendous amount of burden on faculty and staff of MBB, handling 3 different institutes with suboptimal number of staff. But at the same time, you have been doing a great job. As I have noted down here, you have 54 students in NRCPB and 69 in IARI, Ranchi, and 4 in Raipur. So, put together, that is an admirable job. And I am sure that you are doing this ungrudgingly. Of course, it is our national duty to do that. At the same time, we expect more faculty and more teachers. And in this juncture, I also recollect Dr. Jasdeep's suggestion, which I have been mulling over for a long time, since I have come as RAC member of NIPB. It is certainly a discouraging tale or a depressing saga that molecular biology and biotechnology haven't been given due importance in ASRB positions. And I request Anupama ji, and through her, the Director IARI, and also Director NIPB, at some platform, at some juncture, in some context, please mention this, the respective persons in ICAR as well as ASRB. Because our young students who have been doing such enormous work, considerable work of excellent quality, should not get dejected or should not get depressed for lack of opportunity. It's very, very important. Very, very important. And as far as the research in NIPB goes, you have been doing so much work. I fail in my duty if I point certain things and do not point others, because everything has been of equal importance, great importance. But at the same time, I have particular reference to tDNA-free gene-edited mutants, where 3 genes for blast or blight, blast resistance are being worked out. Is something I found very exciting, something novel. And this kind of thing actually should go straight to patent office, not for publication. Once you make this, you go to the patent office first. After an year, you can be a good journal. Then similarly, a lot of work pioneered by Dr. Mahapatra, Dr. N.K. Singh, Dr. T.R. Sharma, and it has now percolated down in the past 5 to 10 years through enormous work on identification, dissection of QTLs, genes, and SNPs, then genomics-assisted breeding, and I find a lot of students working in this area, which is really, really good. And these students who have been doing this kind of work, molecular breeding or whatever, you name it, whatever you like, would have very good opportunities in corporate sector, the agri corporate sector. So it is certainly very useful and very important for the young students. Then another important thing I found worthy of mention is work on nitrate transporters in wheat. Because this is where, as you all know, I need not emphasize again, I have been telling this, I had been telling this to our colleagues. Nitrogen use efficiency in cereals like rice and wheat is of prime importance because of the fertilizer costs and also the energy costs and so on. So when you develop a genotype, rice or wheat, that can take up nitrogen and even other nutrients more efficiently from a soil that is poor in nitrogen. It should be able to take up enough nitrogen from a soil poor in nitrogen. And that's where the nitrogen use efficiency and the respective genes or QTLs or whatever, and even going beyond molecular breeding to gene editing will come into play, and I am happy to see that some projects are already on in that, either through students or research projects of the scientists. Then the aphid effectors, this is something novel I found, aphid effectors. Earlier we were just prospecting for genes that give aphid tolerance or aphid resistance or whatever. But this is a concept which I have found very novel, that is aphid effector genes in mustard. Let us hope this particular kind of approach will solve the most vexing problem of sucking pests, that is aphids, in crops like mustard. And another thing which I found novel, something worthy of mention, is green silver nanoparticles. I did not know this. To be very frank, I only knew about nanoparticles made of some, some metals, but not green silver nanoparticles. If it is really going to help you in ameliorating chickpea wilt, it's a great, it will be a great achievement. Again, if you really succeed in that, you have only one way to go, patent office. No other way. Then I would like to know from Debashish, is he around here? What is this FTO, human FTO? I did not know. What is this human FTO? Because it is expressed in potato. What for? Please excuse me for my ignorance. What is FTO? Yeah, human FTO. What is that?
It is mainly for growth. So they have found the autolog of that gene.
Oh.
This is basically this work was from Nature paper.
Oh.
From where they took their print.
That's interesting. So that is already expressed in potato by the student of Debashish. Huh? Very good, very good. I'm really gratified to know that. Same thing, the cold-induced sweetening is a very, very important trait in potato. Potato after harvest, they need to be stored for a long time. And in northwest India, including Himalayan foothills, it's a very important trait. Cold-induced sweetening is something that should be prevented. And that is something I found very interesting. Student of NIPB working through gene silencing, again worthy of patent once it is ready. Then during RAC, I listened to construction of novel CRISPR-Cas vectors, and the vector also has been validated, I think in tobacco or somewhere. And that is something, I think the director NIPB has also made a promise that it will be given to all sister institutes in ICAR, which is a very good proposal. But at the same time, you take initiative first to patent it. It should be a patent of IARI, since it is done by a student of IARI. So IARI and NIPB joint patent. And it will sell like hotcakes, not only in ICAR, but also in other systems like CSIR, other universities, etc. Genome editing is the latest revolution. It has not yet taken off in India. It is about to take off. But once you have this kind of vectors, which are our own property, our own patents, there will be many, many, many buyers. So it's, I really appreciate that novel CRISPR-Cas vector, very good. Then BT isolates, I have some attachment to BT. So when you find that chitinases in BT also give resistance to fungi, I'm really happy. It's not only resistance to insects, but also to fungi. So the student who has been doing this work should be congratulated. and very good record of publications from NIPB, 48 from students. But in commensurate with the number of students, number of publications, number of projects, number of thesis, dissertations, patent applications are only 2. Please don't misunderstand me. I expect more patent applications. Not only from the faculty or staff, but also from students. So please try to improve this number of patent applications in the next subsequent years. And plant physiology, as I mentioned right at the beginning, has almost totally transformed itself in IRI to molecular breeding and genome editing, which is a very, very, very happy thing. I think this happened not suddenly, but it has been slowly happening in the past 1 decade. And I'm really happy, Renuji, your work also is, I really commend it. Because earlier we were doing classical plant physiology, classical crop physiology, unmindful of what is happening on second floor or what is happening in NRL first floor. Although we are little slow in converting ourselves, you have now caught up with the others. That is MBB and biochemistry. So Vishwanathan also need to be complimented and congratulated for this. Now the whole thing has become an integral whole. So again, publications, record of publications 53, but little poor. Where is Anil? From student side, 14 only. 53 from the staff and 14 from students. So 14 has to be uplifted. Not Anil, Anjali. But 14 has to be taken up. When it is 53 from scientists, it should be 25 from students. So please don't mind if I say these things, okay? This is for everybody's good. Do you agree, Anupama ji? So, with these few comments, I don't think I should stop you from having a nice dinner at home or elsewhere. I once again thank Director IRI, Dean Anupama ji, Monica ji, and Shruti ji for giving me this opportunity, and also thank all of you for patiently listening to me.
May I, sir, kindly be allowed for just a point in continuation to what you said? I really salute you for your, emphasis on protecting the IP part of this, because this is what, during all my deliberations with the professors, with the HODs and with the student fraternity, I always say this, that, please protect, please know the worth of work that you are doing. And for that purpose, sir, dear audience, you are all aware, but again, in continuation, just with reference to what sir has said, the moment you have your idea on paper, the moment you have your POC in the very first semester of your work, means your student's work or your work, immediately go in for filing the provisional patent. Do not wait for completion of the entire verification and validation exercise. File the provisional patent. You have 1 year full window to validate, and then that gives actually guide and edge to actually orient the student to complete the work at that speed, so that you can write the complete specification with the claims. Moment you file the patent, you can write the paper, you can submit the paper in any good journal, leaving aside the only core novel part. Leave aside that, but then the data, that secondary data and the lot of things you have generated around that Venn diagram, go on publishing the paper, giving reference to the IP that you have protected while you are coding. Some of the codes, sir, this is what you know from my lab, we do it, that if we do not have to disclose that, okay, formulation is performing like this, we say F1. And that F1, we say that, okay, this is protected, the composition cannot be disclosed. So similarly, this is it. And that is the winning proposition. So as sir has said, because by the time you will file the complete specification, you know, China or other countries and these countries, they would have taken, you know, the entire clue, because let us not be in silo that we only are, you know, thinking on that or we only are doing on that. Everywhere, humans are basically thinking in the similar manners at some or the other point of time. So this is it, sir. Thank you very much, sir, for sensitizing our basic sciences group. They are doing just wonderful, sir. Thank you so much, sir.
So thank you so much to the chairperson for his very kind remarks. Sir, all the heads, professors, faculty will take care of the inputs given by you. And so now it's time to felicitate the respected chairperson. May I request Dean Madam and all the heads and professors of the 3 disciplines to kindly join on stage along with Dr. Brahmanan and Dr. Bhattacharya, please.
Celebrating together.
So as the co-convener of this session, I would like to thank Dr. Anand Kumar. Sir has been with us since the morning, since the first session. And he has been here for the last 4 sessions, and it is very appreciable. Thank you so much, sir, for being here with us. And thank you for applauding the achievements of the students and the faculty. And I am sure, sir, we will take care of all the suggestions what you have given to us. Sir has been he has been so excited since morning. And sir, it just shows that your love for research. So thank you so much, sir. It is an inspiration to all of us. And I would like to thank the other audience, the audience who is sitting in this hall. I would like to thank the director NIPB, PDWTC, or our competent authority, the faculty, heads, and the dear students whose presentations have been made. Today we started late in the morning, so tomorrow I think we'll begin in time at 9 o'clock sharp. I will request everyone to pass on the message to fellow colleagues, to students, to please be here on time at 9 a.m. sharp so that we start on time and we do not delay the sessions.
Kindly also, try to spare your time to listen something outside the School of Basic Sciences so that you can understand what is being duplicated or what is new, something which you missed out. And where crosstalk can happen. So the whole idea of this session is because of that. And may I take this opportunity to request Dr. Ramcharanji to kindly think of how we can engage Dr. Anand Kumar sir for benefit of the biotechnology as a discipline. You got my point. And we have the BTech biotechnology also. We have the everyone, waiting for his acumen to be actually utilized. So any proposal, I'm going to take the approvals immediately. Yeah. Thank you.
Thank you, everyone.